Good morning, everyone. Good morning in Brazilian time. So it's a great joy to open the first Immune-Leish Simpósio. Welcome on behalf of the Federal University of Espírito Santo, Federal University of Rio de Janeiro, and Oswaldo Cruz Foundation. I would like to thank you, everyone involved in the organization. So everybody was so helpful, and it was not so hard to organize the meeting because everybody was very collaborative. So, and we know that in the pandemic situation, it has been hard. So hope to find everybody well and safe. Of course, we prefer to be together in a presential meeting with coffee breaks and talking. Unfortunately, we are not allowed. So here in Brazil, the situation is still very hard. But in the virtual meeting, we have this possibility to have all together in several different countries at the same time talking about science. So Danielle and I had our graduation together in the same lab. And after we finish and become permanent professors, we usually talk once a week. So in one of these conversations, we decided to organize a meeting of specialists in immunology of leishmaniasis to discuss deeper into some aspects of or new aspects of immunobiology of leishmaniasis. So we hope you enjoy this meeting. Now I give the word for my friend Danielle.
Thank you very much, Herbert. Hello, everybody. Thanks so much for the attendance, the audience for this meeting. As Herbert says, we used to talk, and also we met a long time ago. I'm not going to give you the details, otherwise you always going to confirm our age. But anyway, this meeting supports one thing that we agree, that science is a wonderful place. It's a place where we can meet people, or we can also make friends, make brothers. We can find work partners, as Herbert and also I have with other speakers in this meeting. And also we can also have life partners sometimes. So considering this hard time that we are facing now, science also is a place where we can shelter from adversity and prepare yourself for challenges. So in this, I would like also to say thank you for all speakers and chairs that accepted to attend this meeting, who promptly agreed also and make this meeting big as it today. So thank you so much for this. I would like also to say thank you for the organizing committee, which was formed in large by part of our lab staff, and who manage extra activity brilliantly and contributing to the scientific party, as we're going to celebrate today, starting the celebration today. I would like also to say thank for the partners, which is the Brazilian Society of Protozoology, Brazilian Society of Parasitology, and Brazilian Society of Immunology, for their noble commitment in promoting science and helping to publicize the Immune Symposium. And finally, I would like to say thank for the sponsors, which is the Federal University of Espírito Santo, Federal University of Rio de Janeiro, and Fundação Oswaldo Cruz, and as Herbert said. And also I'd like to say thank you for Beckman Coulter, which supported financially part of this event. Thank you so much for the audience. And also I'd like to invite the part of our partners to give a brief speech. So I'd like to invite Ana Caetano from, is a president of Brazilian Society of Immunology. Please, Ana.
Hello. Well, good morning, everybody. Thank you very much for inviting me. It was very generous from you, because actually I'm not an expert in in leishmaniasis. And I have been introduced in the field by a student of mine who is working with me, which is Tatiane Maioli. And I also want to first to congratulate Danielle and Herbert for the great initiative. I think it's they are young scientists with a lot of talent and energy, and they have been organizing these symposiums and meetings, which they say are very important, especially at this time that we are living now, and to keep us together, to share experiences. So I think it's a great initiative. So I just want to say that congratulations. I think it's a great meeting, and I hope and I'm sure it's going to be a success. So good morning, and thank you for the invitation.
Okay, I mean.
So also we'd like to invite Professor Peralta, from President of Brazilian Society of Parasitology, to say some words.
Okay, good morning, everybody. Thank you very much for the invitation. I think that is a good initiative of the two young fellow that was, I think, Herbert was my student since the undergraduate in the Federal University of Rio de Janeiro. Danielle, I know very well, that is works near my laboratory for a long, long time. And this is a good initiative, and this Brazilian Society of Parasitology is proud to have participation in this event, since that we know that parasitology is one of the most important scientific area of research in Brazil. And the leishmaniasis is very important. And as I belong a long time ago to Immunology Society, we started our Immunology Society working in trypanosoma and the leishmaniasis. That I think that was most of the research in Brazil start probably in immunology. It start a lot in leishmaniasis and the trypanosoma. Then the symposium is very important, and I believe that's going to be a great and a nice event. And I would like to congratulate both of you, Danielle and Herbert, and all the collaborators that accept to participate in this event. I'll be the Brazilian Society of Parasitologists is open for everybody that work in this field, that can help the society and that you can move to a strong society in Brazil. Thank you very much for the invitation, and have a nice meeting for all of you. Thank you.
Thank you very much, Peralta. Also, I'd like to invite Luis Carlos Croco, is a president from Brazilian Society of Protozoology, to give some words.
Hi, Danielle, Herbert, thank you very much. I'm not the president yet. I'm president-elect. I'll be in charge next April. But anyway, it's FPPZ is very proud to sponsor this meeting. It's a great initiative, and it's good to see all these faces, friendly faces, and I'm pretty sure that this initiative is going to be very successful. You guys are you work too hard, and you deserve a very good meeting. I would like to take the opportunity to announce the SPPZ meeting is starting next Monday. So since all you here are interested in leishmaniasis, SPPZ meeting is a very good place to learn a little bit more about the subject. So please join us at this meeting starting next Monday. Anyway, I wish you all a very good meeting, and thank you for the opportunity, and good luck to everyone.
Thank you very much, Luis. Before we moved for the scientific party of the meeting with the opening conference, so I just would like to tell you that I'm delighted to report that the meeting has achieved the great success or more than we expected, to be fair. We had more than 700 registrations from 270 different institutions from all the world, which was like in 16 different countries. So it is the first Immune-Leish Symposium, but so far I can say that is the biggest event ever that we start. So thank you very much for everybody. And I would like also to invite Alda Maria, who is going to be the chair of the opening conferences.
Good morning, everybody. Thank you very much for the invitation for this important meeting. And I also would like to congratulate Abertura and Daniel for this initiative. And I am sure that I will have a very nice time during these 3 days. And it's also very nice to see this face so friendly together here, that we are working in this field for a long time. So it's really a honor to be here. Then we can already move to the presentation. We start right now.
Yes, definitely.
So let's start with Dr. Paul Kay. Dr. Paul is professor of immunology at the University of York, and he's also a Wellcome Trust Senior Investigation and elected fellow of the UK Academy of Medical Sciences. Dr. Paul has a great contribution for understanding different aspects of post-leishmania and vector relationship, not only in the lab, but also in the field. And right now he's working in a vaccine trial in Sudan. So, Dr. Paul, thank you very much for accepting our invitation, and it's a pleasure to hear from you again.
Okay, well, thank you very much. And it's a great pleasure to have accepted the invitation from Herbert and Danielle to attend this first meeting and to give the opening conference. So much of the meeting, I note from the program, is going to be devoted to cutaneous leishmaniasis. So I thought I would choose as the topic of my presentation visceral leishmaniasis and diseases associated with Leishmania-Donovani complex. So let me just find the first slide and share the screen. Okay. Right, hopefully you can all see that, okay?
Yes, it's okay.
Good, right. Okay, so just before we start, one thing I will want to say is that much of the program is devoted to, I think, really in-depth and exciting immunological characterization of the host response to Leishmania. And we're going to hear lots and lots about many cells, many molecules over the next 2 days or so. So I thought I would take a slightly higher level view of the issues associated with the immunology of leishmaniasis caused by the Leishmania-Donovani complex, and give you really some thoughts about what I think is interesting, what we've been working on over the last few years, and some of the challenges that we have going forward. So let me just move on. So I'm going to start with just a few preliminaries, and these are really nods to the fact that although immunologists think, of course, that we can solve all of the world's problems related to infectious disease, there are other considerations that we need to take into account. And I just want to highlight a few of those. I'll then go on to spend a little bit more time specifically talking about visceral leishmaniasis and addressing some of the issues that we've been able to learn from transcriptomic analysis, and also, as one might predict, inviting me to give a talk a little bit about organ-specific immunity in visceral leishmaniasis. And then I'll move on to two other aspects of L-Donovani complex disease. One is the unusual form of cutaneous leishmaniasis seen in Sri Lanka, and then to post-Kala-Azar dermal leishmaniasis. And hopefully over the next 35, 40 minutes or so, you'll get a flavor for some of the sorts of questions that are still really tough to address in this disease, and perhaps make it slightly distinct from the more focused immunology that takes place with cutaneous leishmaniasis lesions. So let us start with just 3 slides, really, just as a nod to other factors we need to take into consideration. And the first really is hats off to my molecular parasitology colleagues and my molecular genetics colleagues, who really recognize that Leishmania is a complex parasite. And I think as immunologists, we've perhaps in the past tended to treat Leishmania as a single entity. It's very clear that we have multiple groups of Leishmania donovani parasites. We have a single group of Leishmania infantum. They cause different diseases in different parts of the world with different aspects of clinical presentation. But there is also a number of complexities around just the basic biology of these parasites related to how their genetics operates. Copy number variation, aneuploidy, the intriguing problem now being felt through in terms of interspecies hybrids, and of course the fact that many of these parasites carry endogenous symbionts. And I think it's really beholden on us to think as immunologists about the influence of parasite diversity on immune responses that we measure. And this is really very much uncharted territory, perhaps with the exception of the really elegant work that Nick Fazell and others have been doing, looking at the role of endogenous symbionts. So that's my first slide. The second slide is really to think about genetics. The era of genetics in leishmaniasis has somewhat closed down, I think, over the recent years. We had a heyday back in the 1980s, 1970s, looking at natural resistance genes, the NRAMP gene, first identified by David Bradley and colleagues, a very important innate resistance gene in the mouse. And we also had the work of Jenny Blackwell and others looking at MHC restriction as a determinant of the outcome of leishmaniasis, and particularly identifying MHC class 2 loci in both mouse and human that are associated with different aspects of cure. But I think we've probably ignored over the last few years how we could use host genetics to really hone in on the control of specific traits associated with different aspects of Leishmania donovani disease. And I think that would be an area I'd like to see pursued more in the future, perhaps. And then the third thing that, again, particularly as modelers, many of us working in mouse models, we love to have our mice specific pathogen-free, beautifully contained within wonderful microenvironmental containment. And yet we know from the field that protein allergy malnutrition, micronutrient deficiencies are all major risk factors for the development of Donovani-associated diseases. And yet, besides a few fairly cursory studies on how lymph node barrier function is altered, how T-cell cytokines are altered, and a little bit about immune metabolism, we know very, very little about how many of these markers that we associate with protection against leishmaniasis really operate in the real world under situations of protein energy malnutrition. And I think this is probably also true for the impact of co-infections. I think these are areas, again, that we need to really consider as we look both at our clinical cohorts, but also as we think about the value of our animal models of disease. So that's just a brief preamble, almost to get some things off my chest, if you like. Let me just go on now to talk a little bit more about visceral leishmaniasis, and just split this into two sections. Firstly, talking about transcriptomic studies that have been performed over the last few years, and then we'll go on to organ-specific immunity. Just to remind those who are less familiar with visceral leishmaniasis of the cardinal features of this disease. First of all, we have good models of visceral leishmaniasis, ranging from different mouse strains through the hamster model, through canine VL, rhesus macaques, and all the way through to humans. And there are clearly differences between the immune responses that we see in these different settings, but I think there are a few common features that one can bring out. In general, CD4 T-cell responses seem more dominant, particularly in primary resistance to infection, than CD8 T-cell responses. And CD8 T-cells perhaps come into play more prominently during secondary re-challenge infections, particularly in animal models. We don't have the strict dichotomy of Th1, Th2 responses that have dominated the mouse literature of cutaneous leishmaniasis for many years. In fact, we have a much broader mixed phenotype with contributions to resistance of both type 1 and type 2 cytokines. But there's clearly a dominant role for the balance between interferon gamma, on the one hand being host protective, and interleukin 10 being immunoregulatory or host detrimental. When one starts to look at chronic stages of infection in really all of these settings, it's associated with T-cell exhaustion, and that covers both CD4 and CD8 T-cells, and some degree of non-specific immunosuppression, possibly reflecting the anti-inflammatory environments created by IL-10. But these are also systemic diseases, unlike cutaneous leishmaniasis, and the systemic disease really impacts on tissue structure, tissue morphology, and a whole range of physiological processes, particularly those related to the coagulation cascade and to anemia, which are really not within the remit usually of standard immunology textbooks. So we have to be very cognizant of those processes taking place. At a histopathological stage, we have, oops, I'm just going a little bit crazy, yeah. At a histopathologic stage, one of the common features is granulomatous inflammation. This is mononuclear cell enriched foci surrounding infected macrophages, which are associated by and large with protective immunity. And it's fair to say that under many situations, chemotherapy is believed to restore some degree at least of immune competence. And we'll talk a little bit more about that later on. So I just want to switch and pick up on a few points that come out from that series of facts. And the first is just to think about the central role of IL-10 in regulating immunity to VL. So IL-10 being much more dominant than other cytokines often associated with susceptibility, such as IL-4. And if you look at this data, this transcriptomic map here, this is a study from the NIH-funded TMRC group out in Bihar, just showing what the effects of blocking IL-10 in an in vitro restimulation assay are on peripheral blood mononuclear cells. And you can see what's upregulated is a whole host of genes associated really with interferon gamma, TNF, NF-kappa B, IL-1 and IL-6 as pro-inflammatory factors driving that cascade. So really IL-10 providing this overall dampening of the pro-inflammatory response during active disease. And of course, one of the difficulties with IL-10 is it's not a simple cytokine to work with. And the second cartoon over on the right here just illustrates a study from Chris Nguerda's lab looking at IL-10 production in IL-10 GFP reporter mice in two different target tissues, the liver and the spleen. And I won't go into detail here or even define these subsets, but what you can see very clearly is that IL-10 is produced in multiple different cells and varies in its production depending on target tissue. Quite interestingly, FOXP3 positive CD4 T cells in both the clinic as well as in many of the models don't seem to be associated with the major production of IL-10, even though they may have some immunoregulatory role. And I think as we understand more about IL-10 and its regulation, including its regulation by microRNAs, long-coding RNAs, for example, it becomes clear that IL-10 still represents an untested immunotherapeutic target in visceral leishmaniasis. Just want to borrow a little bit of data from a review from Christine Pedersen on canine leishmaniosis. Canine leishmaniosis, a great model system for understanding human disease, but also very important in its own right, and particularly so in Brazil. And as with many models, it serves to exemplify and exaggerate certain aspects of immunoregulation. And one that's very much exaggerated in the case of canine leishmaniosis is the role of B cells, and in particular, the role of B cells as a source of IL-10 and also as a source of ligand for checkpoint pathway inhibition of T cell responses, in particular the overexpression of PD-L1 on B cells during chronic disease. And I think that really begs the question, actually, have we really given B cells as much attention as they deserve, both in human disease and also in some of the mouse models? So just thinking about transcriptomic analysis and what we can use that for, and one of the early things that we started to try and use transcriptomics for was to understand a little bit about how concordant the results we were seeing from different models of visceral leishmaniasis might be. And this is just one example, and whenever you do a study like this, it's never perfect because technologies vary across different experimental platforms. Conditions of infection may vary across different experimental platforms, but I think some general messages might come out. Firstly, when you look at transcriptomic responses in any of these models, they're really dominated by interferon-regulated genes, and you can do standard bioinformatic analysis to map downstream transcription factors from interferon gamma, which are related to really very large proportions of the differentially expressed genes that you see during infection. And if you try and compare data sets, this is a mouse data set, and this is one of the hamster data sets from Peter Melby's group, you can see that actually there is a large concordance of genes really related to immune and cell cycle regulation, and also related to vascular remodeling. The vascular remodeling, not surprising, being associated with splenomegaly in visceral leishmaniasis. But you can also see that there are many genes which are segregated and differentially expressed either in hamster or in mouse. This is slight an over-exaggeration in the mouse here because of the technology used compared to hamster. It's interesting to pull out a few of those and just think about how one would use these models to study different aspects of immune regulation. In the mouse, we seem to have a much more dependence on cyclooxygenase pathways for inhibition, whereas in the hamster, indolamine IDO1 is actually a much more prominent immunoregulatory target. And really, you can use this data also to address an old question of why do hamsters get sicker than mice? And at least part of that explanation might be related to this increased abundance of M2-associated genes that you see upregulated in the hamster relative to the mouse. So there clearly is a type 2 bias within the hamster relative to the mouse, but whether that is fully responsible for the differences in clinical outcome remains to be determined. We can try and do the experiment the other way around, and this is a recent study that's just been submitted, really just in revision, from Ana Paula Lima and Jeremy Mottram's group. And what was compared here was a dual-seq analysis, now not switching different model organisms as host, but switching different parasite strains of the species. So this is now Leishmania donovani versus Leishmania infantum in the same host under the same infection conditions, or at least as best as one can manage that. And what you can see here then is a massive concordance really in the response across these two, with very, very few host genes differentially expressed in these two different infections. And where you do see those, they are often related not to immune function, but to various metabolic processes in the host. Surprisingly, when we looked at parasite genetics, parasite transcriptomics in these situations, there was really very, very few, just 4 genes differentially expressed between donovani and infantum through the course of that infection. So we can get some interesting comparative studies like this. And I think we should really be thinking about doing this type of comparative analysis much more frequently, with focuses on specific areas of interest in disease. What about using transcriptomics for biomarker identification? And I think this is a great study in one respect, just because it tells us that something we should have known to start with. And so this is an analysis that we did a couple of years back now. Sorry, I said a couple of years back. That's because you rule out the 2 years during COVID lockdown. So it's actually almost 4 years ago we did the study now, where we tried to look for systemic signatures that were common across blood, liver, and spleen that would allow us to have potential biomarkers of systemic disease. And this study was done in the mouse, and we could identify this largely type 1 interferon regulated signature associated with all 3 target organs that we could use differentially to study the blood. And this was also largely conserved in the hamster as well. But as soon as you take that data set and you then try and apply it to human VL data sets from peripheral blood, comparing rodent blood to human blood, you see a massive discordance in response. And so I think what that tells us, perhaps, as I said, really biomarker discovery is probably best left to clinical studies rather than trying to make wild exaggerations from animal models. That's not to say that we can't get some values, and that was a slightly negative outcome for transcriptional profiling, but we can also get some really exciting insights into disease mechanism. And this is, again, a recent study from Chris Nguerda's group, working with the Bihar group, where they compared CD4 positive T-cell transcriptomics with mouse splenic responses. And they could identify common sets of differentially expressed genes. And the top hit here was a molecule called NKG7, which is a molecule involved in granular exocytosis. Very nicely could do comparative studies in the human to show that presentation of clinical disease, you have high levels, and similarly you have high levels in the mouse. And also that when you knock out NKG7, you can dramatically change the outcome of disease. So I think we can learn things by these types of comparative studies, but I suggest that biomarkers are not perhaps the way to go. We can also use this to monitor drug responses, and this is a study that I think was really fueled by the observations many years ago that one of the main risk factors associated with relapse from visceral leishmaniasis is splenomegaly. And we've always been interested in understanding how well the spleen responds following drug treatment and how well immune homeostasis is restored after drug treatment. And what you can see in this PCA biplot here, if you look at the transcriptomic data from naive animals, they all sit on the left-hand side of this image. From infected animals, they all sit on the right-hand side of the image. And when you drug treat and cure these animals, you get a reversal from the left going back, from the right going back to the left, but it's only a partial reversal. And there is a big discordance here between what you see post-clearance of parasites and the full return to homeostasis. And we're now just in the process of doing similar studies in East Africa, looking at various patient cohorts to see whether this is reproducible in humans, to understand really what we mean by immune restoration at time of cessation of chemotherapy and release of the patient back into their home environments. There's been a couple of studies coming out over the last few years on transcriptomic signatures being used as alternate tests of cure. This one I just highlight from the ITM group in Antwerp, picking up this 4-gene signature associated with an outcome in HIV VL patients. And we've also picked up a innate macrophage-dominated signature associated with cure from PKDL. But I think with all of these signatures, we're still in this early discovery stage, and very few of these have actually been taken out into the field to actually test their reproducibility and specificity. And really to do that, we also have to get access to scalable point-of-care diagnostics to be able to do that. So let me move briefly on to organ-specific immunity, a subject we've worked on for many, many years. And just to give you a few of the outstanding issues, I think, here, and some of the approaches we're using going forward. So the first is just to remind everyone that splenomegaly is a classic feature of visceral leishmaniasis. In some parts of the world, you'll also have lymphadenopathy, such as in Sudan, but that's not generic. And you can spend a lot of time mapping the immune responses that take place in different target tissues. And I just show this cartoon here from a review that Marcelo Montes de Oca wrote recently, which is deliberately complicated. And it's really complicated because, in fact, that is the reality of what's happening in the tissues. And when we are doing many of our experiments, we ignore most of what's happening here and focus on our cell of interest or our cytokine of interest. But the reality is this is all taking place simultaneously, side by side, in a very complex microenvironment. The other key feature from this is that not only are immune cells involved, but there are also other cells from completely different lineages, in particular vascular cells and cells that regulate vascular neogenesis. And we've been able to conduct studies to determine the impact of these different non-immune cell populations and processes on disease outcome, probably leading to the most significant conclusion here that if we block neovascularization using a drug called sunitinib, which is a receptor tyrosine kinase inhibitor, we can actually then augment immune-dependent chemotherapy in the context of antimonial treatment with very good dose-sparing activity by using a preconditioning regime to block angiogenesis. And these types of studies, I think, really tell us that we need to understand microenvironmental aspects of human disease much more than we do currently. And people like Washington Luis and others in Brazil are perfectly placed to take us down that route, I think, in the future. We've been very interested in another target organ in leishmaniasis, and that is the bone marrow. It's always encouraging to still be able to show a piece of data from 1948. It's bad enough showing data from my own lab before my students were born, so it's nice to show something from before I was even born. And this just shows the hematological changes that you see in patients in relation to increase in splenomegaly. And so we've been interested in trying to understand how parasite infestation of the bone marrow has essentially impact on normal bone marrow function. And what we've been able to show in a series of studies is that both hematopoietic stem cells are affected and also various niches associated with platelet production, megakaryocytopoiesis, and also anemia. And I won't go through the details of all of this for time, but I think one of the important consequences of these types of studies is to pose the question, if we have long-term systemic infections, such as visceral leishmaniasis in the bone marrow, how do those accumulate over time to impact hematological health, and whether there is a predisposition then for subsequent co-infections or even perhaps malignancies as a consequence of that. What you start to see when you look in any tissues involved in leishmaniasis, visceral leishmaniasis, is that there is massive heterogeneity in terms of the distribution of parasites and parasite foci within those tissues. And we've really analyzed that in two major target tissues. One is the skin and the other is the liver. And in the skin, you see a very patchy distribution, and that patchy distribution has a major impact on transmission out to the sandfly. But it also gives us a way into looking at what are the potential pathways for dissemination of leishmania within different target tissues and also between target tissues. And the approaches we've taken here are, again, to step outside of immunology and move more towards ecology and to borrow models from ecology that allow us to essentially map parasite distributions, turn those into fixed points, build network maps, as you can see here, through a process called spatial point pattern analysis, and then use those simulations to construct hypotheses about how dissemination occurs. And this work from Joe Dahl is elegantly describes the process where initial seeding of infected cells is followed by parasite expansion through reinfection of local macrophages, seeding of those macrophages out into new foci, and eventually coalescing of those foci to form larger patches with heavy parasite infestation. We think those approaches are going to be incredibly instructive in terms of directing future experiments to understand parasite dissemination. But probably the more well understood and more well known form of dissemination and patchiness is within the liver, where seeding of individual Kupffer cells in the liver, as shown in these very original micrographs from Hank Murray's lab, leads to the generation of these focal inflammatory responses, granulomas. These are inherently patchy in their distribution through the liver parenchyma, based on where stochastically parasites are taken up. And very importantly, these granulomas develop in a very asynchronous manner. And you can see that even a month or 2 months out to infection, you can have fully sterile granulomas which have already progressed to this stage and have killed parasites. And you can have other infected Kupffer cells which have yet to even mount an inflammatory response. And we've tried all sorts of approaches experimentally, but also including in silico computation to try and understand that type of heterogeneity. And what this graph on the right-hand side just shows is that different granuloma subtypes can be identified through computational modeling that either clear their parasites very well, as you can see on the left here, or allow repeated waves of parasite recrudescence. And those waves of recrudescence are associated with waves of macrophage activation or deactivation. And so we've started to use various approaches now to dive even deeper into understanding what these granuloma subtypes are. And I'm not going to show you too much data now, but just to show you some of the approaches we're taking. We're essentially taking a multi-omics approach. We're using protein profiling, largely on the nanostring DSP platform. And that's already allowing us, even with fairly low-plexed protein profiling, to differentiate different functional categories of granuloma. We're using lipidomics through mass spectrometry imaging, again, able to distinguish granuloma subtypes based on their lipid profiles. And we're also now using single-cell RNA-seq to map the heterogeneity in hepatic immune responses, map those cell populations back onto the liver microarchitecture and overlay them with where granulomas are or are not. And that really allows us to get a very, very precise and in-depth view of individual granulomas and their cellular composition, really at the level of 3,000 to 4,000 genes or so per sample. And I just put this one picture up on the right-hand side just to really ram home the idea that microenvironmental control is probably going to be important in understanding pathways of immune protection. What you can see in this top left-hand image is RNA for messenger RNA for alpha-tubulin, for Leishmania donovani alpha-tubulin. So we have 250 micron regions of interest here, both with roughly equivalent Leishmania message. Over here, we can see one of those regions of interest, highly abundant for the Kupffer cell marker CLEC4F, simultaneously expressing NOS. Another population, another region of interest here, probably only about 30 microns away, high expression of ARG, very minimal expression of Kupffer cell markers. So we have incredible levels of heterogeneity in these tissues that we really need to be able to understand, I think, more than just taking a global view of what the whole liver is actually doing. So I'm just going to skip through to some studies we've been doing now on Sri Lankan cutaneous leishmaniasis. And just to refresh people's memory on this, this is an unusual setting where a Leishmania donovani sensu stricto is essentially causing cutaneous leishmaniasis. There are also Leishmania donovanis that cause visceral leishmaniasis in Sri Lanka. And if you compare, this is the cutaneous leishmaniasis lesions, you see they range from small nodules, papules, into ulcerative lesions, very reminiscent of old world leishmaniasis. And if you compare these in mouse models, cutaneous versus the visceral strains that one can identify, then this is the work from Greg Matlashewski's lab, really showing that the visceralizing parasites do very well in mice, in mouse viscera. The cutaneous parasites do very poorly in mouse viscera. Conversely, the cutaneous parasite does very well in the mouse skin, in the foot pad, whereas the visceral strain does very poorly in the mouse foot pad. And Greg's group were able to show that that is one of the best examples, perhaps, where differential expression of parasite-related proteins, this is the A2 protein, might relate to these different levels of tropism. So this is a complicated disease. It's a disease which has it's very tough for chemotherapy, usually somewhere between 12 plus weeks of intralesional SSG required for complete cure and resolution. Again, granulomas form a major feature of the histopathology. And so what we were interested in doing, and this is work with our colleagues out Shalindra Ranasenghi and others at the University in Sri Lanka, we were interested in applying targeted transcriptomics followed by spatially resolved protein analysis to try and understand treatment responses in these patients and whether we could get an idea of what might determine immunologically how well they responded to treatment. So this is whole tissue transcriptomics using the nanostring platform, and you can see just 2 weeks after initiation of treatment, there is a change in expression, a downregulation in immunoregulatory molecules such as IDO1, LAG3, PDL1, as well as some T-cell activation markers such as granzyme B, for example. And if you look at these by T-cell function, we chose to focus on 2 or 3 of these, particularly IDO1 and PDL1, as being pathways tractable for host-directed therapy. And if you take those markers and you now start to do protein expression profiling, this is using that DSP platform I mentioned before. This is actually printed the other way around. So this is now changes prior to treatment. You can see again, there's a much higher level of PDL1 as well as granzyme B and IDO1 prior to treatment compared to after treatment. Importantly, we can use single cell approaches using RNA-scope to identify amastigotes in tissues and correlate expression of PDL1 in this instance with the presence or absence of parasites at the single cell level within lesions, and show that amastidin positive or parasite infected cells have a higher expression of PDL1 ligand than amastidin negative CD68 positive cells. And then finally, and this is probably the most important clinically, is that if we now stratify patients into whether or not they have high PDL1 at the beginning of treatment, or whether their ratio of PDL1 before and after treatment changes more significantly, you can see that in fact, patients with high levels of PDL1 actually have a slower rate of cure compared to those patients that have low levels of PDL1. And so what that gives rise to is a model where we suggest that PDL1 expression in particular is leading to a dampening of T-cell responses locally within the lesion. And that inhibits the synergy between SSG and immune responses that we know is required for effective therapy. And that if we come in with therapy, we kill off some of these amastigotes, we relieve that pressure, we downregulate PDL1 amongst other things, and that allows infected cells to then become better cognate targets with CD4 T cells or CD8 T cells. So finally, just in the last minute or so, a few things about PKDL. I'm not going to dwell on PKDL in great detail in this talk. But again, just to remind people of PKDL, a varied clinical presentation with macular lesions, hyperpigmentation associated with that, related largely to a loss of melanocytes, papular lesions, and also nodules that can grow in size quite dramatically. We know that it develops usually after or sometimes concomitantly with VL treatment. And we know various risk factors for the development of PKDL, including the presence of IL-10 in skin prior to VL treatment, and also a negative LST response after VL treatment. Curiously, and we really have no explanation for this at this current stage, there are great differences in presentation, in time to onset, in duration of disease, in treatment response based on geography. Again, this may well relate to differences in parasites or other features that we're unaware of. Some people have described the immune response here as reminiscent of an immune reconstitution syndrome, like you see in HIV patients. There is a loss of immunosuppression associated with visceral leishmaniasis, which allows a rebound of immunity targeting parasites that are in the skin. When we see parasites in the skin and we see the immune features of those lesions, they're very dominantly M2 polarized. There's usually a lot of IL-10 around. There's plenty of exhausted CD8 T cells, and also, importantly, quite a lot of B cells, again, which we know very little about their function. Unusually, at least in India, we find very few CD4 T cells in the lesions, and that's quite distinct from the situation in Sudan, where CD4 T cells are also found dominantly within the lesions. And in terms of what underpins PKDL from an immunological perspective and from other perspectives, I think we just have a big list of questions, really, and these are just still a few of them. We don't know why PKDL is limited largely only to L. donovani infection. It's very rare to have reports of L. infantum giving PKDL. Even within L. donovani regions, such as East Africa, we don't know why there is a restriction, such that it's very rare, again, to see PKDL in Kenya or Uganda or southern Ethiopia, much more common in northern Ethiopia and in Sudan. We don't know what determines this difference between macular versus nodular presentation. And immunologically, perhaps the most fascinating question really is, why is there this disengagement of systemic versus skin immune responses, both in time and in place? And I think we're really challenged here that we have very few animal models of this disease, where we have no animal models of this disease. The disease is very protracted clinically and very difficult to conduct clinical studies, and ethical challenges in countries where PKDL is prevalent usually preclude serial sampling. So we're often doing cross-sectional cohort comparisons, which are quite challenging for various reasons. So that's really my quick survey through all of those three Donovani-associated diseases. But I want to leave you with just two slides to sum up. Oh, I thought I should mention something about vaccines. Probably people were expecting me to say something about vaccines. So I should just mention where we are with vaccines for the development of against Donovani complex disease. So our own study, which is the PKDL therapeutic Chad vectored vaccine, is still ongoing in the Sudan. It's obviously had some hiccups over the last year or two in progression. We've recently published the study for the phase 2A data, which shows the vaccine is safe, immunogenic, and also looked at some of the transcriptomic responses. And we're just, hopefully by the end of this year, we'll have completed the randomized control trial, which will give us an efficacy signal or not. So that's where we are, and that's really the only clinical vaccine trial that's currently ongoing for any form of leishmaniasis that I'm aware of. I just wanted to mention one very important paper that's come out very recently, and that's from Hira Nagasi's group and others looking at the Leishmania centrin knockouts. This is a genetically attenuated line. And I think this paper in Comms Bio is a really important one, because it shows for the first time, really, that you can protect against fatal L. donovani sandfly infection using heterologous vaccines based on Leishmania major. And I think that's a really important stride forward in terms of thinking about prophylactic vaccines for Donovani complex disease. And then finally, we have a human challenge study, which we're just starting recruiting for now, to see if we can use sandfly-initiated L major to understand not only early stages of pathogenesis, but also as a way of testing these particular vaccine candidates in a prophylactic setting. So the two conclusion slides very quickly. Here are some unanswered questions, and maybe we can discuss those in the forum or the round tables. I think we really do need to understand more about parasite genetic variability, how it impacts on immunophenotype, and how immunology impacts on parasite diversity. And colleagues like Dan Jafares in York are really interested in these issues, and I think are likely to make major strides in the next few years. Parasite dissemination, both in Donovani complex, but really across all of the Leishmania diseases, I think is still a critical thing for us to understand at a molecular and cellular level. We need to have better understanding of human tissue pathology and how we can use this to improve our models. And from an immunological perspective, an organ-specific perspective, why really is the skin so permissive for disease reactivation? And of course, we can't do any of that without knowing a little bit more fundamentally about what all of these cytokines and cells do and whom they do it to. But the last slide is slightly more philosophical and problematic, I think, for us. As immunologists, we love to have reasons for doing our immunology. And I think when I started writing grants, every grant said that if we understand the immunology, we'll have a new vaccine. Now we seem to be in a situation where every study we do concludes by saying we have a new target for host-directed therapy. I'm a great believer in host-directed therapy, but so far, with the exception of imiquimod, we've all failed to get these host-directed therapies into the clinic. And I think we have to think beyond just the immunological proof of concept that we have from models and think really hard about how we get proof of concept from humans using experimental medicine approaches, how we define the arguments for host-directed therapy, how we engage with things that matter for pharma and for funders, such as value propositions and health opportunity costs. Why do we need to have host-directed therapy is the first question that I often get asked when I talk to funders about this. And I think collectively we need to put a little bit more effort into thinking about some of these issues. So finally, just a long list of colleagues who've worked with us over the years. I won't dwell on all of them, but just to mention Helen Ashwin, Nidhi Day, Shumit Day, and Sarah Forrester, who've done a lot of the mouse work that I've been talking about today, and also the human immunopathology work. Charles Lacey, who's helped us with all of our clinical trial activity, which has all been done really in collaboration with Ahmed Musa, and Shalindra Ranasinghe and her colleagues in Sri Lanka, really for access to materials related to Sri Lankan CL, and a whole lot of other people that, again, I won't mention in detail. So thank you very much. I'll stop there, I think.
Congratulations, Paul.
Paul, for your overview, just passing by different aspects of leishmaniasis immunology, especially in visceral leishmaniasis. Then, as we have time, maybe we can start with some questions right now. What do you think, Herbert?
Perfect, thank you.
I think that there is no question on the chat yet, but maybe we can start with us.
Sure. Can I start?
Yes.
Paul, thank you so much for your talk. It was fantastic. So your data about the expression on PD-L1 on the treatment, the reduction of expression of PD-L1 on treatment, it's very beautiful. We talked before. So do you have plans to think about the possibility to do treatment or combinations with drugs and anti-PD-L1 or anti-PD-L1 in the clinical trials? Because it would be fantastic, this idea.
I think we have thought about that, but then we thought about anti-CTLA-4 and combined chemotherapy probably 20 years ago and have never progressed that. So the challenge is making that next step forward, and perhaps Phil Scott might discuss this a little bit in relation to some of his work as well. Engagement with pharma to allow these types of studies to go ahead is by no means easy. And that's where I think we have to use not just immunological arguments, but we have to use healthcare arguments, we have to use policy arguments, we have to use things that they understand more, perhaps financial and commercial proposition arguments. That's not to say that anyone's going to make money out of host-directed therapies, but you have to show that the cost of delivering such a therapy like that is not prohibitive in the settings that one wants to use it.
I'm sure that, again, it's not for all diseases, all patients, but some have some problems, immunological.
It is, and yeah, and so it's not like a vaccine issue where with a vaccine you have to actually justify why you would go into multi-million-dollar production. We're probably talking about drug repurposing in most settings here, so it's not the actual value of that, but it's those are the types of figures that I think they understand more to allow release of the products for study.
But just my finish. I think the advantage of anti-PD-L1 or anti-PD-L1 in relation to anti-CTLA-4, I think that's more specific. Now they are called immune regulators because they don't know how, but they can precisely increase the level of interferon gamma that is necessary. So several people are called that. So I think that's interesting because sometimes, of course, we can have more interferon gamma that is necessary, but usually for cancer, when you use this treatment, you produce the amount that is required. So because that they are calling immunoregulators. Maybe it could be a very good possibility of a treatment. Yeah.
I think if you put a whole bunch of people working on checkpoint inhibitors into a room, they can argue themselves as to which molecule is the best pathway, perhaps.
Yes.
I think the other important part of the data, I think, though, is that there is another way to think about this. One is interventions using PD-L1, but the other is if you have a disease such as Sri Lankan CL, where we have 10 weeks of intralesional therapy, it may be that we could use PD-L1 expression perhaps as a marker for those that are going to cure early or not cure early. But it also leads to the possibility that if you've reduced your PD-L1 expression significantly over the first couple of weeks, maybe we don't need to keep drug treating. Maybe the immune response would have kicked back in again sufficiently to allow cure. That's actually a much easier clinical study to design than a true intervention study with anti-PD-L1.
Thank you so much.
Yes, go ahead, Val.
Hi, Paul.
Val.
Thank you for a fabulous talk. Just a quick question regarding this issue with the use of checkpoint inhibitors to treat leishmaniasis. Do you think it would be potentially a problem if you release too much of the immune response? You could control the parasite, but as we see in the cutaneous forms, if you have an exacerbated production of inflammatory cytokines that could lead to the disease, we know that in visceral is quite different because what you have is actually too much of a control of the response with IL-10 and all that. But do you think this could potentially lead to a different type of pathology or a associated, let's say, a secondary pathology?
I think it's quite possible. We always run that risk of turning on things that we don't want. My feeling is that we'll only find the answer to that by actually doing the studies, and I'm not sure the models will give us the answer to that. I think we need well-designed, controlled, safe trials to really address that issue. I think the fact that we would probably use these in a therapeutic setting in a much more restricted way compared to how they're used in cancer immunotherapeutics. I think almost single shot administration in combination with chemotherapy is the approach that one would be looking at. But yeah, jury is out on that. And that was a consideration with anti-IL-10 as well.
I think it would be a dream come true if we could figure out a way of using some kind of intervention that would be targeted to a particular cell type in such a way that you could maintain the response, you know, in other cells, but then interfere with just one population.
Yeah, and sometimes that type of data only emerges from the clinic. I think there are reasonable examples where, you know, antibodies that target multiple cell types, at least by flow cytometry, when you put them in vivo, the pharmacokinetics actually ends up being selective. And that's true with a lot of the small molecule inhibitors as well, that they tend to have cell-specific effects in vivo that you don't demonstrate necessarily in vitro.
Thank you. So we have questions from the chat. First, Patricia Escobar told, thank you for your nice talk. Dr. Albert de Coutu, also very nice presentation, Paul. And we have a question from Lino Neves da Silveira. He asked, can you bring some overview about immunology associated with recurrence of cutaneous leishmaniasis?
Sorry, I didn't quite, associated with what?
With cutaneous leishmaniasis.
I think we're going to hear a lot about cutaneous leishmaniasis over the next few days. I think what's been quite surprising for us, actually, looking at Sri Lankan cutaneous leishmaniasis caused by Donovani, is how hard it has been to find good comparative data for old world leishmaniasis, old world cutaneous leishmaniasis. You know, you guys in Brazil have done a fantastic job on characterizing Braziliensis infections. But actually, there's very, very little similar types of studies that are done with Leishmania major or even Leishmania tropica in the old world. So I think that's again, something that we might be able to use transcriptomic analysis as a broad brush to look at, particularly lesion-based transcriptomics. But at the moment, I couldn't really say whether cutaneous Sri Lankan disease is different from old world cutaneous disease. Probably has a few differences from Braziliensis.
Sure. So, Leda Vieira also, when you see these different phenotypes of cells in the liver, where the macrophages or Kupffer cells, do you think there is quorum sensing going on?
Well, that's an interesting question. Quorum sensing. I think the immune system has, you know, obviously there is fantastic coordination of cell location based on chemokine signals, based on extracellular matrix, based on other factors. I don't think that really falls necessarily into quorum sensing, but certainly there is communication across space and time. If the question was about quorum sensing in the parasites themselves, that's something I've raised a few times, but never had an answer to.
So to continue, there is no inducing neighborhood max to become regulatory or healing. And also, are those different cells in the same granuloma?
It would appear so.
Context of the same question.
Yeah, so even within the same granuloma, you would have cells with those different phenotypes, yes. And we sort of know that from conventional immunohistochemistry to a certain extent, but I think the approaches that we have now allow us to look at you know, really quite in-depth transcriptomic profiling of those very specific regions. I don't think we can quite get to single cell resolution yet within granulomas and still maintain that detail, that level of depth, but I think the technologies for doing that will be certainly available within the next year or so. Both 10x genomics and also nanostring are both bringing out single cell resolution in spatial profiling systems over the next 18 months.
Daniel, do you want to make a question?
Oh, yeah. Thank you, Alda.
Hi, Dan.
Thank you, Paul, for this amazing talk. I just wondering why PDL1 and not PDL2? Did you check the whole of the PDL2 in your stuff?
I think we did, but I might have to defer to Nidhi if she's on the line somewhere to answer that question. I can't remember PDL2. I'll let you know later.
Okay, thank you.
Let's continue. Camilo Oliveira. Skin so permissive for disease reactivation with PKDL. She asks if it's a problem with resident memory T cells or failure immigration of effector cells, or safe havens. So she saw many thoughts and say fantastic talk.
Alda.
I think.
Go on. Sorry.
I was just going to say, I think you would have to think that resident T cells, resident skin T cells, will play a role somewhere, but we haven't. There is no data currently. We're still looking at that question.
So I have a question. So you addressed an important point, that is the organ-specific immune response. Did you address the skin, the thymus? What I mentioned this is just because one thing that is not addressed in deep is the reconstitution of immune response after treatment. So we have a reduction, an important reduction of lymphocytes, and how this compartment repopulated after the treatment. So it could be, it's also an issue that I have many curious. We studied it a little bit, just trying to understand how the input of thymic cells could impact on this compartment, or T cell compartments after treatment. So I would like to see if you have already addressed at this point.
We haven't, because there's been a couple of groups in Brazil doing that sort of work for a number of years, and both in leishmaniasis and Chagas, as you know. I think it's a natural extension, really. You know, we've looked mainly in the bone marrow at hematopoietic dysfunction, but it's a natural extension to think about, you know, a disease that's affecting young children when you have high thymic outputs. If you have thymic involution progressing early on, whether there is restoration of thymic output post chemotherapy, we haven't looked. Great question. Be great to do that in humans, actually.
Herbert, Daniel,
we can move. Thank you so much, Paul.
Thank you so much. But we can still maintain the questions. Maybe we have time at the end of this, of the talk of Elvira, for more discussion. Thank you.
Right. Thank you.
So let's move to Dr. Elvira Saraiva. Elvira is a full professor from the Department of Immunology of the Institute of Microbiology from the Federal University of Rio de Janeiro. He's a researcher from the Brazilian National Research Council, our CNPq, and also the scientist of the FAPERJ, that is our council from Rio de Janeiro state. Elvira has also working for a long time in host pathogen interactions, and she has great contribution to understand the role of neutrophil extracellular traps, the NETs, and she will talk about this in this meeting. Thank you very much, Elvira.
So thank you for the invitation to present our data here, and also for the words of Alda. So let me start sharing. So can you see it?
Yes.
Yes, okay. So it's a big change after Paul talk. So not only because we move for the cutaneous form of the leishmaniasis, but also because I'm going to talk about the innate immunity and the first effects that is happening in the beginning of the infection. So I'm going to talk mainly about the neutrophil-leishmania interaction, and also about the neutrophil extracellular traps that the parasite is inducing in the cells. So we became interested in this subject because in the beginning of the infection, the sandfly injects the parasite in a pool of blood, and neutrophils are the main white blood cells, and they are in huge amounts in this blood. And also there is a recruitment of neutrophils to the infection site in the beginning of the infection, as Nathan Peters and Dave Sachs show very nicely. And so we ask if this encounter of the leishmania and neutrophils could induce the release of the neutrophil extracellular traps that I'm going to call nets for the rest of the talk. So this structure is very interesting because it's a new mechanism that was described in neutrophils, where upon some stimuli, they release the chromatin, as you can see here. In human neutrophils, I'm going to talk most of the time about human neutrophils. So upon interaction with the promastigotes, we see that the neutrophils release the chromatin, labeled here for the DNA and for histone. And also this structure is associated with different proteins from all the granules of the neutrophils, and also some cytoplasmic proteins, as you can see here, elastase that is from the azurophilic granules. And all these are associated, as we can see in this merge picture. So this structure is already described as an important structure with the function to restrict the spread of the parasites, holding these parasites, as you can see here in the ultrastructure of this net, holding 3 different parasites. And also it was able to kill the parasite. So we can see here that promastigotes attached to this net, they are dead, as shown here by this live and dead assay, where the dead parasites appear in red, and the live ones that are not interacting with this structure are labeled in green. So we show that if we incubate the neutrophils with leishmania, and we treat these neutrophils with PMA, that was a standard net inducer molecule. So we decrease a lot the parasite survival, the promastigote survival. We have increased the survival if we decrease or if you inhibit the phagocytosis of these cells, and much more if we, more than phagocytosis, if we incubate this with an anti-histone antibody, neutralizing the histone that is associated with this structure. So showing that histones are really toxic to the parasite, as you can see here. And also, again, by the live and dead assay, where some parasites are still moving. This is a live picture. As you can see, the flagella beating, and some others are completely dead. So trying to understand the mechanism of this induction of nets, we show in this paper by Natalia Rochael that Leishmania is able to induce the two different net release mechanisms already described. So the classical one, that is the more studied, that occurs in more longer time, 60 minutes or more, and is dependent of NADPH oxidase, nitric oxide syntase, elastase, peptidyl arginine deaminase 4, PAD4, but is independent of myeloperoxidase, similar to what's being described for Pseudomonas aeruginosa. But the parasite is also able to induce net release by the mechanism, the early and rapid net release mechanism that occurs in 10 minutes. So in this mechanism, you have elastase dependent, but is independent of NADPH oxidase and PAD4 activity. So the elastase has been shown that it moves from the azurophilic granule to the nuclei of the cell, where it starts to digest the histones and contributes to the decondensation of the chromatin. Also, PAD4 is doing the same, as it's citrullinate arginine residues from the histones, contributing to this decondensation of chromatin. And of course, the ROS production is important for activating this mechanism in the classical mechanism, but not in the early and rapid way to release nets. Then we move to the signaling inside the cells or the other pathways, and this was done by Thiago de Souza Vieira. That's going to talk. I'm going to do a spoil here. Thiago is going to talk this afternoon, and he's going to show the role of nets in the L major model in BAL-C and B6 mice. It's very interesting. So what Thiago shows is that the Leishmania parasites, the promastigotes, are activating actually two different pathways in the neutrophils. One that is dependent of PI3K delta, that is dependent on calcium, participating in the chromatin decondensation. And the other one is the PI3 kinase gamma through the MEK-ERK pathway, dependent on ROS, also contributing to this chromatin decondensation. But then we are interested to see how these structures are, if they appear in the lesions. And so this is cutaneous lesions from Leishmania brasiliensis lesions, leishmaniasis. And this work was done with Fátima Conceição from Fiocruz and her group. And what they see is that in some patients, you have these huge nets that are showing here labeled by anti-elastase antibody. And some other lesions, you have very small of these structures. And some others, you see the parasites here labeled in red in the middle of these nets. So this is interesting because in the beginning, before the description of this mechanism, people are just looking at this and thinking that it was just an artifact. So it's a very common feature, at least in cutaneous lesions. And looking in these lesions more deeply, we can see some nests of mastigotes labeled here in green, in a net labeled with anti-elastase here in A and anti-histones here in B. So you see that the parasite is in close contact with these structures. So we decide to study the signaling pathway that regulates the the mastigote net release using Leishmania infantum axenic mastigotes as a model. And as you can see here, if we incubate the neutrophils, and I'm speaking also about human neutrophils, with the axenic mastigotes, you see an increase in the release of net as we increase the parasite neutrophil ratio. And here is just some nets induced by the parasite, labeled by anti-elastase. And looking the signaling is more or less very similar to what happened with promastigotes. So it depends on calcium, peptidyl PAD4, peptidyl arginine deaminase 4, elastase, because when we incubate the neutrophils with the elastase inhibitor, chloramidine A and BAPTA, we decrease the net release. It depends also from ROS production, because aprotinine is decreasing this release of net. But contrary to the promastigotes, axenic mastigote seems to be resistant to the net-mediated killing. So here we showed that mastigotes incubated with nets in different concentrations, they are still alive. They are not labeling by ethylium bromide, measured by flow cytometry, as well as from a metabolic point of view, they are completely viable. So we don't have the same effect as we described in promastigotes. Although Leishmania donovani was described by De Couteau, that is resistant to the net-mediated, promastigotes from Donovani is resistant to the net-mediated killing. Oops. So then we start to try to approach a more physiological meaning of study this mechanism. And as you know, the sandfly inject the promastigotes in the pool of blood, as I told you, but this blood was in contact with the extracellular matrix. So in order to understand the role of proteins from the extracellular matrix, we start to look and study the effect of laminin in the modulation of the neutrophil extracellular traps induced by the parasite. So this is the work of Gustavo Silva Oliveira. And so we incubate the neutrophils with the parasite in a very low ratio. And we can observe that these three isoforms of laminin that we tested, 111, 411, and 511, all of them increased the release of nets induced by the parasite. But what was surprising us is that the laminin itself is able to induce net release in all these settings. So just the neutrophil with the laminin, we can have some net production induced by these molecules. So we test some other isoforms like 211, 332, 421, and all of them, they induce net release when incubated in solution with the human neutrophils. And here, just to show you the nets induced by laminin 111, and you see the elastase labeled in green and the laminin stained in red, and the three forms that the three isoforms of the laminin that you tested, and you have the colocalization of these molecules to the net structure. So we were interested in seeing how this mechanism is occurring. And again, it depends on elastase migration to the nuclei and the elastase activity, because when we inhibit elastase in the neutrophils, we decrease the net induction by the three isoforms that we are studying. And also depends on the PAD4 because chloramidine inhibit all the net induction by these 3 isoforms that we tested. But interesting, the ROS production of this, the ROS role in this mechanism is inhibit. If you inhibit ROS production, we decrease the net induction by the isoform 111 and 511, as you can see here, but not the net induction by the isoform 411. And this is just, of course, we don't know exactly the why we have this, but just to remind you that the laminin 411 is a different kind of the ones that we are testing, the 111 and 511. So it's the T shape and not the cross shape. So perhaps it's not interacting with some receptors that needs to be activated for ROS production. So then we decide to see if the laminins are inducing net release by the integrin VLA6, that is the one that is recognized most of the laminins. And we can observe that using an antibody that blocks the alpha chain, the alpha 6 chain of this integrin, we decrease net release. So showing that they are inducing the release of these structures by being recognized by this receptor. But all these studies were done in using incubating neutrophils with the laminin in solution, or even with laminin in adherent to the plastic plate where we are doing the interaction. So we decided to do something more close to the extracellular matrix, the natural extracellular matrix. So we polymerized the laminins in acid pH, according to a methodology by Coelho-Sampaio's group. And as you can see here, we have different types of polymerized laminin. The 111, you have a thicker layer and full of holes, space, where you can, the nets are released when you put the neutrophils here, but they go inside the rows, they go down. As you can see here in the blue, this is the neutrophils that are more in the bottom of this structure. But in the 411 and 511 is this layer, this polymerized laminin layer is more thin. So we can see the nets are in the top of the layer, here labeled in green. So showing that even the polymerized laminin, it's also able to induce net release, and we are measuring in the supernatant of these cultures, as you can see here on the right, the net induction by this form of polymerized laminin. So then we test another matrix protein that is fibronectin, and interesting, just the tissue fibronectin, and when this fibronectin was adhered to the plastic plate, it induced nicely net release from neutrophils. But if we incubate the same fibronectin with neutrophils in suspension, and the fibronectin is not adherent to the plastic, we don't see any net release. And this net release was inhibited by 2 RGD peptides. So it's inducing net release from the neutrophils by its RGD domain. And we could not find net release by the plasma fibronectin. It's something very that we are expecting actually. Not when we incubate with the the parasite, the laminin, the fibronectin itself, or the fibronectin with the leishmania. So then we start to think about how this structure, as you look, they could be very big. How this structure can influence the cells in the neighborhood, how they could modulate cells in the neighborhood. And this is the work of Anderson Guimarães Costa, where we look the effect of neutrophil extracellular traps in the monocyte differentiation of to dendritic cells that we do in vitro, incubating human monocytes with IL-4 and GM-CSF. So what Anderson showed, it was that when you add nets to the medium where you are differentiating monocytes with IL-4 and GM-CSF, you increase the labeling of CD14. On the contrary, that is lost because the monocyte is differentiation in this medium to the dendritic cells. And we also increase the CD1A positive cells. That is a marker of dendritic cells. Sorry, we increase the CD1A here when you don't put a net, that is a marker of dendritic cells. And this was observed also when was decreased when we add nets to this medium. And this effect was not observed when we digest nets with DNAs. So we break the structure of the nets when we filter these nets or the supernatant of leishmania alone. And here is just for you to show that nets interact very close to the neutrophil. So we see a net here labeled in pink by the anti-elastase antibody and the cells nuclei in blue. So what Anderson also described is that when you treat the monocytes with nets, even in the presence of IL-4 and GM-CSF, actually the alpha chain of the IL-4 receptor was decreased on the contrary of the receptor for GM-CSF. And I'm not showing all the results, but this was done by the elastase present in the nets, because when you inhibit the elastase activity in the net, you don't see this loss in the IL-4 receptor. And the other important result of this paper is that the cells that differentiate in the presence of nets, they don't have any effect in the parasite entry, as you can see here, because monocytes are interacting with the parasite labeled with CSF equally in the presence of net or not. But if you infect the cells, the parasites survive much better in the cells that are differentiating in the presence of net structure. And here you can see that in all the monocytes from different donors that we test, we have increase in parasite survival. So the monocytes that are arriving to the infection sites, and we have nets in this site, they differentiate more to a M2 phenotype. So we have markers for M2, and also the cells, I didn't show, but we look, they produce more IL-10 and more TGF-beta, which allowed the parasite survival in these cells, on the contrary of normal dendritic cells that are more prone to produce TNF-alpha and IL-12 that are activating cells and contributing to the parasite killing. So till this moment, different proteins were described associated with the nets. as I told you, some from the granules of the cells, some others from the cytoplasm, not all of them, some. And so we wonder if we could have also some RNA associated with this structure. And this is the work of Leandra Linhares Lacerda and Jairo Ramos Temeroso, that we stimulate NET with 4 different stimuli: PMA, that is our control, transthyretin amyloid protein, and Leishmania amazonensis promastigotes. And I should mention that these parasites are all the time fixed, so they are not having any activity. They are used just for induction of the NET structure. And IL-8, that is another already described cytokine able to induce NET release. And as you can see here, we are finding a lot of small RNAs in these preparations, and also some small nucleotides from 19 to 24 nucleotides that is characterized by microRNAs. So then we decided to look one specific microRNA, that is this 142.3p, in the NETs induced by these 4 stimuli. And we choose this microRNA because it has already described that it is expressed in myeloid lineage, and particularly in neutrophils. It is a regulator of cell maturation. So as you can see here, we have this microRNA present in the NETs of all the stimuli. So it's decreased actually in Leishmania amazonensis, but it's present in all the NETs from the 4 stimuli. And as you can see here, it is also attached, it was interacting with the NET structure that we can see here by labeling the DNA with cytox green, and the microRNA with a probe, a fluorescent probe specific for this microRNA. So we can observe that it's linked and it's associated with NET structure induced by Leishmania, as well from the other stimuli. So then we choose 87 microRNAs to study them in the NETs from the 3 stimuli. We didn't analyze the IL-8 NETs, but as you can see from the 87, you have 55 microRNAs that are present in these structures, some that are shared by Leishmania-induced NET and PMA-induced NETs, and some 5 others that are shared by the amyloid protein and PMA. And as you can see here, this shared microRNA is not present in Leishmania, but is present in the amyloid protein in PMA, as well as this other one, that is the 432.5p, is present in the NET induced by Leishmania, but not in the amyloid protein induced NETs. So based on this study, we can say that we have different pattern of microRNA distribution depending on the different types of stimuli. And this is better shown here. As you can see for this microRNAs labeled in red, that you have an increase in the NETs induced by Leishmania amazonensis and not in the other 2 stimuli, as well as some other microRNAs that the NET induced by the parasite are decreased in this microRNA are decreased in the NET induced by the Leishmania parasite. So microRNAs normally are associated with some proteins as Argonaute 2, or some lipid complex like apolipoprotein A1. So we decide to look if these carriers, microRNA carriers, are associated with the nets that we are inducing. And so, as you can see here in Leishmania amazonense, we see the nets labeled with elastase and the Argonaut colocalizing with this structure, as well as the net induced by Leishmania here, again, labeled with an antibody that recognizes DNA and histone H1, and the apolipoprotein A1 also is colocalizing with this structure. And on the last panel, we can see that the Argonaut and apolipoprotein are present in the net structure induced by the parasite, as well as the one induced by PMA that we use as a control. So we show that we have microRNAs in this structure, that they are probably associated with some carriers. So is this structure able to induce the entrance of these microRNAs in a cell, in the macrophage? So we use THP1 differentiated with PMA for macrophages. And then we incubate this macrophage to the nets. And after 2 hours, we analyze by PCR the presence of the mature form of this microRNA, that is 142-3P, and it's increased in the cells that were incubated with nets in comparison with the non-incubated one. And this is not it's not due to a new synthesis of this microRNA, because the precursor of this microRNA was not modulated by this interaction. We also labeled these microRNAs and looked then inside the macrophage by flow cytometry. So we pre-incubate nets with specific microRNA 142-3P inhibitor, that is fluorescence. We pre-incubate the nets with this, then we incubate the nets with the THP1 macrophages, and we look the expression of this fluorescent inhibitor 2 hours after the interaction. Then some cells we wash and put some medium, complete medium, and then we look again the expression of this marker after 18 hours of culture. And as you can see here, we have the presence of this microRNA in the cells after 2 hours. This is not occurring, of course, in the control, in the microRNA scramble control, in the microRNA inhibitor control, just the inhibitor. But if we add the nets with the microRNA inhibitor, we can observe it inside the cells. And we don't see this if the nets are incubated with the scramble microRNA control. And this occurs at 2 hours and also after 18 hours of cell culture. And this is interesting because could this molecule be modulating some events in the macrophage? So because this microRNA is down-modulating PKC alpha, and PKC alpha is related with the induction of different pro-inflammatory cytokines, such as TNF. So we look these two markers, PKC and TNF alpha, if they are modulated in the macrophage that are incubated with the net-associated microRNA. And here you see the frequency of positive cells for PKC alpha, and here is the MFI. So the control has nice activity that is not modulated by just by incubating the cell with the scramble microRNA, or if the inhibitor microRNA. But if we incubate the cells with NETs alone, we decrease this expression, PKC alpha expression, that is more evident here in the MFI analysis. And this is not modulated when we incubate the macrophage with NETs with the microRNA scramble control. But if we incubate the NETs with the microRNA inhibitor, then we revert part of this activity that we can see here better. And interesting, this effect was dependent on actin polymerization, because when you treat the macrophage with cytokalasin D inhibiting endocytosis, NET incubation was not modulating the PKC alpha expression. And this was not also done by DNA, because we had an heterologous DNA, just DNA, and we are not seeing any modulation of PKC alpha expression. So then we look the TNF alpha, because PKC alpha is also modulating the TNF alpha production. And we don't see any modulation by any of the stimuli if the cells are not primed. So we decide to prime the macrophage with LPS, where you can see the TNF alpha production. So we don't see any effect if we inhibit this, if you incubate the cells with the microRNA inhibitor. But if we add NETs to this culture, we increase the TNF alpha production. But this effect was much pronounced if we incubate the cells with NETs that were pre-incubated with the specific microRNA inhibitor. So you have much more TNF alpha being produced here as the picogram per ml, and here is a percentage of control. And again, this effect is mediated by PKC, because if you treated the cells with PKC inhibitor, we decrease the TNF alpha production. NETs with the microRNA inhibitor that have this huge activity. If you add to this incubation medium the PKC inhibitor, we also inhibit TNF alpha production. And of course, NETs alone with the PKC inhibitor, also we have inhibition of this TNF alpha production. So what we are showing is that NETs can modulate macrophage monocytes that are being differentiated to dendritic cells, as I showed in the work by Anderson, and also some microRNAs that are present in this structure is also modulating the macrophage. The resting macrophage, they are decreasing the PKC alpha, and in the LPS activated macrophage, they are decreasing the TNF alpha and the PKC alpha, and also could contribute to the parasite survival in this in the infection site milieu. So this work was being done by different people and collaborators from Federal University of Rio de Janeiro. The work that I present here have participation of all these people. From Instituto Oswaldo Cruz, and also the work from Anderson has a collaboration of Fabiano Oliveira from NIH, and we need to thank the hemotherapy service of Hospital Universitário Clementino Fraga Filho from FRJ, that is giving us the access to the blood, to the human blood, and the financial agencies, CNPq, FAPERJ, and CAPES. And thank you very much for your attention. I hope I could answer questions. So let me, whoops.
Well, thank you very much, Elvira. Very nice job.
Not hearing you, Valda.
It's okay now? Can you hear me?
Now it's okay.
Okay. So I thought, thank you very much and very nice job. We still do not have questions on the chat, but maybe some of us could have questions to Elvira just to start the discussion.
I have questions. First, thank you so much, Elvira. Congratulations for your talk. That was amazing. When I saw your presentations, I was thinking if I have some of mine neutrophils to the experiments, because we usually don't know. So I was thinking that. So, Elvira, what do you think the impact of the NETs on the inflammatory response, for example, in cutaneous leishmaniasis? And so if you have a contribution to control the parasite, at the same time you can contribute to inflammatory process. So maybe there is some balance. I know that you are looking and investigating this possibility. And for example, when you demonstrated the data from Anderson about the NETs impact the monocyte to the dendritic cell differentiation. So NETs is not only innate, but it can affect the adaptive immune response. So what do you think about this inflammatory process impact on the dendritic cells? And what would be the end of this picture? Do you have an idea?
Well, thank you very much for this question. Actually, it's a broad question. What is being described for NETs till now, it depends on the disease. The amount of data on NET effects, in different effects, is increasing a lot since its discovery in 2004. But what it's known till now is that you need to have, as you said, a balance. You cannot have a lot of NETs or no NETs, because if you have no NETs, you have a worse response for mainly different microorganisms and pathogens. And if you have a lot of NETs, you have also some damage, tissue damage and inflammatory response. More or less what Paul was saying, that answering to Val's question, if you have a lot of this structure, then you could have effects of this, all the mediators. There are so many different molecules, some proteases that are destroying the tissue, and it's difficult to know now exactly the right role. But what we say and what we are seeing in different papers, that you cannot have absence of NETs, or if you have a lot, you can have pathology because of the structure too. So,
Alda, I have a question. May I? Elvira, congratulations for the work. Very, very beautiful. I have a question because Val recently she shows that it's possible to get NETs from the lymphocytes, no CD8 T cells in leishmania, and it's a beautiful work that she has done with cutaneous leishmaniasis too. So I am wondering if it's possible to see some influence of these NETs on lymphocytes, for instance, in the lesions that you have the neutrophils and macrophages and a lot of lymphocytes. So independent of antigen presentation, that you can this influence on the production of cytokines by these cells. Do you intend to do something since the NETs seems to carry some kinds of, like the exosomes, no? Carry some proteins that could be important in the modulation of other cells.
Well, NETs is a platform that can carry many different molecules. There are some already described. And it was initially described in neutrophil, is the more abundant net. If you look the nets induced by other, released by other cells, they are not so big and you don't have big nets. But not only neutrophils release this structure. Monocytes, macrophages, lymphocytes, as Val was showing, not only CD8, but CD4 and B cells also release this. It's a very conserved mechanism. It is occurring even in plants, in root plants. And about the work of Val, it's interesting. I'm not working with patients, but if you look the patients here in Rio, that is the one that I showed the lesions with these huge net structures, you have a lot of neutrophils in these lesions. But if you look the lesions from Bahia, from Corte de Pedra, Val was showing that there is no neutrophils in these lesions. So it's something different. I don't know. And the other day we are having a talk from Daniel, and Espírito Santo is in the middle of Bahia and Rio. And so they have some lesions with some neutrophils, not so many as in Rio, but it's a kind of intermediate amount of cells in the lesions. So this is something that we don't know exactly why it's happened, but of course it's possible that the nets liberated by the CD8 neutrophils that are present in the cutaneous lesions in Bahia could have some effect in the of course they have in the lesions. But it's something different with the Brasiliensis infection that I realized just with the work of Val. I didn't know that.
But the idea is the communication, no, between the cells and the nets. So if you have, for instance, the influence of these nets of neutrophils, for instance, on lymphocytes, or CD8 or CD4 cells, sure that you have this kind of microRNAs that could be delivered to the lymphocytes.
To other cells, sure, of course. Actually, neutrophils are cells that are not very studied before, because, well, neutrophils do oxidative bursts, they phagocytose, and they die. Nothing more than that. And now it's there is a whole bunch of papers showing that neutrophils can present antigens. The nets are very close to the B follicle in the lymph nodes, so they could have some interaction with the cells, even in the lymph node where you're having the stimulation of the cells. So it's something that is growing enormously. And of course, the nets can be a platform for transfer different molecules to the neighbor cells.
Thank you.
More questions?
You have write down something on the chat. Maybe you could comment.
Yeah, yes, I think this, what Elvira mentioned, two important things that are very interesting. One is the fact that we do see, we don't see neutrophils in our lesions, and this was backed up by the work of in Moja's lab and then in Phil's lab, showing that there's no transcripts for neutrophils in the lesions from Bahia. But Elvira sees, and there was a paper that came out in Frontiers recently in collaboration with her, showing that the patients from Rio do have lots of neutrophils and make lots of nets. So that would be interesting for Daniel to look in his in-between patients to see what happens there, and if there is a interaction between the neutrophil-derived nets and the LETs derived from the lymphocytes. That would be quite interesting to find out. And the other thing is the concept that the LETs may be a platform for delivering things. I think the study that Elvira showed with the mRNA is fantastic. And we also saw that the LETs from the CD8s seem to be delivering cytotoxic molecules. And it's really interesting because they look like a string. It seems that they were built to deliver something to another cell, but so that it's very interesting that they are quite different morphologically. And the reviewer from the paper suggested that we also looked at the other types of LETs that we see in the leishmania lesions, whether they are coming from CD4 or macrophages, which can make these structures, as Elvira said. And so we started studying that. And it's really cool because when you take purified CD4 cells and you stimulate them, there's a culture of Aedes aegypti here in our house that I have to be killing them all the time. I apologize for that. I prefer to do that than getting bitten by them and risking to get something. So apologies. But the LETs that derive from CD4 cells, they, first of all, they do not look like strings. And when you stimulate the cells and you look at them in a live microscopy, they burst, completely burst, totally different than the string that is released by the CD8 cell. So I'm very interested in understanding a little bit better that structure. That's a very, so I do have to thank Elvira because she was an inspiration for this work, as well as the fact that her former PhD student came to the lab and started that, which was Amanda. So Elvira is very much a part of it.
Thank you.
I think it's interesting also to see all the strains of Albrighensis that it's possible to induce NET or don't induce NET in lymphocytes or in the Espírito Santo. So it will be very interesting to investigate the parasites also.
Yeah, we are stimulating Daniel to look his lesions to see how neutrophils behave in his lesions, the lesions from patients in Espírito Santo. This will be very interesting. And also, I think that this work from Val is amazing because normally people that are looking adaptive immunity in leishmaniasis, of course, you are looking cytokines, you are looking cell activation, and now you have a different mechanism. What is that? I prefer to call DETs because all of them, they have DNA. So DNA extracellular traps instead of putting different names for depending on the cell that is releasing this structure. So what's the meaning? What is the function of this release of DNA that from neutrophils related with the adaptive immune response of the T and B and different T's and B lymphocytes? So it's something very, very interesting. But for
I have a question about that, Elvira, and putting Val in the conversation. So what's the difference in the content of the net? So we are showing some data about content. So maybe are totally different. Of course, DNA we have and some molecules from DNA, but maybe, and my second question is difference in the content of the net when you simulate with PMA or if you simulate with leishmania? So I don't know.
Yeah, there are already some reports showing that you have difference. If they do proteomics in nets induced by different stimuli, you can have some special molecules with depending on the stimuli. Of course, you have this DNA and histone, and I mean, I'm speaking about neutrophils. DNA, histones, and proteins from the granules, but of course you have different proteins depending on the stimuli. In lymphocytes is more difficult because it's not already described, but it's a very new mechanism. We don't know yet. But in relation to neutrophils, you have different proteins depending on the stimuli. So this is already, and we show this also in the microRNA because the different stimuli, they share some microRNAs associated, but there are some others that are specific from the nets stimulated by some of the stimuli. So we have difference.
I have my own question. Promise that to be my last question. Elvira, why do you have a lot of nets from human neutrophils, but from mice, it's a small amount of nets? Is there some difference, right, about this from human and mice?
This was described also by the Arturo Ziklinski, that is the one that described this mechanism. So there is some difference because neutrophils from mouse are different from human. First, we have much less neutrophils in the mice blood. It's around 20%. So it's very hard to work with neutrophils from mice blood. Second, normally we work with bone marrow derived neutrophils or neutrophils that are recruited to the peritoneal cavity by some stimuli. So we are talking about different types of neutrophils because one is from mice and other human. And also we are not comparing the mice neutrophils, the blood mice neutrophils. We are comparing the ones that we can get from the bone marrow that we have mature and immature and all bunch of these cells, and also the ones in the that are recruited by some stimuli. So it's something different. Thiago is going to talk about this in the afternoon because he's going to work just with mice neutrophils. But of course, it's not so big and so abundant net as in human neutrophils. Human neutrophils are the ones that release the bigger nets that compared to the other cells.
So, Elvira, there are many congratulations on the chat for you. Patrícia Escobar, Maria de Fátima Horta, Rosângela Miller, Olivia Bacelar, de novo, Alberto de Couture, Caroline dos Santos. Leda is saying that, Daniel, it's for you, that Espírito Santo is a piece of Minas Gerais that got split off. Minas wants to have a seal.
I can't support this. Otherwise, someone here is gonna kill me in Espírito Santo.
Well, Daniel is from Minas, isn't it?
I'm from Minas.
Yeah,
I'm a Mineiro.
He's living in Espírito Santo. He's from Minas.
I'm from Minas, but I can't support this.
But you should say that the Mineiros are invading Espírito Santo, literally.
Yeah.
Okay, and also Ken Golobi and Bartira Bergman. Congratulations, Elvira, for the very complete work. How do you think neutrophil extravascular coagulation, procoagulant activity contributes to parasite trapping by nets? And I would like to mention that maybe this difference moving to the neutrophils in human lesions, maybe this difference could be related to the purity of the disease. Have you thought about this? Just put those two questions together.
Alda, the last part of your talk, it was not very clear. It was kind of
okay.
Picotado pessoas. Eu não sei como é que é picotado em inglês.
No, have you heard the question from?
Yeah, how the nets contribute to the coagulation.
So my question was, if you have thought that the difference in the infiltrating neutrophils and cutaneous lesions in humans, maybe it could be related to the period of the disease, as the inflammatory infiltrate, it fluctuates along the time. then maybe depending on the period in which the lesion was taken off, you could have more or less neutrophils.
Yeah, when we did this work with Fátima Conceição from Fiocruz, I was wondering to look this kind of difference, but we just got some lesions from chronic patients. So we are looking from chronic patients. And this would be very interesting, but it's difficult to have because patients normally arrive to the medical care when they are already with the lesion. So you can do this in mice, and we are planning to do this, but we didn't at the moment. But in humans, we just look the chronic lesions. So, and the other thing that I would like to a data that I would like to have in this paper with Fátima is that correlates the net because you have this huge structure and some others that are very small, and correlate this with the disease progression and all that. But because they have these lesions already in the nitrogen, so they didn't have the history of all the patients, so we could not have this a correlation. The only thing that we could find in this paper is that when you have more parasites, you have more nets. It's the only thing. But I love to see if you have this since the beginning. I think, yes, we could have, because we have a lot of neutrophils arriving to the inoculation site when the sunfly bites. Also, I would like to see this effect in the whole blood, just to measure net in the whole blood, but we didn't have the methodology sufficient to do that, because we have more neutrophils in the blood than other, besides red blood cells, of course, but we have neutrophils, the major leukocyte population. So I would like to see this, but, and if, about the role of nets in the coagulation, it's already been described. It's very studied. It's being very studied now in the SARS-CoV-2 infection, that you have a lot of nets, and nets are contributing to the thrombosis, and because of they have many molecules able to activate the coagulation cascade, complement activity too. So we have all this together, and it's a mechanism that is very exacerbated in SARS-CoV-2, for instance, and among other diseases, but just because we are having this pandemic, I'm talking about COVID, and is an aggravating effect in this disease, because it has a lot of molecules able to activate the coagulation cascade, and also complement and activation of other cells, and remodeling of the tissue too, because you have a lot of proteases that are able to digest different tissues and all that. So about this effect that we are seeing with the matrix protein, this was something surprise to us, because I was not expecting that these proteins are inducing net release. But actually, it was already described that the laminin 111 is inducing neutrophils Hos activation and some protease release. So it's something that I was thinking that could be when the neutrophil transmigrate in contact with these proteins, it could release this structure to digest the tissue in a very controlled way, because nets are digesting the tissue exactly where they are deposited, so allowing the cells to migrate to the site more easily. So this is what we are thinking, but we need to prove it.
Elvira, can I ask one thing? Just because I was wondering about. do you think that cytokines can drive NET, or because I'm thinking in a pathology context, because you says that the NET can be also delivered by some non-parasitic stimuli. So we can induce by other type of stimuli. I was thinking about some like a inflammatory condition, chronic inflammatory condition, or if neutrophils or maybe CD8 cells are more prone to do NET in this kind of disease.
Well, it's already described that IL-8, TNF-alpha, TNF-gamma, IL-1, and there are some more cytokines that are able to induce NET in neutrophils. I don't know at the moment if they do the same in lymphocytes, but this is already described for neutrophils. Some different cytokines are inducing NET release. IL-8, for instance. IL-8 is a chemotaxic chemokine that is also inducing NET release. So at the same time that it is attracting the neutrophil, is also inducing the release of the structure. Another thing that we don't know is because if you take neutrophils from the blood and you simulate them, it's not all neutrophils that are releasing this structure. Some, around 30% of the neutrophils are releasing NETs, not all of them. So we don't know if it is a different population or if it is a different mature stage. So because neutrophils are not in the beginning not very well studied, we don't have markers to see if there are subpopulations or why some are releasing and some others are not. Why some are releasing through this early mechanism and some others are just dying, releasing this structure. So it's something that's not known yet.
Okay, thank you.
Okay, so I think we don't have more questions on the chat. Leda Vieira mentioned it's true, but the reason was to make the shipping of gold difficult. Regis Gomes, thanks Elvira for showing my cartoons. And Cristiano Santos, also congratulations you, Paul and Elvira. And I think that that is it. I don't know.
Thank you very much, all.
I don't know if I did not mention any question. I'm sorry if I did it, or if I did not mention any question, but I hope that everything was mentioned.
Thank you so much, Alda. Thank you so much, Professor Bouquet and Professor Elvira and Professor Alda for this beautiful start section in the round table. So I have a lot of more questions, but we don't have time. But I want to wish you luck for your research, for your life, all the good, and for Professor Bouquet, very lucky in the vaccine studies, because I think it will be so important for the world, and here in Brazil, it'd be very important. And a very good luck on sites about neutrophils and the other studies, and for the comprehension of the mechanism and the establishment to understand the importance. Thank you so much. So I think it was an amazing, brilliant start of this Immune-Leish. Daniel, I think we decided to have a short break, right, to start Raquel on the time.
I think, Everett, we can have a 10 minutes break and then we can proceed with
I think 6 minutes because we start at
11.
Yeah.
Okay. Thank you very much, Paul. Thank you very much.
Thank you very much.
Thank you very much, Alda.
It was a pleasure to be here.
So we're gonna have a break and then we're gonna start in 5 minutes with the sponsor talk. That's going to be in Portuguese only. So
I think Peralta wants to say something.
No, no,
Okay.
just saying goodbye.
Ah, goodbye.
Thank you, Geraldo. Thank you all.
Okay, I
Alright.
see you. I see
Thank
you
you.
soon, guys.
Okay,
Bye-bye.
bye-bye.
Welcome back. Can you hear me, Herbert? Yeah.
Yes.
Okay.
Sure.
So let's move for the partner conference. I would like to introduce Raquel Nogueira. Raquel Nogueira is from the scientific Beckman culture, and she's going to talk about flow cytometry for us. This talk is going to be in Portuguese, but you can follow the slides if you want. Ok, Raquel? Você pode iniciar sua apresentação se você quiser, tá? Muitíssimo obrigado por apresentar para nós.
Daniel, bom dia, eu que agradeço. Obrigado pelo convite, obrigado pelo espaço. Está sendo um prazer especial para mim falar aqui pela Beckman e falar de imunologia e de leishmaniose, que é todo o meu histórico antes de vir para a Beckman, exatamente nesse cenário. Então, vai ser um prazer poder trazer aí para vocês um pouco do que vem acontecendo na citometria. Eu fiquei pensando muito como é que eu ia abordar o tema aqui, e aí acho que essa sua iniciativa, né, de trazer esse assunto, imunologia e leishmaniose, focado em avanços e desafios nesse tema, e aí eu tracei aqui um preparo da minha apresentação para tentar desafiar vocês, contando um pouco, lembrando, na verdade, um pouco da história, né, da citometria nesse universo imunologia e leishmaniose. Então, gostaria de trazer um pouquinho aí dessas informações e provocar vocês para a gente poder pensar no futuro também, né? Eu estava ouvindo aí a Elvira falar, então acho que tem muita novidade, muita coisa para ser discutida. Deixa eu compartilhar a minha tela aqui. A hora que você estiver vendo, me avisa aí, só me confirma que a tela já foi projetada.
Tem que botar em full screen, né?
Ok. Bom, então, eu queria falar para vocês hoje sobre a citometria de fluxo de alta sensibilidade. A gente vai falar dos painéis multicolor até o sorting de microvesículas. E eu estava ouvindo vocês falarem antes aqui, Elvira falar, Alda, um prazer rever o grupo aí de leishmaniose, a Cláudia, mencionaram a Fátima Conceição também. Então, faz muito sentido, inclusive, começar essa apresentação trazendo um pouco sobre alguns artigos que foram publicados dentro desse universo, né, de leishmaniose e citometria de fluxo, contar um pouquinho como é que essa história se deu e aonde que a gente está hoje, para onde que isso está indo e pode ir, né? Então, acho que um dos trabalhos importantes é a utilização da citometria de fluxo para mostrar, por exemplo, interação de macrófago e leishmania. Esse é um trabalho lá de 92, que já mostrava essa interação por citometria e mostrava isso através de um aumento de granularidade de macrófago, ou seja, um trabalho que utilizava citometria sem utilizar anticorpos, sem a necessidade de usar aí os fluorocromos, só com a característica de aumento de granularidade dos macrófagos, foi mostrado, né, que tinha internalização da leishmania por essas células. E aí ainda na mesma linha, um outro trabalho, inclusive, do grupo da Elvira, mostrando aí o acesso à leishmania na forma metacíclica, também com uma abordagem não fluorimétrica, né, sem a utilização ainda dos fluorocromos. Não que já não tivesse nessa época, mas uma outra abordagem, e que é interessante o uso da citometria nesse cenário, sem necessariamente essa abordagem utilizando anticorpo e fluorocromo, utilizando scatter, né? Especialmente quando tem aí, acesso ao parasito em si e a interação célula-parasito também, né? Então, inclusive, esse trabalho traz um comparativo com microscopia, que é um outro ponto importante. Então, se a gente andar um pouquinho aí nessa história da citometria nesse universo, aí a gente já começa a ver, né, alguns trabalhos, isso do grupo do Golub, já mostrando avaliação de algumas citocinas, identificação de algumas populações celulares, né, que caracterizam a imunopatogenia da leishmaniose, e aí o acesso já, artigos mostrando já os dot plots. A gente tem o grupo da Alda, que trouxe aí também, né, um acesso mais voltado para a resposta celular dentro desse universo da leishmaniose cutânea. Então, fala muito das subpopulações, CD4, CD8, especialmente CD8, com foco, inclusive, já utilizando a citometria não só para fenotipagem, mas para estado funcional dessas células também. Um outro trabalho também que já traça também um desenho experimental voltado para a inflamação, para uma resposta inflamatória. Aí a gente tem que considerar nesse cenário, né, TH1, TH2, depois TH17, identificação de quimiocinas por citometria de fluxo, as células produtoras de citocina, secreção de citocinas também, identificada por citometria de fluxo, e aí a gente vai tendo esse cenário com aumento do número de marcadores que se pode utilizar em um único tubo na citometria. Isso se deve muito ao aumento da capacidade dos citômetros, né, em poder ver mais cores ao mesmo tempo, então isso propicia uma maior riqueza de informações em uma única amostra, e aí, junto com isso, a gente começa já a observar pela própria imagem, né, dos dot plots que são compartilhados nos artigos, a gente começa a ver um avanço em tecnologia também, acesso não só fenotípico e funcional, e aumento do uso, né, do número de cores dentro da citometria de fluxo para poder definir esses perfis imunológicos de subpopulações celulares, né, junto com os estados funcionais. Então, quando a gente fala já a parte, né, de painéis, aí a gente está falando de painéis de 8, 10, 15, até 20 cores. Quando a gente fala em painéis multicolor, a gente precisa considerar que aí a gente não está acessando só as populações majoritárias. Se a gente conta essa história dentro da imunologia, especialmente da resposta celular, seja por linfócito T, da resposta inata, quando a gente fala, eu estava falando aí dos neutrófilos, são populações majoritárias dentro do universo, especialmente de uma amostra biológica, se for de sangue, por exemplo. E aí, quando a gente fala de subpopulações de estado funcional, né, que é o que a gente acessa quando a gente fala em painéis multicolor, a gente tem populações minoritárias em termos de percentual, a gente tem populações que vão expressar pouco antígeno, ou seja, que vão brilhar pouco, e que isso precisa ser levado em consideração quando a gente fala em painéis multicolor, por conta dos desafios para ter uma detecção eficiente de todas as populações que são investigadas. Né? Então, a gente tem um exemplo aqui com essa imagem de uma análise fenotípica e funcional de algumas populações, né, que são majoritárias, mas estados funcionais também, onde a célula tem marcadores que não são constitutivos, ou seja, ela passa a expressar ou perde a expressão de algum marcador, e isso define o estado funcional dela. E para que se chegue nesse tipo de resultado, é importante que você, claro, tenha um conhecimento de como realizar essas análises, mas que tenha qualidade, tanto em termos de anticorpos, como em termos de citômetro e de ferramenta de análise, para poder ter uma análise eficiente e explorar da melhor forma os dados que são oferecidos, né, que são dados multiparamétricos da citometria de fluxo. Então, eu queria trazer, inclusive, o painel que o grupo do Daniel, que cedeu aí gentilmente o espectro de marcadores que ele tem utilizado aí na sua pesquisa. Então, o grupo do Daniel, junto com a Luciana, eles estão trabalhando em determinar os mecanismos citotóxicos mediados pelas células senescentes associados na imunopatologia da leishmaniose cutânea, né? Então, é um painel aí que hoje tem 16 cores, mas o Daniel hoje está trabalhando aí com equipamento que chega a 21 cores, então isso permite uma abordagem bastante completa do ponto de vista de células que têm esse papel na imunopatogenia, suas subpopulações, seus estados funcionais, identificação aí das células senescentes nesse universo, né, de citotoxicidade, aí, com inclusão de granzima B e perforina, por exemplo, e aproveito para destacar aqui já a inclusão de marcadores de sinalização, marcador de viabilidade, que permite ir acrescentando dentro de um painel amplo diversos marcadores para contemplar todo aí esse painel dentro da imunopatologia da leishmaniose cutânea. Bom, então, nesse universo, né, se a gente contando essa história, né, desde os painéis lá de 3, 4 cores, agora já falando em painéis de 21 cores, podendo ter todo esse acesso às informações, a gente pensa também na questão de preparo dessas amostras e preparo do citômetro. Então, eu queria trazer para vocês a informação sobre o Duraclone, que é um tubo seco da Beckman-Coulter, ou seja, ele é uma combinação de anticorpos secos que fica no fundo do tubo, e ele tem painéis prontos para uso. Então, quando a gente pensa nessa citometria multicolor, ao invés da gente pensar em pipetar 21 ou 15, que seja, 10 anticorpos para o tubo de amostra, essa ideia invertida. Então, na verdade, agora se pipeta a amostra para uma combinação de anticorpos monoclonais, que já está pronto, preparado. Esses tubos, eles são armazenados à temperatura ambiente, então tem uma vantagem na maneira de você gerenciar também os seus painéis, e traz também os tubos single color para poder realizar a compensação de cores desses painéis no seu citômetro de fluxo. Então, essa tecnologia, né, que é a tecnologia dura, é dos ensaios de reagentes secos, unificados, né, e individualizados também, porque você tem, para cada tubo, você vai utilizar uma amostra, nesse tubo está concentrada o seu painel. Então, dentro da jornada do citometrista, agora ele adiciona a sua amostra, pode ser sangue total, ou pode ser uma amostra de cultura, ou pode ser uma amostra obtida, por exemplo, de células mononucleares de sangue periférico. Se tiver que lisar, no caso de sangue total, vai passar pela lise, vai lavar e vai levar direto para o citômetro, porque, inclusive, esses tubos já são os tubos de citometria. Bom, então, se eu estou falando de painéis prontos, eu queria trazer para vocês algumas opções para poder pensar, inclusive, nesses desenhos experimentais, que células acessar. Então, o Duraclone, como eles são painéis prontos, a gente traz alguns painéis, por exemplo, de células tronco, tem células mesenquimais, células hematopoéticas, a maioria dos painéis com opções até 10 cores, mas mesmo os painéis que não têm os 10 marcadores, os espaços que têm sem os anticorpos também é possível adicionar na forma líquida. Então, ele traz também uma flexibilidade de vocês desenharem aí os ensaios experimentais e acrescentar alguns marcadores na forma líquida também. Além das células tronco, a gente tem os painéis de imunofenotipagem que a gente chama de funcional, porque tem um painel de ativação, que inclui aí, inclusive, as marcações intracelulares, IL-2, interferon, TNF-alfa, de T-helper, ativação de monócitos e ativação de basófilos também. São os 4 tubos nesse perfil de tubos secos, e para o sistema imune tem uma diversidade, né, uma fenotipagem geral, uma diversidade de tubos aí, incluindo um tubo que a gente chama de básico, que tem CD19, 3, 4 e 8, né, para linfócito T e B. Tem direcionado para os linfócitos B, direcionado para linfócito T e para suas subpopulações, células dendríticas, células Treg, granulócitos, contagem absoluta, e inclusive, que é o mais recente que foi acrescentado, um tubo direcionado para a imunidade inata, trazendo aí alguns marcadores como o CD117, o 335 e o CD294, todos eles unificados e concentrados num tubo só. E é importante dizer aqui, né, pensando em tecnologia, que esses tubos não são liofilizados. Tudo que é liofilizado, você tem que reconstituir. Nesse caso, ele já é pronto para uso mesmo. E eu gosto de fazer uma comparação, se a gente pensar numa linha do tempo de avanço de citometria, né, avanço de tecnologia, o tubo seco ele representa, acho que o mesmo paradigma de que representou lá na final da década de 90, os anticorpos primário e secundário, né? Porque a citometria era feita dessa maneira. Utilizava o anticorpo primário e o secundário com o fluorocromo. E depois vieram os anticorpos já conjugados, prontos, que é o que a gente tem mais acesso hoje. E agora, né, com essa citometria aí multicolor, essa realidade também de você já trabalhar com os painéis prontos, mas tendo também flexibilidade de incluir outros marcadores nesse painel também. Então, eu trouxe alguns exemplos aqui dos resultados obtidos, para a gente pensar também que não é só uma vantagem do ponto de vista do preparo das amostras, mas também em termos de reproducibilidade, né, quando você adquire essas amostras e tem um resultado, claro, amostras bem definidas, marcações bem feitas para identificar aí, nesse caso aí desses dot plots, as populações majoritárias aí, monócito, linfócito TCD3, TCD4, TCD8, as subpopulações desses linfócitos aí, já combinando CD45RA com os marcadores aí das populações efetora e naive, CD28, CD27, CCR7. As células B também, que eu acho que é importante trazer esse exemplo, de amostras e de populações raras, que são mais difíceis de detectar, e aí quando você tem também esses painéis prontos, já com a titulação pronta, você tem uma maior definição e uma maior eficiência na hora que você precisa identificar essas populações raras, que às vezes é exatamente o ponto é uma hipótese de algum estudo. Além desses tubos, a gente tem aí também os painéis para TREG, os painéis para o sistema inato, trazendo aí CD61, CD127 também para essa análise. E além dos tubos secos, né, com essa tecnologia dura, com os anticorpos monoclonais, a gente tem a opção também do Duractive, que é uma combinação também seca, mas do mix de estímulo, para poder fazer os ensaios de estímulo in vitro. Então, a gente tem 3 opções, um deles já vem com PMA, ionomicina e brefeldina, outro com PMA e ionomicina, e uma terceira opção com LPS, brefeldina A, para dar aí também opções para quem trabalha com esse tipo de cultivo celular. Então, da mesma forma, ao invés de botar as células, ter que preparar uma meio de cultura e ter que fazer o cálculo e a concentração desses estímulos, esses estímulos já vêm pronto no fundo do tubo e você acrescenta a sua célula já no meio, leva para a sua estufa e depois, se tiver que fazer um ensaio de citometria, por exemplo, pode inclusive pipetar essa amostra que já está pronta no tubo e transferir para um outro duraclone com um painel pronto, trazendo aí mais um padrão de qualidade para os ensaios, para que esses ensaios possam, inclusive, serem repetidos de maneira igualitária. Bom, então, falando em linha do tempo, em painel multicolor, eu queria trazer também a linha do tempo em termos de equipamento, porque não dá para a gente falar em painel multicolor sem falar desse avanço, né? Então, se a gente contar a história da citometria, eu vou trazer a linha do tempo Coulter, né, que tem tradição em citometria, porque já produz citômetro de fluxo que já era self-sorter lá na década de 70, o avanço, né, as novas máquinas vieram acompanhando os avanços tecnológicos também, né? Isso a gente consegue ver não só pelos painéis multicolor que a gente estava falando, mas até pelas qualidades gráficas que a gente começa a observar, pela possibilidade que os citômetros oferecem cada vez com mais tecnologia, que não é somente número de cores, e acho que essa é uma característica importante quando a gente começa a falar da plataforma Cytoflex, que a Beckman Coulter lançou em 2014, e que é uma tecnologia, uma família de citômetros, na verdade, que vem crescendo, e por conta até do seu sucesso, pela sua característica de trazer alta sensibilidade de detecção, é uma família que vem crescendo. Então, depois do Cytoflex, que a gente chama de BRV, veio o Cytoflex S, o Cytoflex LX, que é o equipamento de 21 cores, né, que eu mencionei que o Daniel está usando, o DXFlex, que é o citômetro para o mercado clínico, né, para quem trabalha com diagnóstico de leucemias e linfomas, que é uma realidade, inclusive, e o Cytoflex Sorter, lançado agora em 2021. Então, o que essa família de citômetros traz em comum e aonde que a gente chega quando a gente fala em alta sensibilidade? A principal característica desse citômetro é que ele não possui mais os tradicionais fotomultiplicadores, né, que todo mundo conhece como a base de detecção da citometria de fluxo. Então, essa família de citômetro, ela não tem PMT, ela só tem fotodiodo em avalanche, que é outro tipo de fotodetector, que é mais sensível, e que a medição dessa sensibilidade é através da MESF. A MESF é uma quantidade mínima solúvel de moléculas de um determinado fluorocromo para que seja detectável. Então, a gente tem uma média aí para FITS menor que 30, para PM menor que 10, né? Ou seja, menos de 10 moléculas para você identificar um sinal. Isso é o que vai mostrar a alta sensibilidade desse tipo de detector. Quando vocês trabalham com citômetro que não tem PMT, você não trabalha com voltagem, então isso tem muita redução no ruído eletrônico, que a gente estava acostumado a ver nos equipamentos que tinham os fotomultiplicadores. Então você vai ter, né, com essa maior sensibilidade, uma maior facilidade de detecção, por essa facilidade de sensibilidade que não é só de fluorescência, mas também das características de tamanho e granularidade, e você vai ter uma melhor resolução dos seus dados, que são multiparamétricos. Então, isso faz muito sentido quando a gente pensa nesse avanço do tamanho dos painéis, porque quando a gente não tem mais só as populações majoritárias, os marcadores constitutivos, a gente vai precisar de mais eficiência para detectar essas marcações pouco expressas, esses marcadores que não são constitutivos. Então, só para contextualizar esse tipo de fotodetector, né, no cenário da citometria, tem um artigo que foi publicado comparando o fotodiodo com o PMT, não é um artigo que está falando especificamente de nenhum citômetro, mas ele compara a eficiência quântica, né, de fluorescência entre um fotomultiplicador e o APD. Né? Então, ele mostra aqui que um PMT, que é o tradicional da citometria, aqui na linha vermelha, ele já tem um decréscimo no seu desempenho conforme a gente anda no comprimento de onda. Então, tem um exemplo aqui, por exemplo, FITS, APC, e o APC sai 7 a 750, já tem uma performance bem diminuída, comparando só dentro do universo do fotomultiplicador. Quando a gente fala da citometria com fotodiodo em avalanche, a performance é bem superior e estável para todos os comprimentos de onda dos fluorocromos disponíveis aí no mercado. Então, como é que isso vai impactar no ensaio de vocês, né? Nesses ensaios multicolor? Quando a gente olha para um citômetro que tem fotomultiplicador, especialmente as fluorescências mais fracas, né, daqueles antígenos pouco expressos, eles não são tão bem definidos e você não consegue diferenciar uma população, às vezes, negativa daquela que passa a expressar ou perde uma marcação. Enquanto que quando você trabalha com um equipamento que tem o fotodiodo em avalanche, você tem uma melhor resolução, uma maior definição das populações, especialmente daquela população rara e daqueles antígenos que são pouco expressos. Bom, além dessa maior sensibilidade para a fluorescência, fundamental para esses painéis multicolor, essa maior sensibilidade dos APDs, ela também serve para o scatter, especialmente para o side scatter. E tem um artigo que foi publicado recentemente, acho que tem 2 anos, na Scientific Reports, que aí sim fala do Cytoflex, né, essa plataforma que trabalha com fotodiodo em avalanche, e que mostra a detecção de nanopartículas, não só de beads de poliestirene sílica, chegando aí a 60 nanômetros, mas também a detecção de vírus. Então, isso muda um pouco também dentro da história da citometria, aonde que se pode chegar, com que nível de detecção e com que tipo de resolução para esse tipo de amostra biológica, inclusive. Então, para nanopartículas em geral, é o equipamento ideal também, por conta dessa autossensibilidade, que vai além da sensibilidade de fluorescência, uma sensibilidade para scatter também. Então, só para a gente exemplificar esse mundo nano, esse artigo ele também traz alguns dados de detecção de exossomas e de vesículas extracelulares, já aí com alguns marcadores, combinando CD9, CD61, CD81, que são alguns dos marcadores desse universo da nanocitometria, e esse artigo traz aí esses resultados da plataforma Cytoflex, que tem aí esse fotodiodo em avalanche. Então, se a gente começa a trazer aqui, né, nessa linha do tempo, esse universo nano, dentro da história da Coulter também, a gente já na década de 90 tinha equipamentos que chegavam a 500 nanômetros, né, que já consegue detectar um espectro das microvesículas, mas já é sabido também que existem as pequenas vesículas e as médias vesículas e que elas têm um papel diferente na resposta imune. Já foi mostrado que quanto menor as microvesículas, mais atividade de comunicação célula-célula elas desempenham. Então, às vezes, quando a gente fala de uma diferença de 80 para 200 nanômetros, de 200 para 500, isso vai ter um impacto dentro do estudo imunológico também. E o Cytoflex, né, que são esses equipamentos, essa família de citômetro com fotodiodo em avalanche, chega a 80 nanômetros, então a gente já está falando em nanocitometria. Para a gente falar em nano, vamos introduzir um pouco esse universo, não sei se todo mundo já pensou em trabalhar com microvesículas, ou se já ouviu falar. Tem uma questão aí de nomenclatura, tem microvesículas, vesículas extracelulares, os exossomos, tem uma diferença de onde eles são produzidos e liberados, né? Alguns direto do citoplasma, com informações da membrana plasmática, outros vêm com informações, inclusive, de RNA, especialmente os exossomas. E essas nanovesículas, né, que são liberadas pelo que a gente chama de uma célula-mãe, que são as células que participam da resposta imune, vamos pensar aí no universo de leishmaniose, elas podem desempenhar diversos papéis. Pode ser na apresentação de antígenos, levando esses antígenos de superfície para outras células, de outros tecidos, inclusive. Pode desempenhar atividade pró-inflamatória, anti-inflamatória. Tem um papel na coagulação, tem um papel em eventos trombóticos, inclusive. E como é que a gente trabalha, então, na citometria, identificando essas vesículas? Utilizando aquela informação básica da citometria, que é buscando, especialmente, antígenos de superfície, né? Sempre buscando um marcador que ela traz da célula-mãe, fatores teciduais e a fosfatidilcerina, que é um dos marcadores que a gente chama de marcador PAN de microvesículas. Muito bem, então vamos trazer então as microvesículas para a citometria de fluxo. A gente pega um forward side aqui, uma imagem tradicional de um sangue periférico, né, onde a gente identifica por forward side linfócito, monócito, granulócito. Aí ali para a esquerda, aqui embaixo, a gente tem os que a gente chama de debris, que a gente não quer saber. E para trás, onde a gente coloca o threshold, né, onde a gente apaga aquela informação, estão algumas das microvesículas. E que a gente, até muito tempo, por muito tempo, a gente ignorou essa informação. Mas esse é só o começo das microvesículas dentro da citometria, né? O universo das microvesículas, vamos dizer, fisicamente, aí está bem para trás por conta do tamanho. E quando a gente tem algo na citometria que está muito colado no eixo, que a gente não consegue ver, o que a gente faz? Aumenta a voltagem. Quando a gente aumenta a voltagem, a gente aumenta o ruído eletrônico. Então, por isso, a gente precisa também pensar, em termos de tecnologia, que a citometria precisa de algo que tenha uma tecnologia que consiga chegar nesse tipo de detecção. Além do fotodiodo em avalanche, uma outra característica importante que se combina para poder detectar partículas tão pequenas, não só vesículas provenientes aí da resposta imune, mas inclusive vesículas do próprio parasito, seja de leishmania, seja de trypanosoma, por exemplo. Então, uma outra característica da citometria que é importante para essa detecção é utilizar, além do side scatter do laser azul, o side scatter do laser violeta, que vai dar um maior índice de refração e vai propiciar um maior sinal, possibilitando, então, detectar partículas que são muito menores e que, portanto, teriam um menor índice de refração. E aí, qual que é a realidade de quem está trabalhando com a resposta imune celular e de quem quer ver o papel dessas microvesículas na resposta imune que seja da leishmaniose? A realidade é mais ou menos essa, né? Você está trabalhando aí, se a gente pensar num linfócito, mais ou menos 10 micrômetros. Quando a gente fala em microvesículas, a realidade de tamanho é mais ou menos essa. Mas tem uma outra coisa que é importante levar em consideração, que é a oferta de antígeno, que, claro, vai ser muito menor, né, para as microvesículas. Então, vai ser algo que vai brilhar muito menos quando você usar o marcador. Esse sinal vai ser um sinal com um brilho muito menor na hora que você for abrir lá o seu dot plot. Então, qual que é a realidade, né, quando você trabalha com célula, quando você trabalha com microvesículas? Com microvesículo, você vai ter algo que é muito menor do que a célula, e você tem muito menos receptor, muito menos oferta de receptor. Então, você vai ter um resultado que vai ter um brilho muito menor na hora de você definir lá negativo e positivo. E aí, na hora de você ajustar o seu citômetro, se você não tiver especialmente a tecnologia adequada, você vai tentar aumentar a tua voltagem, e aí você vai ter um sinal que vai estar cheio de ruído, vai ficar na dúvida se aquilo de fato é o teu sinal de microvesícula, é a tua marcação para entender de onde que vem essa microvesícula, ou é ruído eletrônico. Então, por isso que a gente fala dessa combinação do fotodiodo em avalanche com o side scatter do laser violeta para detectar esse tipo de partícula dentro desse universo nano. Qual que é o impacto científico das microvesículas, né? Se a gente pegar aí o número de artigos publicados só em exossomo, né, que é uma fatia aí do universo nano, nos últimos anos tem um aumento muito acentuado, especialmente nos últimos 10 anos. Se a gente pensar só fazendo um comparativo aqui, que a direita no alto tem um comparativo, mais à direita aqui o tamanho de um linfócito, né, vai variar de 8 a 12 micrômetros. A gente tem os corpos apoptóticos, que já é considerado uma microvesícula, que tem aí mais ou menos 1 micrômetro. A gente tem as microvesículas, né, que vão variar de 100 nanômetros a 1000 nanômetros, e os exossomas, que vão variar de 30 a 100 nanômetros, tá? Mais ou menos do tamanho de alguns vírus. E aí, se a gente também pegar o número de artigos geral em vesículas extracelulares, um aumento super acentuado também dos últimos 10 anos, e do estudo de vesículas extracelulares por citometria de fluxo, exatamente para explorar a técnica como uma técnica que aborda de maneira multiparamétrica. Então, você consegue combinar informações de tamanho e expressão de moléculas. Bom, tem um artigo publicado na Cytometry há 5 anos que fala exatamente de como detectar, como obter essas amostras de microvesículas em plasma, como armazenar essas amostras, como ajustar o citômetro, e traz também informação de alguns marcadores importantes. Recentemente eu fiz uma live junto com a André Teixeira, do René Rachou, né, da Fiocruz de BH, e a André falou bastante sobre as microvesículas como biomarcador do prognóstico e resposta clínica do tratamento, tanto de doenças infecciosas como câncer, E ela mostrou bastante resultado que ela já obteve, inclusive, em diversas doenças, em sepsis, em câncer, mas também em algumas doenças infecciosas, e ela mostrou o papel dessas microvesículas como preditores de desfecho clínico, papel protrombótico, especialmente na sepsis, e falou também da identificação dessas microvesícula de neutrófilo, de plaqueta, e um trabalho que está sendo realizado aí na plataforma Cytoflex, focada em biomarcadores de prognóstico e de resposta a tratamento. Essa live está disponível lá no Facebook da SBI, enfim, traz bastante informação, desde a parte de obtenção de amostra até a escolha de marcadores e como analisar, e as características de citometria também. Bom, então, saindo um pouquinho desse universo nano, vamos para a citometria, para algumas temáticas aqui da citometria que são importantes levar em consideração quando a gente pensa no futuro de citometria, especialmente em painéis cada vez com mais cores, e pensando em tecnologia também, porque não adianta avançar em cor e não avançar em tecnologia. Quando a gente fala em compensação, né, especialmente fala compensação de 8 cores, 10 cores, 15 cores, como é que isso é feito? Isso geralmente gera um pânico, mas que atualmente é um pânico desnecessário, especialmente para esses equipamentos que têm o fotodiodo em avalanche, que é um detector que é totalmente digital. Isso traz algumas vantagens para os ensaios que vocês fazem por citometria. Por quê? Porque qualquer ajuste de ganho, porque nesses citômetros não tem voltagem, qualquer ajuste que seja necessário, às vezes para um marcador que é pouco expresso, você precisa aumentar o seu ganho, a média de intensidade de fluorescência, ela aumenta de forma proporcional nos equipamentos com APD, como a gente vê aqui nesse gráfico aqui de cima. No gráfico aqui à esquerda, na parte de baixo, os citômetros com fotomultiplicador, né, com os antigos fotomultiplicadores, quando você mexe na voltagem, seja aumentar ou diminuir, a média de intensidade de fluorescência não aumenta de forma linear, proporcional. Isso quer dizer que você tem que fazer novamente o teu ensaio de compensação, tem que adquirir novamente os tubos. Quando você trabalha com os fotodiodos em avalanche, com os APDs, essa compensação ela é atualizada automaticamente, exatamente porque ela é proporcional. Então, o software é compatível com esse tipo de tecnologia e a sua matriz de compensação, mesmo que ela seja enorme, com vários marcadores, com 10, 15, 20, ela é atualizada de forma automática, você não precisa adquirir de novo os seus tubos single color para realizar a compensação. Muito bem, então estou falando de Cytoflex, ainda não apresentei quem é essa família de citômetros, então a gente tem hoje aí dentro do universo da pesquisa 4 diferentes membros dessa família, o Cytoflex BRV, que tem 3 lasers, o Cytoflex S, o LX e o Sorter. O Cytoflex a gente chama de BRV, tem 3 lasers, e ele vai de 4 até 13 cores simultâneas. O Cytoflex S, ele sempre vai ter 4 lasers, sendo que o 4º laser é da escolha de vocês, então ele sempre vai ter azul, vermelho e violeta. E um 4º laser que pode ser o yellow green, pode ser o near UV, pode ser o infravermelho. Ele parte de 6 cores mínimas, podendo chegar até 13, conforme vocês vão avançando em upgrade. O Cytoflex LX, que tem 6 lasers, todos eles, né, azul, vermelho, violeta, yellow green, near UV e infravermelho. Vai de 14 cores até 21 cores, e a gente tem agora o Cytoflex Sorter, que tem 4 lasers, azul, vermelho, violeta e yellow green. É um equipamento que vai de 5 a 15 cores e faz 4 vias de separação simultâneas, né? Lembrando que ele é um cell sorter. Muito bem, é importante dizer que para toda a plataforma Cytoflex, o upgrade ele é sempre eletrônico. O que isso quer dizer? Que se vocês começam com o equipamento, tem um exemplo aqui, né? B2, R2, V2, ou seja, o equipamento que faz 2 cores no laser azul, 2 no laser vermelho, nenhuma cor no laser violeta. No caso de fazer um upgrade, o equipamento sempre vem pronto, com todos os lasers, todos os filtros, todos os APDs. Esse upgrade, então, ele é um upgrade eletrônico. Vocês compram a chave que vai liberar, que é um USB, que vai liberar uma configuração mais avançada e vocês vão ter acesso a mais cores simultâneas. Ou seja, mesmo que você comece com uma configuração básica, todos os filtros estão sempre disponíveis. Então, se você tem 2 cores no azul, são 2 cores simultâneas, pode ser FITS IPE, pode ser FITS IPC5, FITS IPC7, cada dia pode ser de um jeito, porque todos os filtros estão sempre liberados. Então, acho que é interessante também, quando a gente pensar em tecnologia, ter também flexibilidade desse ponto de vista tecnológico também. Bom, já que ele traz no nome dele a questão da flexibilidade, um equipamento pequeno, que inclusive é compatível com cabine biológica, as versões de 3 e de 4 lasers, né, que tem essas medidas, equipamento que pesa 23 quilos e que tem um alinhamento fixo, que é uma das preocupações de quem é citometrista, é desalinhamento de laser. Então, essa é uma outra característica e vantagem também. Até poucos dias eu estava com um aqui na minha casa, inclusive, e não precisei nem da engenharia para instalar. Então, ele traz outras vantagens para um laboratório também, até para, se inclusive você quiser mover ele de lugar, já que espaço físico é um problema também hoje em laboratório. E a gente tem o Cytoflex Sorter, né, que faz a separação das amostras também, e é um equipamento, né, como eu já mencionei, que chega a 15 cores com 4 lasers, faz 4 vias de sorte simultâneas. O ajuste de sorte é todo automático, em até 30 minutos o equipamento está ajustado para realizar a separação das populações de interesse. Ele traz a mesma tecnologia já conhecida da Beckman, de fazer sorte misto, isso é uma característica super importante, né, que se você tem uma população multiparamétrica, você pode fazer no mesmo sorte, um sorting de pureza, priorizando a pureza, com o mínimo de contaminação da sua população de interesse, e numa outra via você faz um sorting de enriquecimento para uma população que é rara, você não precisa fazer 2 sortings subsequentes, você faz tudo no mesmo processo. É um equipamento que traz alta pureza e alta estabilidade de processo, para não ter que ficar refazendo ajuste de sorte, e ele é um nanosorter, porque ele traz toda a tecnologia que a gente vem falando aqui, de Cytoflex, né, do APD, do Sidescatter do Violeta, chegando aí a 200 nanômetros de limite de detecção. É um sorter de bancada, então ele, além de ter as medidas aí compatíveis com sorte de bancada, é um equipamento que é leve e ajustável a qualquer laboratório. Para quem nunca trabalhou com sorte, que é inclusive uma das aplicações que tem vários artigos dentro do universo, não só da leishmaniose, mas da imunologia em geral, todo sorter é um citômetro e passa pela parte de análise, né? Então, você tem a sua amostra pré-sorte, que é a sua análise multiparamétrica, e você tem as suas populações de interesse que você deseja, que a gente chama de sortear, que é purificar essa amostra, sempre pensando que o sorting, o processo, para quem trabalha com sorte, sempre está pensando no ensaio posterior. Então, a citometria, ela é uma parte desse caminho, essa amostra obtida, né, agora homogênea, vai para uma aplicação pós-sorte, e aí são várias. Análise funcional, biologia molecular, genética, tem os ensaios de proteômica também, clonagem, que é o single cell sorting, cultivo celular, engenharia de proteína, teste funcional com as microvesículas, não só identificando essas microvesículas, mas utilizando essas microvesículas obtidas por sonde em ensaios funcionais, desafiando algumas células com essas microvesículas. Então, para quem pensa em cell sorting, né, tanto na parte em si de cell sorting como no ensaio posterior, precisa ter algumas coisas em mente do ponto de vista, né, de um desenho de projeto que é pensar em alta sensibilidade para você ter as populações de interesse, assim como alta especificidade, percentual de viabilidade pós-sorte, especialmente se você for fazer um ensaio funcional depois, um cultivo celular, para você não perder a viabilidade dessas células e isso comprometer o teu ensaio pós-sorte. Percentual de pureza, para você não ter contaminação e ter um ensaio fidedigno pós-sorte. E também um alto percentual de recuperação, que é a quantidade de célula que você recupera. Não adianta você querer sortear 1 milhão de células e não recuperar nem metade, por exemplo. Então, o número de célula recuperada também vai ser importante para o teu ensaio pós-sorte. Então, algumas características importantes para quem já faz cell sorting ou para quem pensa em fazer. Outras questões que precisam ser pensadas quando a gente desenha um ensaio experimental de sorting, né? O sorting no Cytoflex Sorter pode ser feito, né, em 4 vias simultâneas, pode ser em 2 tubos de 5 ml, 2 de 15, ou 4 tubos de 15, pode ser feito em placa, ou seja, você já pode preparar a sua placa com seu meio de cultura, seja a placa de 6 postos, 24,96, com um soro, por exemplo, se você for levar para um cultivo, e aí a célula já cai num ambiente propício para ela poder se desenvolver e você ter o seu ensaio pós-sorte. E o modo de separação misto, né, para poder encaixar aí dentro do seu ensaio multiparamétrico diversas funcionalidades simultâneas. Além do sorting misto, a gente tem também o single cell sorting, que a gente chama de clonagem, reforçando aqui que, inclusive, o single cell sorting ele é bastante utilizado para sorting de parasito. Né? Você coloca uma célula, um parasito por poço, e você tem um ensaio aí de clonagem. Então, você tem dentro do software a possibilidade de dizer quantas células você quer colocar por poço. No caso do single cell, você vai botar uma, mas você pode, inclusive, selecionar para sua placa 500 células, 1.000 células, 1 milhão de células dentro da mesma placa, inclusive fazer o teu controle positivo dentro do ensaio, tá? Quando a gente fala de Cytoflex Sorter, e é um equipamento, né, que é automatizado, então todo aquele ajuste de sorting ele é feito em até 30 minutos, o equipamento está pronto para você fazer a tua análise, teu sorting, obter a sua população de interesse, então isso traz uma outra realidade para quem trabalha com cell sort. A eficiência de sorte, que é super importante para quem está pensando, né, em obter, por exemplo, uma célula produtora de uma determinada citocina, ou uma célula Treg, por exemplo, que é uma célula rara, pensar, né, na pureza que a gente pode atingir quando a gente fala em cell sort. Então, a pureza desse equipamento chega a 99%, e a viabilidade chega a 94%, a recuperação chega a quase 92%. Então, isso mostra a alta eficiência, que é um padrão importante para pensar nesses ensaios de cell sorting, no ensaio pós-sorting também. E, além disso, a gente traz a opção também, sempre isso tudo vinculado com o tipo de resultado que vocês buscam dentro da hipótese de vocês. Então, a possibilidade também de agregar o cell sorter dentro de uma cabine biológica e acoplar também um banho-maria está de acordo não só com a proteção do usuário, no caso da cabine, mas como com a realização dos sortings assépticos, né? Para quem vai fazer, por exemplo, cultivo celular, e o banho-maria que vai controlar a temperatura tanto da sua amostra pré-sorte como da amostra pós-sorte, para que você também não tenha um impacto funcional das suas populações de interesse. Muito bem, então, chegando aqui ao final dessa apresentação, nesse universo aí, eu tentei trazer um pouco do que a gente já tem hoje, do que já é realidade, de como que a gente pensa a citometria de fluxo de agora e do futuro, dentro desse universo, né, da imunologia e da leishmaniose. Então, alta sensibilidade para a fluorescência, já que a gente está falando, né, de painéis multicolor, né, a gente já fala de uma realidade de pelo menos mais de 10 cores. Então, pensar em identificação de populações raras e antígenos pouco expressos, que é uma realidade de painéis multicolor. Pensar em equipamentos que têm possibilidade de mais cores, né, que passam aí das 10 cores, chegam aí pelo menos 20 cores. E com essa vantagem da possibilidade de um upgrade eletrônico, em que você já tem um equipamento totalmente pronto e ao longo dos anos pode ir avançando nessas configurações. Os tubos secos, com os painéis prontos, que vai trazer a reprodutibilidade e agilidade para a obtenção desses resultados multicolor, com padrão de qualidade. Flexibilidade para a realidade de pesquisa de vocês, né? São equipamentos compactos, com alinhamento fixo, possibilidade de aquisição em tubo, em microtubo, em placa. Quando a gente fala em sorting, pensar em sorting misto, onde você consegue combinar enriquecimento, pureza, recuperação de células abortadas e a clonagem, né, o single cell sorting, no mesmo ensaio. Alta eficiência, ou seja, pensar em percentual de pureza, de viabilidade e de sensibilidade no seu processo, e pensar em nanocitometria, que já é uma realidade com alta sensibilidade aí chegando a 80 nanômetros. Eu trouxe um pouco dos nossos resultados aí em termos dessa família que eu falei tanto, né, de Cytoflex, como é que a gente está hoje na América Latina, tanto no universo da pesquisa como clínico, é uma realidade essa citometria de alta sensibilidade, e trazer para vocês também informação que, para além da citometria, a Beckman Coulter também tem toda a linha de automação, né, são os preparadores, inclusive de bibliotecas para GNS, para NGS, desculpa, genômica, os contadores de partícula e toda a linha de centrífuga também, desde as centrífugas de bancada até as ultracentrífugas também, dentro das nossas linhas. Bom, então espero ter trazido atualizações, ter trazido avanço e desafios para vocês aí nesse universo da imunologia e da leishmaniose. Obrigada mais uma vez, Daniel, obrigada a todo mundo que assistiu aí até agora.
Raquel, eu que agradeço esse painel enorme de tecnologia que você passou para a gente, né? É sempre bom a gente acompanhar, principalmente eu que já me sinto mais da velha guarda, né? Já não dou mais conta de acompanhar os upgrades tecnológicos da máquina, né? A gente acha ainda que as antigas máquinas, né, que vêm nos ajudando há muito tempo, são ainda altamente tecnológicas, e cada vez mais a gente fica surpreso com os novos avanços, né? A tecnologia é isso, a cada momento você tem um lançamento que melhora aquilo tudo que já foi batido martelo no passado e que nos abre possibilidade de expandir para o futuro. Eu agradeço você, agradeço a Beckman, né, por nos ajudar nesse evento, e fico muito feliz mesmo com a sua participação, porque realmente citometria e imunologia, né, são coisas que andam juntas e são indissociáveis. Né? Não dá hoje para a gente fazer imunologia sem a citometria como uma ferramenta, ou eu ouso dizer que é uma das mais importantes ferramentas da imunologia hoje. Então, realmente, muitíssimo obrigado.
Eu que te agradeço, Daniel. Não sei se tem alguma pergunta no chat, acho que não tive acesso a alguma outra pergunta aí.
Eu tive no chat e as pessoas não fizeram não, mas não sei se você gostaria de fazer alguma pergunta, Herbie.
Eu queria agradecer a Raquel. Eu gostaria de ter anticorpo para fazer umas 16 cores ao mesmo tempo, não acho que é o sonho de todo mundo, né?
Eu gostaria de ter esses tubos secos, daí a gente pode usar, porque o citômetro já tem. Se eu usar, comprar o tubo seco, ele funciona em outros citômetros ou só no Citoflex?
Não, os tubos secos são compatíveis com qualquer citômetro, na verdade, desde que respeite a combinação de fluorocromos que ele vai ter. Então, a gente tem, inclusive, alguns painéis ali do tubo seco que são em 8 cores, tem alguns painéis em 6 cores. Então, eles são adaptáveis, sim, a outros citômetros, seja da Beckman...
Né? Podia, de repente, ser flex, né? Que nem você tem o Luminex Flex, o CDA Flex. Quer dizer, você escolhe, vocês fazem e a gente pode ser.
Ótimo ponto, porque a gente também oferece esse serviço. Além dos tubos que a gente já tem pronto, desses painéis, a gente tem o serviço que vocês desenham o painel e a gente produz também. Então, se você tem, por exemplo, um projeto que vai ser desenvolvido aí pelos próximos anos, a gente tem alguns clientes no Brasil, inclusive, que já tem esses tubos padronizados, que é o seu próprio painel.
Precisava ver agora o preço, né? Não deve ser uma coisa barata, né? Então...
Eu acho que bota mais medo do que a verdade, viu?
É só, Claudinha, é só você falar. É só você pedir. Só você pedir. Claudinha, só você pedir desconto como participadora ativa do Immune-Leish. Então, você fala lá com o pessoal da Beckman, fala assim, olha, a gente quer um mega desconto.
Graça para a gente no início, para a gente experimentar, para ver se gosta.
Sim,
né?
isso tudo.
Se a gente conseguir publicar um trabalho com esse, a gente pode até pensar, né? Falar,
com certeza, por que não?
A gente aperta a Raquel aqui agora.
Pode apertar. Conta para mim qual que é o painel ideal e a gente conversa. Acho que isso é uma realidade, sim. Acho que a gente tem que trazer isso, tornar realidade na pesquisa no Brasil. Acho que no universo clínico já tem bastante gente no Brasil que já utiliza, mas para pesquisa é interesse nosso também. Então, conta para a gente quais são os painéis de interesse e a gente conversa.
Vou contar.
Raquel, agora uma coisa assim, claro que vocês setando, né? Uma coisa que eu sinto no dia a dia da citometria é que quanto mais cores a gente coloca, mais complexo é. E independente do citômetro, né, assim, a compensação se torna sempre mais complexa. Claro que esse é um sonho, é possível. Quando você tem moléculas que são muito expressas, então você fazer o multicolor é mais fácil, né? Eu não sei a experiência da Cláudia, do Daniel, né, de outros, né, mas assim... Eu fico me colocando nisso. E quando eu tenho moléculas que são pouco expressas, o multicolor fica bem mais difícil. Não é? E, assim, aí você vai agora com a bit com o mesmo multicolor, mas brilha, é lindo, né? Mas na hora assim, de achar, sabe? A Beckman dá esse tipo de assistência, de de poder ajudar a chegar nessa... Assim, a gente no dia a dia, a gente vai conseguindo, vai aprendendo, mas tem uns sistemas que são complexos, né?
Perfeito, ótimo ponto, porque não dá para a gente desassociar. Quando a gente começa a falar de painel multicolor, mais de 6, 7 cores, inevitavelmente vai ter uma população com um antígeno que é pouco expresso. Né? Ninguém trabalha com 7, 8 cores só com antígeno que é super bem expresso, que é antígeno constitutivo. Então faz muito sentido, mas não necessariamente a compensação ou própria identificação dessas moléculas precisa ser difícil. Eu acho que aí entra a questão de você ter um equipamento de alta sensibilidade, porque aí você tem uma facilidade também de identificar aquilo que é pouco expresso, se você tem mais resolução, tá? Então tem duas coisas. Um, você trabalhar com esse detector que tem maior sensibilidade, então você vai ter mais facilidade de identificar o que é pouco expresso. Tem a questão da compensação, que quando você trabalha com esse tipo de fotodetector, que não é só a questão dele ser mais sensível, tá, Herbert? Ele é totalmente digital. Então, ele te permite outros tipos de ajuste que quem está acostumado com a citometria mais antiga talvez tenha até medo de aumentar os painéis por conta dessa dificuldade e que, na verdade, dependendo do painel ali que você vai fazer, é até mais fácil, porque o software consegue atualizar essa compensação. E respondendo sua última pergunta, sim, a gente dá essa assistência. Eu sou uma das pessoas que dou essa assistência em como fazer esse ajuste, em como preparar esse ensaio. Toda vez que a gente vai fazer uma demonstração ou dar um treinamento de Cytoflex, uma coisa que a gente faz muito na Beckman é perguntar qual é o painel que vocês usam. Eu não vou com um painel que eu tenho de base aqui com BID e te mostrar o que eu tenho. Eu quero saber o que você tem e a gente vai trabalhar em cima do painel que você tiver.
Porque lá na Fiocruz, né, lá no LipMed, tem, né, um da Berkman, né, uma plataforma, né? Eu queria muito um dia, né, a gente ir lá, porque assim, eu estou acostumado com outros equipamentos, né? Mas lá na Fiocruz tem um da Berkman, acho que o Daniel agora tem um da Berkman, né, Dan?
O Daniel está com um de 21 cores.
A gente...
Tá chique,
assim,
né?
Vou falar só do...
Eu preciso
isso que a
aprender
Raquel,
a mexer.
isso que a Raquel... eu passo longe dele. Isso aí eu não tenho mais HD na minha cabeça para poder... O máximo que eu chego, eu vejo assim, ele de longe, assim, acho que morde e vai embora. Mas a Lu já tem feito bastante carinho nele lá junto com os meninos. Mas assim, a Raquel ela fez os testes no equipamento, né, quando a gente instalou ele, e aí o que ela disse eu posso confirmar nesse sentido. Assim, a gente já chegou com um painel que a gente tinha iniciado, que era 12 cores, para ela setar o painel nosso lá. Então, assim, o teste do equipamento foi feito com o nosso painel e um painel já de 12 cores. E aí, devagarzinho, a gente foi subindo o painel. Hoje a gente tem um de 16, ensaiando chegar a 18. Mas tranquilo, assim, a gente não tem tido muitos problemas em detecção, não. Na detecção daquilo que a gente vem... De vez em quando empaca alguma coisa assim, mas nada que você quebrar a cabeça um tempinho lá na frente, né, diminui a amperagem aqui, aumenta a voltagem ali, e por aí vai, você chega no negócio. Agora, para a gente, aí assim, não estou nem falando muito de equipamentos, isso acontece com todos, inclusive com o equipamento da Beckman. A gente sentiu bastante dificuldade, e aí não é o equipamento, e sim são os anticorpos, quando a gente vai fazer fosfoflow, né, analisar a proteína fosforilada na citometria. Porque aí você usa metanol, o metanol detona o marcador, e aí alguns marcadores são incompatíveis para metanol. Aí, assim, até agora a gente não conseguiu ter um cenário de muitas cores fazendo fosfoflow. Aí a gente tem que baixar a nossa bola, geralmente, para diminuir a quantidade de marcadores do painel para poder analisar essas proteínas fosforiladas. Assim, não... E de todos os equipamentos que a gente já usou, seja da Beckman, seja BD, seja o equipamento da Thermo, em todos os casos a gente sempre precisou diminuir a quantidade de marcadores para poder ter um bom painel funcional de proteína fosforilada. E aí aquele sonho de vou ter um painel de 18 cores com proteína fosforilada, Isso eu desencorajo qualquer um, assim, não quero jogar água na fervura para apagar o fogo, não, mas eu acho bem difícil, né? Mas, no geral, assim, o equipamento, agora, nesse equipamento em si, ele tem nos atendido muito bem, a gente tem gostado muito do equipamento. A Luciana com mais propriedade para poder dizer sobre isso, né, junto com os outros meninos do laboratório. Mas,
Daniel,
enfim...
esse é um ponto importante que você fala de, às vezes eu quero para essa aplicação fazer 18 cores e tive que reduzir. Microvesículas é uma realidade também. Às vezes tem gente que consegue fazer microvesículas e quer fazer um painel de 8 cores. E não é a realidade, não por conta de equipamento, até por conta de oferta de antígeno. Você não coloca tanta oferta de anticorpo para microvesículas. Você vai fazer ali um painel de 3, 4 cores, assim. E que não quer dizer que é pouco, você tem informações multiparamétricas ali de algo que é absolutamente inovador e que tem muita coisa para responder em imunologia. Então, claro que todo mundo quer fazer painel multicolor para uma determinada aplicação, mas tem muita coisa também com painéis não tão grandes, mas que também representam um avanço em tecnologia, né? Não é só aumentar o número de cor.
Mas é isso, Raquel, ó, te agradeço de novo pela participação, tá?
Eu que
Muitíssimo
agradeço.
obrigado mesmo pelo bate-papo, né, científico em relação à citometria, ou quem devia estar aqui, quem que é o nosso especialista na citometria para poder conversar mais, né? Mas, enfim, a gente vai conversando e Herbert, quando você quiser, se quiser vir passar um tempo aqui para poder fazer um test drive, tá?
Muito obrigado, meu amigo. Eu estou precisando aprender mesmo.
E pode entrar em contato também, de qualquer coisa a gente combina e faz, nem que seja aí por videoconferência, a gente conversa um pouquinho e discute aí esses painéis e o seu acesso ao Cytoflex da Fiocruz, no Rio mesmo.
Pô, muito obrigado, Raquel. A Joana, ela fez uma pergunta que eu acho que você respondeu para a Cláudia, né? Gostaria de saber se o Duraclone, por enquanto, estão com painéis prontos ou no futuro poderemos escolher, né? Acho que foi a pergunta que você respondeu. A Alda, ela precisou sair, ela te mandou um abraço, tá? E te desejou parabéns.
Ah,
Tá?
legal,
Eu
legal.
te agradeço, tá, Raquel, pela sua participação e pelo suporte na Beckman, tá?
Imagina. Eu que agradeço a participação e retribuo aí um abraço para a Joana e um abraço especial para a Alda também. Obrigada.
Nada, um abração, Raquel.
Vai ter um tempinho para almoçar ou não?
Esse foi o almoço no palestra no almoço, Cláudia, perdão.
É porque o evento, o primeiro dia ficou bem corrido, assim, ó. Let's change.
Mas lá disse que tinha um intervalo de meio-dia 1.
Let's change for the English, because David
Eu não falo
is
português.
Sorry, David.
We are discussing about the lunch time, so.
So sorry, Claudia. We put this conference in the lunchtime.
It's fine. I will take something to eat.
Just close the camera and take some snacks or this kind of things. So, Dave, thank you very much for accepting. Nice to see you.
Arnie's couldn't come, huh?
No, yeah, he sent me a message. Yeah, he had some problems, so he couldn't chair the first part of the conference. So
Alda did a good job.
Yeah, but anyway, we should try to find a way to meet everybody now after UKB opened.
Hello, Nathan.
Oh, Nathan is here.
Just came on.
There we go.
Dario is here too. So
how are you?
Fine.
Hey, how are you guys?
Hi, Dario. Hi, Nathan. Thanks so much, you all.
Yes, Daniel, I would much rather be sitting in Espírito Santo right now giving this talk.
Yeah, I think we're all done with the format. Hi, guys.
Hi. Good call. Yeah, I'm afraid my caipirinhas just don't live up to scratch.
Oh yeah, maybe next year, we can meet in person to have some caipirinhas, have a couple of drinks, and discuss a little bit about leishmania and immunology. This is such painful time with this social distancing.
And let's see if World Leash manages to get up and running in Colombia next year.
Yeah.
Oh yeah, it's gonna be in such a nice place in Colombia.
In Cartagena,
yeah, it'll be amazing. Anyway, I think we
could
start.
Bring some cachaça to make caipirinha.
That's good.
Make us feel at home.
So, what do you think, guys? May I start?
So is Thiago here? Thiago is here. Okay.
Just arrived. Yes.
Hey guys.
Good.
Thank you.
So I just to briefly discuss the section, Herbert and Danielle. So it's, thanks for the invitation to be a chair of this section. So I'm gonna make a very brief introduction about Nathan, Thiago, and David. Is that correct? And then I'm gonna open the mic, and then you guys just like give the 45 minutes presentation, and then we have all the discussions after the last talk. Is that correct?
Yes, it's like a round table. Yes, thank you.
I was a little bit confused because I thought that we were gonna have a lunch break between the two sections.
I'm so sorry. We put the support in the lunch. So presentation conference during lunchtime. So we're missing the lunch. No, we don't have lunch more we're not in the Congress, but the idea was like that.
Yeah.
Sorry, Thiago.
It's okay.
So Thiago, you are still in the NIH, or you are back already?
My contract ended this month, so now I'm traveling. I'll be back to Brazil next month.
Because I got your short CV, and it was not clear for me if you were already in NIH or if you returned to Brazil yet. Not yet.
Yeah, I'm kind of in the limbo now. I need to.
He's looking position, right?
Yeah.
Good.
In Brazil, though, my home country.
Exciting.
I'm taking a break now, like I'm traveling before, like I need to process everything.
Nice.
So you can start there. Thank you so much. Daniel, it's you, sorry.
Okay. Thanks so much, guys, for accepting the invitation. So it's a pleasure to us join all here in this scientific meeting. So I'm just gonna introduce Dario. I mean, for sure he doesn't need, but I'm gonna give a short introduction. Dario is a professor, titular professor from Faculdade de Medicina de Ribeirão Preto, University of São Paulo. And Dario is going to chair the conference, the afternoon conference of this meeting today. So Dario, you can take the word. So thank you very much for accepting.
All right, Danielle, Herbert, I have to thank the invitation to be chair of this very interesting afternoon conferences that we're going to have today. It's a pleasure to be part of this satellite meeting of the Brazilian Society of Protozoology. So it's a very, very interesting. And today we're going to talk a little bit about innate immunity and host pathogen interaction, which is a fascinating topic. So I'm going to just introduce all the three speakers we're going to have this afternoon. And then we're going to have 45 minutes conference from each speaker. And then all the questions and discussions we're going to have around 3:30 p.m. So we're going to have all the questions and discussions open in a roundtable format for about 1 hour. So feel free to take notes of your questions and feel encouraged to submit the questions. So we're going to have time to discuss. And it's a very non-formal and presentation. And I strongly encourage all the students to ask questions and discuss, and there is no not smart question. All the questions are valid, and I'm sure all the speakers are going to be happy to discuss, and it's going to be a fun moment for all of us. So I'm going to start introducing Nathan Peters. He's the University of Calgary in Canada, and he's going to deliver a talk about the interplay between innate and adaptive immunity in cutaneous leishmaniasis, focusing on a central role of monocytes. And Nathan, as I mentioned, it's an associate professor in the Cunningham School of Medicine, University of Calgary. He's for many years interested in understanding how the leishmania and sandfly interactions and goes, and this impacts the overall infection in mammalians. So focusing in inflammation, how the inflammation alters the regulation of the expression of immunity. And this is obviously very important for vaccine mediated immunity as well. So, and he's also interested in new therapies and vaccines to combat chronic infectious diseases such as leishmaniasis. So after Nathan's talk, we're going to have a talk by Thiago Souza Vieira, which is the Brazilian member of this afternoon conference today. So Thiago, I'm sure most of you guys know Thiago from previous meetings. He's very active, and he graduated in the program of Immunology and Inflammation in Instituto de Microbiologia Paulo de Góes in University of Rio de Janeiro, Federal University of Rio de Janeiro. And then he was in the Massachusetts Medical School and was a visiting fellow at the NIH, where he's still in the NIH now. I think he's going to deliver this talk from the Bethesda. So it's a pleasure to host Thiago, and Thiago is interested in understanding the plasticity of neutrophils and how these responses regulates the susceptibility to Leishmania major infection. This is going to be the topic of his talk. So feel free to discuss and to talk to Thiago during the roundtable and maybe after the roundtable as well. And the third talk we're going to have is from Dr. David Moser. David Moser is in the Maryland, in University of Maryland in United States. And he's going to deliver a talk about the macrophage response to Leishmania. It's a fascinating topic for all of us. And David is interested for many years in the heterogeneity of the macrophages and how this heterogeneity affects the response, the host macrophage interactions with Leishmania, and how this interaction impacts the adaptive immune responses. So there are many years of contributions by Moser in this field. So I'm sure it's going to be a fascinating talk as well. So that said, I'm a little bit ahead of in advance. We still have some time, so it's good. I'll just give a very short introduction about the speakers, which I like, because then people feel more encouraged to discuss and to ask questions to the speakers. If the chair usually say about how many papers, how the impact of the publication, citations, usually the students get so shy about asking questions to the students. So I like to just talk about the science and how amazing these researchers are. With that said, I'm going to give the word to Nathan Peters, and again, in name of Brazilian Society of Protozoology, it's a big pleasure to host the section, and thank you very much for accepting the invitation. And now, feel free to start your talk, Nathan.
Oops. I have to give Zoom permission. Okay, I'm gonna have to log out and log back in. Sorry, I've been on vacation. This is my first use of Zoom after an update. So I'll be back in 1 second.
this type of a meeting, it's a great opportunity to us to see each other, because it's a long time without any congress. So today morning I feel very happy, and about this possibility to find it. It's a great pleasure to be here.
Yeah, it's for sure with a caipirinha and having fun in Caxambu would be much more pleasant for sure, Herbert. But I agree with you. It's always good to see friends and colleagues. So it's very nice to see each other, and even via web conferences, discussing science, watching the research progress. It's always fun.
It's good to keep the environment, so the scientific environment and fun environment. So
yeah,
it's nice. Even by internet, we can keep it.
Okay, how's that?
Yeah,
good. Sorry about that. Just a quick change to the preference there. So Herbert and Danielle, thank you so much for the invitation. I think all of us are probably feeling very disconnected these days, and any opportunity to really settle in with colleagues is appreciated, even if it's in this Zoom format. So, like Dario mentioned, I'm really today going to be talking about innate immunity, but also adaptive immunity, and the interactions between the innate and the adaptive immune system during cutaneous leishmaniasis, and specifically looking at monocytes. So I'm going to apologize in advance. Paul K. Kind of alluded to the complexity of the Leishmania parasites, even within a single strain. I'm going to jump between major and amazinensis a little bit today. So not to underemphasize the importances of the differences, but some of you may find it a little bit annoying. So I just want to start off by really acknowledging who did most of the work here, and I really want to emphasize Mateus Carneiro, postdoc in the lab. I'll be talking about a lot of his work, but also Audrey Romano, who I worked with while I was at the NIH, as I talk about a little bit of older work that she was involved in. So I'm really going to start off with the basics here, and to sort of lay a foundation for the work I want to talk about, and that is touching on what are the effector mechanisms that the immune system uses to deal with infection. You can categorize these into two basic camps. You've got the innate immune response, which kicks in quickly to help and try and control an infection. And when we talk about leishmaniasis, we really predominantly think about phagocytes acquiring this intercellular pathogen and generating things like reactive oxygen species in the phagolysosome to eliminate it. And then on the other side of the spectrum, you have the adaptive immune response, which takes a little bit of time to kick in, but when it does, you can generate a response like the Th1 immune response mediated by CD4 cells, interferon gamma production, and the activation of phagocytes that may be having trouble eliminating the pathogen that's adapted to live inside the phagolysosome and induce the inducible nitric oxide synthase and the production of nitric oxide and eliminate the pathogen that way. So while immunology can be quite reductionist, and certainly I'm going to talk a lot about animal models today, and again, the study of immunity can really focus on innate immunity or adaptive immunity. We know as people that work in infectious diseases that these two arms of the immune system are working at the same time for the majority of the time during an infection. And so, as you can imagine, there's a lot of crosstalk between the innate immune response, think about effector mechanisms like complement, phagocytosis, and the adaptive immune response, where you think about B cells and T cells. And really, although the adaptive immune response takes some time to kick in, once it does, it really harnesses the innate immune response to fine-tune it, improve its specificity and improve its type. And when it comes to how adaptive immunity fine-tunes the type of immune response that is being generated, we often fall into this definition of CD4 T cell subsets and how these can tailor immunity to multiple scenarios. In the context of parasitic infections, we often think about the Th2 response controlling parasitic worms, or the Th1 response controlling something like Leishmania infection. And of course, this paradigm is quite broad now. Don't worry about the details here, but basically there's lots of different T cell subsets, and you could tailor the immune response to multiple scenarios. And one of the sort of feathers in the cap of leishmaniasis is that understanding Leishmania responses was really central to understanding these different T helper subsets, and specifically in the context of Th1, Th2, where the infection of monocytes and macrophages in the presence of a Th2 response has been associated with the generation of M2-like or arginase-producing phagocytes, production of polyamines that assists the parasite in its growth, versus the Th1 response, interferon gamma production, and the activation of M1-like phagocytic cells, nitric oxide production, and parasite elimination. And everyone knows the paradigm. Most of us find it slightly annoying, overly dogmatic, not very useful. But we think about Th2 being non-protective and Th1 being protective, certainly in the context of cutaneous leishmaniasis. So while the paradigm has some limitations, we can't forget that it does have a quite a relevant application, and that is when we talk about prophylactic vaccination. Most people are trying to make a vaccine that generates Th1 immunity, not a vaccine that generates Th2 immunity. So the interesting thing about this Th1, Th2 paradigm, and all the T effector subsets, is that they cross-regulate each other. So one response can downregulate the other response. And this can happen in several different ways, probably more ways than I'm going to allude to here, but this can happen at the level of the CD4 T cell. So an individual cell could be exposed to IL-4 or IL-12 or interferon gamma, and that will push it towards these different subsets associated with control or not of an infection. So this cross-regulation can occur at the level of a single cell, a single CD4 T cell, whereas maybe if you are exposed to a little bit more IL-12 or interferon gamma, that will lead to the STAT1 phosphorylation and the induction of TBET, generation of these Th1 cells. So it's at the single cell level. If you do that over time during priming, that can lead to the accumulation of more Th1 cells at the population level. And then really what I'm going to be talking about today, which is how these different cytokines or these different environments can impact phagocyte activation and parasite elimination. And the example I'm going to use here is maybe you've got some phagocytes that are initially exposed to interferon gamma, but in the context of subsequently being exposed to Th2 cytokines, you sort of lose that ability to mediate protection and actually can become permissive. So it's within the context of these different immune concepts that Matthias and I had a real interest in understanding and observation using Leishmania amazonensis infection in C57 black 6 mice. And this basic observation is that, despite the fact that we know this interferon gamma, this central Th1 cytokine, very important for Th1 immunity and protection, during the early phases of L. amazonensis infection, the impact of being lacking interferon gamma is really undetectable. So here we're looking at the lesion size, so the disease, and here we're looking at the parasite load. And interestingly, if there's no interferon, if you have a knockout mouse for interferon gamma, it doesn't really seem to affect things at all during these early phases of disease. Now later on, you get the phenotype that you expect. an enhancement of disease. But we were really interested in what exactly is going on here. And one of the first things we did was, because of our history of looking at Leishmania infection using sandfly transmission, is we wanted to repeat this using sandfly transmission. And you know, I want to point out before I move to that is that this observation where interferon gamma doesn't seem to have a role during early infection, despite its importance, is actually quite common. You can look through the literature and lots of different infections where during early infection doesn't seem to be important. So this is with the needle challenge. You know, does it hold true when we think about the actual physiological transmission of Leishmania? And so we did this experiment, and in fact, this phenotype where the lack of interferon gamma doesn't really seem to be important was actually extended. So here we are, 4 weeks of infection, and the lack of interferon gamma just seems to have no impact. So we really delved into these early immune responses to try and figure out what exactly is going on. And early on, what we found was, you know, really what we expected. If you have an interferon gamma knockout mouse, here we're looking at mRNA expression. Obviously, you don't find any mRNA expression, but you push things towards IL-4 production. So where in the wild type mice, you previously did not get IL-4 production. If you don't have interferon gamma, then you push the immune response towards this Th2 pole. So this whole idea of cross-regulation seems to hold true, but despite these, you know, dramatic changes in the immune response, it doesn't really seem to be affecting the infection at all. And it's not because there isn't enough interferon gamma around to make a difference, because if you actually look at one of the effector outcomes of interferon gamma production, which is iNOS production, you know, very early, you can see day 14, day 21 after infection, the wild type mice are making iNOS, so it is having an impact. And you know, if you don't have interferon gamma, you completely lose that. So our takeaways here is that, you know, the early response to wild type mice is not, you know, silent, quote unquote. There is interferon gamma around, and it does seem to be inducing a response. And it's a fairly robust response. It doesn't seem like it's weakened or it's not skewed Th2. And in fact, despite the skewing in the setting where you are lacking interferon gamma, you don't enhance the disease at all. And in fact, what's really curious is that once the interferon gamma is induced here, starting at day 7, you actually get a very dramatic increase in parasite numbers, not a control of parasite numbers. And so, you know, we started thinking about this, you know, in sort of the context of parasitism. And thinking about parasitism not as just simply immune evasion or immune deactivation, but rather a coevolution. And of course, we like this idea of the Red Queen hypothesis, where the host and the pathogen, this interaction, are adapting to each other, and they both just kind of stay in the same place. So, you know, the question was, you know, has Leishmania abyssiniensis, you know, adapted to depend upon the Th1 immune response rather than turning it off or evading it? Maybe it's actually adapted to make use of it. And how we went about trying to figure this out was really heavily influenced by this previous publication that we had made in 2017, where we were looking at inflammatory monocytes and the different roles of inflammatory monocytes during primary infection or secondary infection. Secondary infection, essentially you have pre-existing Th1 immunity. So the question that we were trying to answer sort of kind of stemmed from some imaging that we had done following sandfly bites. And we were sequentially imaging these probe sites after sandfly bite. And we were sequentially imaging the same site over time. And what we found is kind of was interesting or curious to us. And that was, early on the parasite is associated here in red with a GFP high cell, which in this case is, it's what, a 99% chance this is a neutrophil. But then over time, it no longer seems to be in those neutrophils. And it doesn't really seem to be doing anything in terms of proliferation or even moving. During the first 4 days of infection, I've highlighted day 4 here because I'll be talking about this time point quite a bit. Nothing seems to be happening until about day 6, when you start to see the parasite begin to proliferate within a cell. So we were really curious as to, visually, it doesn't look like there's much going on here, but what exactly is happening with the parasite during this early phase. And we knew that a lot of the parasites ended up inside neutrophils early on, but what was this cell type later on, and what exactly was going on? So one of the things we did is we looked at sandfly bite sites, and we obviously spent a lot of time looking at neutrophils. But if you wait a little bit longer after that initial phase and the peak of neutrophil recruitment and infection seems to be around 10 to 16 hours. But if you wait a little bit longer, here we're looking at 18 hours. Sandfly bites induce not only neutrophil recruitment, but not surprising, also inflammatory monocyte recruitment. And there's a lot of these cells that come into the site. So we got very interested in inflammatory monocytes and what might be going on with these cells. So just, by way of background, inflammatory monocytes, they're generated in the bone marrow, they're a mononuclear cell, they're phagocytic, they circulate in the blood. And then if you've got something like a Leishmania infection, a site of infected skin, you'll recruit those monocytes into the skin. And then the interesting thing about monocytes is they're incredibly plastic. So in response to environmental cues, they can become monocyte-derived macrophages, dendritic cells. It's a whole spectrum of just a lot of heterogeneity here. And so they can take on different effector functions in response to different inflammatory cytokines. They can become M1-like, associated with pathogen elimination. They become M2-like, associated with tissue healing. They can migrate or mature towards a dendritic cell phenotype where they make a lot of nitric oxide, this idea of this TIP-DC population. And they can also do antigen presentation and involved in T-cell priming. So you get this maturation cascade once they're recruited into the skin, and you can actually phenotype this or follow it by flow cytometry in an adoptive transfer system where you take CCR2 positive inflammatory monocytes, put them into a mouse, and then track them in the skin over time. So here the monocytes are going into a day 10 infection. And of course, at the beginning, they're all classical CCR2 positive, LY6C positive monocytes, but over time, this population matures, you lose that inflammatory monocyte phenotype, and then they start to become these other cell types. So we were interested in these cells because they also seem to be around after sandfly bite. And using flow cytometry and an RFP expressing parasite, we were able to track where the parasite is going and what it's doing. And by flow cytometry, we were able to recapitulate what we saw by imaging, and that is during the first hours, the phagocytic cells load up, but then they sort of go through this stall where they don't really undergo any proliferation. So there's no increase in the total number of infected cells until here, looking at day 7, and they kind of take off again. And what we found here using a needle inoculation was that a lot of these parasites during the stall period seem to start loading up in inflammatory monocytes or inflammatory monocyte-derived cells. And you can enumerate this and track it over time, where initially a lot of parasites load up inside neutrophils, and then you go through this transition period. There's no change in the total number of infected cells, as you can see over here on the left, but you go through this transition where they are now transitioning into monocytes. This was a bit curious because while there was some work I've referenced down here that maybe the parasite really would like to be in a monocyte, there's also a load of work done by some people here where actually monocytes were important for parasite killing and Th1 immunity. So we wanted to follow this up a little bit more closely, and what we did is we sorted out these infected monocytes, and we just wanted to know, these parasites that end up inside these monocytes, is this really a good host cell? And monocytes really are a good host cell, both in mice and in people. So you can sort out these infected monocytes from the skin, and the parasite will continue to grow very happily over time. And we also did this with human monocytes. I'm just not going to show that. So this work, which is with Leishmania major, you know, what does this have to do with this interferon gamma question that I started out with? And really the link here is what happens when you start to look under conditions of pre-existing immunity. So if you have a mouse that already has Leishmania specific Th1 immunity, what you find is that the monocyte recruitment is greatly enhanced in the presence of that Th1 immune response. And you can reduce that fairly significantly if you just block the interferon gamma production. Okay, so when you have pre-existing Th1 immunity, you have interferon gamma, you get a lot more monocytes. And this is associated with a very large increase in several monocyte recruiting chemokines, such as CXCL9 shown here. The interferon gamma, as you might expect, leads to a high proportion of these monocytes and monocyte-derived cells making iNOS, and you reverse the protection that's conferred by the pre-existing Th1 immune response. So here you're controlling the parasite load, but you lose that control if you block the interferon gamma. So all these things seem to go together to show that Th1 immunity, interferon gamma recruits monocytes and activates these monocytes to kill parasite. And just to really drive that point home, we also looked at this, in the previous slide, I talked about blocking interferon gamma. Here we're eliminating monocytes out of the system using a CCR2 DTR mice treating with diphtheria toxin in mice that do not have pre-existing immunity, or mice here that have pre-existing immunity. And what you find is if instead of taking the gamma out of the system, you take the monocytes out of the system, here you lose this iNOS production, and then you lose this control of parasite numbers. So these studies really support the role of monocytes in the setting of pre-existing immunity, both their recruitment and their activation. And so our thinking is that we sort of go back to this early phase of disease and the role of interferon gamma. It seems to be there, but it's not impacting the disease. Maybe it's actually benefiting the parasite somehow. The parasites come to depend upon this interferon gamma production. We just thought about it a little bit differently. So if you have interferon gamma, you get these chemokines produced, you get monocyte recruitment, and perhaps in the setting of LM sinensis and the primary response, that monocyte recruitment is going to actually facilitate parasite expansion because you're providing permissive host cells. Okay. So we started looking at these cells more carefully, and what we found is that when you don't have interferon gamma around and you look at the CD11b positive population, you lose the iNOS producing cells, which is precisely what you expect. But you also start to lose arginase producing cells, which isn't what we expected. So here we thought that these iNOS positive cells, these double positive arginase iNOS positive cells, would just sort of shift over here in the absence of interferon gamma, and you would just get a lot of arginase producing cells. But in fact, the overall frequency of arginase producing cells also dropped. And we also looked at this phenotype. We talked in the last session about PDL. We also looked at this alternatively activated macrophage phenotype, and that is the MHC class 2 PDL2 expressing phagocytes. And in wild type mice, you get about 40% of these 3 weeks after infection. If you don't have interferon gamma, this frequency also drops, and this was a bit surprising to us. We thought, well, if there's no gamma, and we've skewed things Th2 with all that IL-4 around, this should increase, but it actually dropped. And so why is this? And what does it have to do with monocytes? Well, the interferon gamma knockout mice, as you might expect, are really deficient in several monocyte recruiting chemokines. So here we're just looking at CXCL9. If there's no interferon gamma around, you basically have very little CXCL9 production. And at these later time points, when you've got this interferon gamma induced monocyte recruitment, what you find is that there's a deficiency in monocytes, okay? So early on, the innate monocyte recruitment is fully intact in the absence of interferon gamma. But remember, the gamma kicks in at about day 7, and now we start to see this, this deficiency in monocytes, okay? So that's by frequency and also by numbers. And if you look at the infected cells, because we can track the Leishmania amazonensis infected cells using RFP, what we find is you start to see a deficiency also in the number of infected monocytes. So the whole idea here is that, this is the way we sort of think about it: interferon gamma produced by Th1 cells interacts with a phagocyte, you eliminate the parasite, it's all good. But in fact, there's a caveat to this, and that is that these Th1 immune response via interferon gamma can also induce these monocyte recruiting chemokines. So you're providing more permissive host cells. And maybe these two things just sort of balance each other out during early infection. And that's why we see this phenotype. So we started to follow this up. And one of the first things we did is we did a bone marrow chimera experiment to see, just what proportion of the infected population actually loads up in these, inflammatory derived cells. And so what we've done here is we've created a bone marrow chimera so that we can discriminate between tissue resident cells and recruited cells or inflammatory monocyte derived cells. And what you find is that you can sort of track the bone marrow derived cells and the Leishmania amazonensis RFP and look at this population. And what we find is that most of the total cells in the site of infection are inflammatory cell derived. And if you look at the infected cells, then even a greater proportion of those, about 90% of the infected cells are these inflammatory bone marrow derived cells. So it seems, these cells are, very important. So the next question was, well, let's take these inflammatory monocytes out of the system during this early time point. And what we found when we did this is based on a lot of previous literature, is exactly what you would expect. So when you take the monocytes out of the system, you see deficiencies in the Th1 immune response. Okay, this has been previously shown. It's due to the fact that monocytes become efficient antigen presenting cells and reduces Th1 immunity. So you take them out of the system, you lose that Th1 response, and you shift things very strongly towards the Th2 response. Now, interestingly, when you fairly carefully look at parasite loads during this early window, this massive change in adaptive immunity does not translate to enhanced infection. And in fact, we saw lower parasite loads in the absence of the monocytes, despite this transition here. Here, looking at parasite titer, or even just the number of infected cells with a CD64 phenotype. And again, looking at this arginase inos or MHC class 2 PDL2 phenotype, when you don't have monocytes around, you see big deficiencies in the frequencies of cells that are making arginase and the frequencies of cells that are adopting this alternatively Th2 associated phenotype, even though you push things strongly towards Th2. And this seems to be associated with the fact that, when you don't have monocytes around, there's these big deficits in the frequency of infected monocytes and monocyte derived cells. So, we started working with this model, and that is, and that is the gamma has an anti-parasitic role, but it may also have what I call a parasite neutral, which is essentially a pro-parasitic role, where it can induce the the recruitment of more permissive cells. So it kind of counterbalances. So you can have these multiple outcomes depending on whether the cell receives a subsequent gamma signal or not. So at this point, we really want to go back and really understand, well, are these monocytes, are they killing any parasites at all during this phase, or is the gamma response just completely backfiring? And the way that we did this is we needed a way to get the monocytes into the site, but not have them respond to the gamma. So what we did is an interferon gamma receptor knockout bone marrow chimera. What that allows us to do is you get the monocytes into the site because they're responding to the chemokines, but they cannot respond to the gamma in terms of their activation. So equivalent numbers of monocytes, and now we get the monocytes into the site. And although the changes aren't huge, here we're looking on a log scale. What we find is that if the cells are there, they can't respond to the gamma to become activated. Then you start to get enhancement of the parasite load, about a 10-fold enhancement in the parasite load. So what this tells us is that the lack of parasite expansion, when you shift away from the Th1 response towards the Th2 response, is not a parasite intrinsic limitation. This isn't because the parasites just can't proliferate fast enough to take advantage of that. The other point is that it confirms that monocytes are a more permissive host cell in the absence of the interferon gamma signaling. So the gamma is doing something, and the monocytes do mediate some killing. Now, we made use of the fact that we also set up mixed bone marrow chimeras. So just to focus your attention on this group here, which is now we can directly compare head-to-head wild-type monocytes or monocytes that are missing the interferon gamma receptor. So they get into the site in equal numbers, but this population cannot respond. And what we found there was that the parasite does seem to prefer, or when you look at the infected cells, you find that most of the cells are bone marrow derived, and then these bone marrow derived receptor knockout phagocytes, inflammatory monocyte derived phagocytes, as shown here when you look at the total population. Now, because we were tracking these by flow cytometry, we could really delineate more specifically where exactly this preference for the receptor knockout phagocytes or monocyte-derived cells existed. And this was really interesting. So when they come in as really immature monocytes, the presence of the interferon gamma receptor doesn't really seem to make any difference. So it seems like these immature inflammatory monocytes aren't responding to gamma yet. They're pulled into the site, but they harbor the parasite in equal numbers, whether they have the receptor or they don't. But as these cells mature and acquire the ability to respond to interferon gamma, the receptor starts to make a difference. You start to find the parasite in those cells that are missing the interferon gamma receptor. So our conclusions here is that, interferon gamma mediated killing occurs in monocyte-derived cells. The monocytes they can respond to gamma, but you still find that a significant portion of the monocyte infected cells, even if they have the interferon gamma receptor, are still maintaining infection. So it doesn't go down to zero once they undergo maturation. So you still find the cells here. And the last thing is that it suggests that recently recruited immature monocytes are really quite a permissive host cell. Okay, so this population here, the lack of interferon gamma receptor didn't make any difference. So they're not responding to gamma. So it actually is quite a good place for the parasite to be. So we really wanted to understand, what are the characteristics of these infected monocytes? And the way that we went about doing this, which seemed like a great idea at the time, but which was technically quite challenging, was to sort infected monocytes or uninfected monocytes from the same site of infection, and then using nanostring look at gene expression. And the thing that actually popped the most here was the fact that infected monocytes, at least at this time point, really produced a lot of chemokines. There's a lot more chemokine gene expression that would lead to subsequent inflammatory cell recruitment. And this sort of fit the idea that if the parasite gets inside the monocyte, that monocyte will recruit more monocytes, which is what the parasite wants. In addition, using flow cytometry, we also looked at some other characteristics of these infected monocytes derived from the site of infection. And interestingly, we found that the affected monocytes really pushed mannose receptor expression and IL-4 receptor expression. And this phenotypes, here looking at the red bars, are the infected monocytes. This phenotype really had nothing to do with Th1 or Th2 immunity. If you sort of knock these pathways out, you still get this robust upregulation of surface expression of these Th2 or M2-like cells. So this seems to be very dependent on the parasite itself. And so essentially what's happening here is it seems like the parasite is infecting the cell and really pushing the expression of the IL-4 receptor alpha, as well as the mannose receptor, trying to create a niche for itself. And this had nothing to do with the Th1 or Th2 immune response. Also following up on this other phenotype, the MHC class 2 positive, PDL2 positive, M2-like phenotype. Again, the infected cells were more likely to have this phenotype, shown here by these flow plots. In the blood, these are immature monocytes. They don't express these markers. But if you look at the infected cells in the ear, a big push towards the MHC class 2 PDL2 M2-like phenotype. In the absence of interferon gamma, you start to lose these cells because it's the monocytes that are becoming these cells. So there's deficiency in the recruitment of these cells. And in this case, it was dependent upon the Th2 immune response. So if you have the STAT6 pathway knocked out, so that's the pathway induced by IL-4, you start to lose these cells with this phenotype. So, this all really fit. It seemed like the parasite was really trying its best to create a niche within cells, monocyte-derived cells that were adopting a more M2-like phenotype. And so the question was, well, how dependent actually is the parasite upon Th2, the Th2 immune response? So instead of using interferon gamma knockout mice to knock out the Th1 response, we use STAT6 knockout mice to knock out the Th2 response. And as you might expect, in the absence of STAT6, things get pushed very, very hard towards the Th1 response. Here you're looking at interferon gamma, TBET. You lose IL-4 production. Not as dramatic, but you lose GATA3 expression. So this is the transcription factor associated with Th2 CD4 cells. A big shift towards way more INOS positive cells. A huge drop in the number of araginase positive cells. So I thought, well, this has got to move the needle in terms of the infection and the parasite loads itself during this early phase. And if you look at lesion sizes, the STAT6 mice do have lower lesions, but this has nothing to do with parasite load. The parasite loads are the same, and in fact, early on, the lack of Th2 immunity, this huge push towards Th1 immunity, early on, it seems like there's actually more parasites there. And if you look at the mean fluorescence intensity of the RFP signal, it's actually higher across the board. And this touched upon some older observations that I'll just list here if you're interested, where people had done these experiments in STAT6 knockout mice, and the results kind of didn't make sense in the context of the Th1, Th2 paradigm. So, from this, we decided that, leishmania doesn't really seem to depend upon the Th2 immune response, and actually, over-enhancing the Th1 response doesn't seem to help clear parasites. So something that we wanted to do is we had to kind of confirm that this was all associated with CD4 T cells. And so you have to look at this data very carefully, but essentially what we did is we looked at gene expression of Th1 or Th2 associated genes, and we depleted the CD4 T cells before doing this. And what we found is these switches, these polarizations in these different knockout environments were required. We didn't pick them up if we depleted the CD4 T cells. And you can also look at this a slightly different way, just looking at the ratio of gamma positive cells to IL-4 positive cells in the different knockout scenarios. And what you find is, in the wild type setting, you got it for every 5 gamma producing cells, you've got 1 IL-4 producing cell. And if you knock out the gamma, you lose that. But if you knock out STAT6, you enhance it. Okay, so this is definitely associated with CD4 T cell immunity. So what's going on here? It seems like a big disconnect. Well, as you might imagine, as you push things towards Th1, you're also generating a lot more chemokine production. And here, if you count the number of infected cells, it's actually resulting in more infected cells. And when you look at what's being influenced by this shift, it's an increase in the number of monocytes coming into the site. And if you look specifically at the infected cells, you can see that the parasite is loading up into the monocytes. And so this heavy skewing actually kind of backfires because of this dual role for the interferon gamma. Sure, you're probably more efficiently killing, but you're also pulling in more cells that the parasite can infect. So that explained what was going on in terms of the total population. But at the single cell level, we also had this observation where the individual cells seem to be harboring more parasites based on the MFI of the RFP, which is linked, of course, to the number of parasites per phagocyte. So we also wanted to understand, well, what exactly was going on here? Well, something that was associated with this heavy skewing towards Th1, and this is where we get to the complexity of the host pathogen interface. You also pushed more IL-10 gene expression and more FOXP3 gene expression. Now we know in leishmaniasis, the IL-10 is coming from multiple sources, as Paul alluded to, but we also have the expression of this transcription factor associated with T regulatory cells. So it seems like the system is trying to compensate for the hyper Th1 environment, and is the IL-10 mediating this enhancement of infection on a per cell basis. And that's exactly what we found in the STAT6 knockout environment. Here's the enhancement of infection on a per cell basis. If you block IL-10, it goes back to normal. All right, so STAT6 doesn't really, from one sort of the take we have is that STAT6 doesn't really facilitate parasite growth. It regulates Th1 mediated monocyte recruitment and IL-10 production. That's its primary role, actually, which is I think quite different than how we might have thought about it before. So in terms of this L-amazonensis project, the last little bit of data I want to go to before I hopefully can get to some newer data is that I can't overemphasize this enough. It's really critical to be able to trace, because of the heterogeneity of what the monocytes can develop into. It's really critical to be able to do lineage tracing experiments. And so what we did is we've got a CCR2 Cree ZS green mouse. And the experiment we set up was to label the monocytes that came in during that innate wave, interferon gamma independent wave, and then label the monocytes that came in during that interferon gamma dependent Th1 mediated wave, and just see, like, where does the parasite like to be? Well, early on, it loads up inside the monocytes that come in during the innate wave, as you might expect, but then that really falls off quite dramatically. And you see this large transition into the monocytes that are recruited during the Th1 phase. And that's where the parasite is, and that seems to be where it likes to be. And if you look at the phenotype of these cells, because we know at some point they were a blood-derived CCR2 expressing inflammatory monocyte, even if they develop into something else, what we can see is that at day 14 post-infection, those monocytes have really matured into cells that we associate more with parasite elimination, whereas the cells, the fresh monocytes that have been pulled in by the Th1 response, have this very immature phenotype that is non-responsive to signaling through the interferon gamma receptor. Is probably a good spot for the parasite to be. And that's just based on the surface staining that we can do that allows us to trace all the cells that were originally CCR2 positive. And we also did imaging to look at this, so you can see that the green cells that were labeled during the innate wave, innate recruited monocytes, the parasite's not really associated with those cells so much. They're associated with a different population. Whereas if you look at monocytes that were labeled during the adaptive wave, that's where the parasite seems to be. And you can also look at these more carefully, these beautiful big Leishmania amazonensis phagolysosomes. The parasite looks very, very happy there. Okay. And you can also take this a step further and look at these different phenotypes, and what you find is that the monocyte-derived cells, whether they maintain that monocyte phenotype or are these monocyte-derived macrophages, or different populations of monocyte-derived macrophages, they tend to have this M2-like phenotype, whereas if they really differentiate fully and become dendritic cells, then they lose this PDL2 expression and just are expressing MHC class 2, and you might think these are much better at controlling parasite numbers. And just the last bit of data to tie this part up is we also looked at the mean fluorescence intensity of these different lineage traced monocytes, and we found that it was the monocyte-derived macrophages that seemed to have the most parasite. And if you sort these populations, and this is just two different assays to looking at the viability of the parasites within these monocyte-derived populations, depending on their maturation status over time, what you find is once they start to adopt dendritic cell-like phenotype, the parasite is less able to expand in vitro culture. If you look at the frequency of cells that are harboring at least one parasite that can subsequently proliferate, you see that as they go through this maturation cascade or process towards dendritic cells, this frequency drops. So early on, when they're monocytes, the parasite is in a happy place, but as they mature, they start to be eliminated. So the take home really for us has been, as you sort of skew things back and forth, and why you get this weird phenotype, where if you negate Th2 immunity, push a Th1, or negate Th1 immunity and push a Th2, why you get this null phenotype early on in infection is because of the dynamics. It's actually somewhat complicated, the dynamics of what's going on in terms of both activation, but also recruitment. So when you have a strong Th1 immune response, you do get a lot of killing, but you're also pulling in a lot of monocytes. Push things the other way, you have less killing, but you're also pulling in less monocytes. So as you increase killing potential, you're also increasing the permissive monocyte recruitment, and so it cancels itself out. So one of the things that we've been thinking about a lot is that when you really highly polarize Th1 immunity, it might actually backfire. There's a caveat here that we're going to have to keep an eye on in terms of how we generate protective immunity. Okay, so with that, I'm just looking at my time, and I think I'm going to cut short some of the newer data and just move along to my acknowledgement slide. So I'll have to do this again in the not too distant future, so I can talk about the newer data. But basically, just thanking the funders, and as I said, A lot of this work was done by Mateus here, postdoc in the lab, Audrey Romano. And this is the lab from, this is a pre-COVID picture because we haven't actually been all together since. So again, thank you for the opportunity, and I will stop sharing.
So thank you very much, Peter, Nathan Peters. We are right on time, so I'm moving straight to Thiago Souza Vieira from Federal University of Rio de Janeiro. So need to share your screen, Thiago.
Yes, just a second. Can you see? Is it working?
Yes, we can see. All right, so thank you, Daria, for the introduction. And I also like to say thank you to the organizing committee for organizing this event and for also inviting me to share my results here. So the data that I'm going to present here was mostly generated during my PhD at the Federal University of Rio in Brazil with Dr. Vida Saraiva, who spoke earlier today during this, this seminar. But I'm also sharing some new data that I have generated in the NIH with Dr. David Sachs. So just a little bit of background. So neutrophils are the major population of leukocytes circulating in the blood, and they can and they possess a series of granules, which are involved with neutrophil microbiota response. And I'd like to call attention for one class of granules, which are the eosinophilic granules, which contain myeloperoxidase and neutrophil elastase. And those enzymes will be important later on during the presentation. And to remember that they come from those eosinophilic granules, are also called primary granules. Second. I was trying to use this laser point, but I don't think that this is gonna work. We can see the laser pointer. That's all fine. But there is
Yeah.
if you couldn't remove this.
Yeah, but I cannot do it with the laser pointer. Let me just. Yeah, there you go. How does it look now?
Still the subtitles going on, but I think that's okay if you cannot remove this.
Yeah, I actually have no idea how to remove this thing. Yeah, okay, let me just continue. So neutrophil microcellular response can be subdivided into 4 different categories. Phagocytosis, oxidative burst, the generation of reactive oxygen species, either from oxygen or nitric oxide orange. Degranulation, the release of those granules to the extracellular space. And one of the fairly recent mechanism, it's also the release of nets. So nets are web-like structures composed of chromatin decorated with cytoplasmic granular proteins. And they have been initially described by the group of Abertura as a slow cell death process that happens in vitro, that it's characterized by a series of morphological changes in the neutrophil cellular architecture. And it starts with chromatin condensation and evolves to the loss of the integrity of the nuclear membrane, allowing the chromatin to get mixed with the cytoplasmic content of those cells until finally this structure is extruded to the extracellular space. But recent observation done by the group of Paul Cubes actually has demonstrated that in vivo, in a model of Staphylococcus aureus infection, that this process happens much faster, and neutrophils, they remain viable as they release those nets, and they can even respond to chemotatic stimuli, even as cytoplasts, as you can see here on this video. And in vivo, this process, which has been redesignated as vitonetosis, has a very characteristic staining with the cell membrane of the neutrophil involved, like kind of shield with this DNA staining. So leishmaniasis is a disease endemic worldwide in 98 different countries. It's believed that over 1 billion people are at risk to contract the disease, and is a neglected disease associated with poverty and poor housing and weakened immune system, and all of those factors. Also malnutrition leads to increased susceptibility to infection. So the cycle starts with a sandfly taking a blood meal, an infected sandfly, and then the parasite, and then the vector will regurgitate a metacyclic form of the parasite into the host skin. And they will elicit inflammatory infiltration, including neutrophils and monocytes, as nicely described by Nathan. And that will lead to and then disease will evolve and then lead to a series of symptoms that will depend on the species of the leishmania, which can include cutaneous leishmaniasis, which will lead to symptoms and local lesions, or mucocutaneous of visual leishmaniasis, as are you very aware of it. So susceptibility to disease, to leishmaniasis, has been largely attributed to a balance between interferon gamma and IL-10 production, as very well shown by Yasmin in the early 2000s. But there's a recent body of evidence showing that neutrophils actually play a major role in increasing host susceptibility to infection. Data on neutrophil depletion has shown that it decreases it increases actually parasite load. So depletion of neutrophils is involved with parasite clearance. It also affects antigen presentation, since neutrophils can suppress an adaptive response and also affect proliferation of T cells and production of interferon gamma by both CD8 and CD4 T cells. So, but the mechanism and the role of the neutrophil effector response is not very well understood in this scenario. So to investigate the importance of neutrophil microbicidal response modulating this immunity against the parasite, we took advantage very well described experimental models of infection with Leishmania major using B6 and BALB/C strains. So we started by evaluating the production of neutrophil extracellular traps from bone marrow-derived neutrophils incubated with increasing concentrations of Leishmania major. And we compared those strains. What we see is that the susceptible strain, and I'm going to name BALB/C as susceptible strain, B6 as resistant strain throughout the presentation. Shows that neutrophils from a susceptible strain have a much higher capacity to produce nets at higher amounts and also faster, as you can see in this kinetics with cytoxin staining. But interestingly, when we prime those neutrophils in the peritoneal cavity with casein, and then we re-stimulate those neutrophils ex vivo with Leishmania major, what we see is that priming of those neutrophils in an inflammatory scenario, the disparity, the response between those strains, it's way more robust. So we see that inflammatory priming leads to dose-dependent increase in the amount of net production from inflammatory neutrophils in the susceptible strain, whereas neutrophils from the resistant strain does not seem to respond very efficiently. We also did a series of other analyses, but for the interest of time, I'm cutting down short a little bit on some of the results. And we also seen that nets from resistant mice, actually, they have significantly higher, lower, amount of elastase decorating those structures, as you can see here in this fluorescence microscopy. Though they're generating those nets, they seem to have not as much elastase decorated to this structure. So nets are well known to trap and kill many microbes, including Leishmania major permastigotes, as you can see here from this paper from Guimarães Costa. But there is several reports already showing that microbes, they can evade net killing by expression of DNases or nucleotidases, for example. So we compared the capacity of neutrophils from susceptible and resistant mice to kill permastigotes in vitro. And interestingly, although neutrophils from resistant mice, they cannot produce higher significant amounts of nets, they seem to be way more toxic. They can control infection way more efficiently than neutrophils from BALB/C. So interesting to understand a little bit of how this mechanism was done. So we used DNase to digest those nets in vitro, or we just blocked phagocytosis by using cytokinase D. And what we have observed is that DNase doesn't have significant effect on parasite survival, whereas if you block phagocytosis in the neutrophils from the resistant strain, you significantly increase about 50% the amount of parasites that survive in this culture. So demonstrating that phagocytosis, it's way more efficient at controlling infection than the production of gnats. And accordingly, when we compare the capacity of neutrophils from those strains to internalize parasites, to gobble up those promastigotes, what we see is that neutrophils from susceptible animals, they have a significantly lower capacity to internalize those parasites, whereas neutrophils from the resistant strain, they have significantly higher potential to internalize neutrophils. And that phenotype, it's also found in vivo. So we label those parasites with PKH26. And then after intradermal injection, we evaluated the amount of neutrophils that had internalized those parasites 3 and 6 hours after. I'm showing here just one of the data. And you can see that neutrophils from the resistant strain have a significantly higher capacity to internalize those metacyclic promastigotes. So those observations with regards to the gnat formation phenotype were done in Brazil. And so there is a lot of discussion in the literature stating that there's some trouble reproducing data generated from multiple labs because there is, differences in animal husbandry, which might affect the microbiota of those animals, which is well known to affect the immune response. And in fact, there is several papers already demonstrated that the microbiota can affect not just the immune system in general, but also specifically affect neutrophil effector response. Neutrophil aging, production of L17, which is critical for neutrophil priming and infiltration, and neutrophil survival in several models. So therefore, we decided to evaluate if we could reproduce this phenotype in a different setup. So we started a collaboration with Dr. David Sachs to try to reproduce this data there and also enrich our results generated in Brazil. So, and indeed, we were able to reproduce the same phenotype that we observed in Brazil during my PhD. Neutrophils, so this time we use highly purified neutrophils by negative selection. And what we see is that neutrophils from the susceptible strain, again, has a significantly higher capacity to generate gnats, whereas neutrophils from B6, they remain unresponsive. So this is not a phenotype specifically from our animal facility in Brazil. It's reproducible. Gnat formation, it's also associated with histone H3 citrullination, here shown in this plot, and can be used as a marker to distinguish random loss of cell permeability due to necrosis. So therefore, we use this marker to selectively identify neutrophils undergoing netosis. So we incubated those neutrophils with leishmania major, and then we compare the levels of netosis and necrosis and in neutrophils from those strains. And as you can see, in agreement with our net DNA data from the supernatant, neutrophils from the susceptible strain, they increase the amount of histone citrullination in response to infection, whereas there's not much difference in neutrophils from the resistant strain, whereas there's kind of a skew towards necrosis in this resistant strain. So this mechanism, it's also netosis, it's also dependent on the nuclear translocation of elastase and mieloperoxidase, which will cleave histone residues and allow chromatin to unfold. And this process can be visualized as early as 60 minutes upon activation with PMA, a well-known positive control for this process. Both PMA and Immune-Leish. So we decided to compare if there's any inherent differences in the activation profile of those neutrophils, which would justify that somehow the environment, the immunological environment in susceptible and resistant animals drives a distinct differentiation status on those neutrophils that makes them more prone for phagocytosis or netosis. So we started by comparing we started actually just evaluating whether PMA immunomycin activation will lead to nuclear translocation of MPO on activated neutrophils in a biopsy strain. So this is positive control, and as you can see, we observed a significant local translocation of MPO as early as 15 minutes upon activation with PMA immunomycin. And we confirmed the nuclear translocation of MPO in those activated neutrophils. And that reached its peak by 30 minutes after activation. So when we compare the subcellular distribution of MPO in both strains, what we see is that, interestingly, there is a depletion in the amount of MPO in the cytoplasmic fraction of the susceptible animals, and not much difference in the levels of the cytoplasmic MPO in the resistant mouse strain. Whereas when you take a look at the nuclear fraction, what you see is that though there is a transient decrease as well in the amount of MPO in the nuclear fraction of both strains, at 60 minutes, you see a significant amount of MPO getting translocated to the nuclear fraction in the susceptible strain, whereas that's not much MPO, there's significantly less MPO getting translocated to the nuclear fraction of the resistant strain. And also I'd like to point it out that it seems that at steady state, there is already a higher amount of MPO present in the nuclear fraction of those neutrophils from the susceptible strain, indicating that somehow those neutrophils, they exit the bone marrow. Those are data from bone marrow derived neutrophils with already more prone to produce those nets. At this stage of the activation, we didn't see much differences in the levels of double-stranded DNA being released to the cytoplasmic, to the extracellular space, which is in agreement with the literature data, showing that this is it's a process that takes several stages. You're not just, like random, like cell death. When we evaluated so we confirmed this data by flow cytometry. We evaluated the amount of primary granules that are being translocated to the extracellular space. So to do that, we evaluated the expression of CD63 a marker for azulophytic granules, and we activated those neutrophils with increasing concentrated leishmania major. And as you can see here, the cell surface, the cell membrane of neutrophils from the susceptible strain, it's highly decorated with CD63, indicating that those cells are degranulating. There is a great mobilization of those granules on activated neutrophils from susceptible strain, whereas there is not much mobilization of those granules to the cell membrane of neutrophils from resistant strain. So in order to visualize in vivo NET formation on biopsy mice, what we did was to generate an antibody to label neutrophils that contains only the FAB portion. So, and why we decided to do this? So we decided to do this because if you just like label neutrophils with the whole antibody, right, with the whole IgG, you cannot guarantee that those cells are dying because of NETosis or whatever other mechanism, or if they are being subjected to antibody-dependent cell death, right? Since the FC portion is there, you know, like calling a bunch of, you know, like phagocytes to kill that opsonized molecule, right? Ultimately, so so I chopped off the FC region of the LY6G antibody by papain digestion, and then I purified this FAB fragment with protein G purification that generated a nicely pure 50 kilodaltons fragment. We took this fragment and then by flow cytometry, I compared I confirmed that it's staining neutrophils. So I took total splenocytes, and it's and I compared the staining of the FAB antibody that I generated and the regular whole IgG, and the staining, the binding nicely correlates and shows also specificity, because the amount of neutrophils, the percentage of neutrophils that both antibodies identify, ranging from 1 to 5%. It's the percentage of neutrophils normally found on the spleen. So we also did a titration of this antibody, showing that it's not just specific, but it is high. There is high. It has a dose-dependent effect, also binding neutrophils. So next we move to the actual experiment, and then here in pink you see neutrophils, in red Leishmania major or FP parasites, in blue staining for the endothelium, and we use cytoxin to visualize those nets in vivo. And what we see is that we will be able to observe pockets of net formation within the swarm of neutrophil infiltration in the skin following intradermal injection of Leishmania major. And interesting, what we see is that those neutrophils that have, they are producing nets, right? They're staining here for cytoxin, which they actually also have a characteristic staining for vital NETosis. Most of them are not carrying any parasites. Most of the infected cells, as you can see, are located in an opposite region, demonstrated that there is subpopulations of neutrophils that are more prone to release those nets, whereas others subpopulations are actually at this stage, most of these cells, they are infected. It's a mix of neutrophils and TRMs, T2 resident macrophages, so I cannot guarantee that those are only neutrophils, but there is a clear, separation between the cells that are producing gnats from the cells that they have internalized or infected. Those were data done 2 hours post-infection. We extended the imaging up to 24 hours, and we still see some pockets of gnat formation in the skin, but this time it's with much lower frequency than during the acute neutrophil infiltration. And at this stage, we also quantified. So the intravital imaging was data done with biopsy mice. So when we compare those strains, we performed a Western blot to detect H3 citrulination. What we see is that during the acute phase of the inflammatory reaction up to 24 hours, what we see is that there is significantly higher amount of histone citrulination in the biopsy strain, whereas we did not detect any citrulination of histones in the B6 strain, confirming that in vivo, this phenotype of massive production of gnats in the susceptible strain, it's preserved. So we also evaluated gnat formation in the context of Leishmania amazonensis infection. And interestingly, although it's not very well studied, but we observe a similar pattern with regards to Leishmania amazonensis with regards to the effector response of those neutrophils in Leishmania amazonensis model. So here in the B6, you see a massive swarming of neutrophils in response to amazonensis infection, but there is not much gnat formation in this mouse strain. Whereas if you infect those biopsy animals with what we see again, it's just massive formation of gnats surrounding this swarm of neutrophils, indicating indeed that there is some sort of this distinction that leads to that favors gnat formation in this susceptible background that might be associated with higher susceptibility to infection as well. So we moved to evaluate actually, okay, so what is the contribution of that formation to the final pathology, right? So to do that, we infected both strains with Leishmania major, and we gave them a series of injections with DNase IV in order to disrupt gnat formation accordingly to the continuous waves of neutrophil infiltration. So we observe a dual role for gnat formation. A dual role of gnatosis in regards to pathology in those strains. If you compare here in the blue line, the animals that received DNase treatment in vivo, what you see is that there is a transient increase in susceptibility in those animals. Whereas if you treat the B6 strain with DNase, what you see is that they become more tolerant to infection, right? They become even more resistant. But both effects were transient, and they kind of merged again during the chronic phase of infection, which might be due to the fact that we did not digested those gnats during the second wave of neutrophil infiltration. And actually, there has been a lot of data showing that neutrophils they continuously like migrate towards those lesions, specifically in the susceptible strains. So it's difficult to account for that. We will have to just like keep giving those animals DNAs constantly. That's obviously not ideal scenario. At the final time point, we evaluated lesions and parasite load, and we didn't see much difference in the parasite loads in the lesion. But very interestingly, what we see is that in both strains, if you treat those animals with DNAs, there is a massive amount of parasites that can migrate to the draining lymph nodes in order 500-fold increase or even 1,000-fold in certain animals, and this is in both strains, which indicates that although NETs does not affect permanently but transiently pathology, they have a very important impact on physically of creating a physical constraint for parasite dissemination, right? So that seems to be critical to control even leishmania major, which is the least sterilizing strain dissemination to draining lymph nodes. So finally, we evaluated whether those differences between those strains would be associated to any, expression distinction in genes involved with neutrophil microbe pseudo-response. So to do that, we analyzed the whole mouse skin transcriptome of naive animals. We also analyzed the whole skin transcriptome of infected ears 4 weeks post-infection. I don't have the data here now. It's not, the analysis is not finalized. But what we see is that there is much higher expression of genes involved with phagocytosis in the resistant strain, so indicating that it is inherited difference in the probably in the skin environment that favors phagocytosis in neutrophils from NB6, whereas there's significantly higher expression of MPO and other genes involved with netosis in the BALC strain. And that's also associated with expression of a more robust expression of genes involved with general aspects of immunity, such as T-cell activation, cytokine production, and so on and so forth in the resistant strain, whereas the same genes are significantly suppressed in susceptible animals. But conversely, what we see is that there is a high expression of certain genes involved with metabolism in the BALC skin transcriptome. And it's been well described that netosis, it's also associated with a shift in the metabolic response of those neutrophils, so that might also be associated with that. So with that said, I would like to thank the several people that were invited this research throughout those years, especially mentor Vito Saraiva, Heitor, who participated in the intravital imaging, Felipe Lima, my student during my PhD, who helped me a lot with many of those experiments, David for accepting to do this collaboration for those few months, and everyone from his lab, the biological imaging facility here in the NIH, who also supported all of those imaging experiments, and funding agencies and many other collaborators that were involved. Marco Preti also, that was involved in the transcriptomic analysis. Thank you.
So thank you very much, Thiago. Excellent. So good to see we are keeping on the right time. So I'm gonna move straight to the next speaker. Is gonna be David Musser. Thank you again, David, for accepting the invitation. As you wish, you may start sharing your presentation.
Okay, can everyone see my screen?
Yes, we can see.
Thanks, Dario. Okay, so first, thanks to Daniel for inviting me to participate in this presentation. I thought what I would do in light of a summary is to start with some old unpublished data, and then to seamlessly transition into even older data that will never be published. And then finally, once Daniel has figured out that he'll never invite me to Brazil to talk again, then I'll go into some slightly newer data where we'll see if we can tie this all together. And I think the main point that I'd like to make was a point made earlier today by Paul, and then earlier in this session by Nathan, that the in vitro observations that we make can be very informative, but often the in vivo observations are much more complex and complicated, and especially when it comes to macrophages, because every time you change the environment that a macrophage arrives in, that macrophage will change its physiology in accordance to that environment, and therefore it will behave differently when it comes to things like host defense. So the first thing I'm going to do is talk about toll-like receptors in leishmaniasis. And there have been some very important papers on the role of toll-like receptors in leishmaniasis, and the best ones have shown an increased susceptibility to mice that were lacking toll-like receptors or toll-like receptor signaling. And so we wanted to look directly at toll-like receptors on macrophages and Leishmania interactions. And I'll say that we did this at a time when toll-like receptors were in their heyday, quite a long time ago now. We weren't really sure of some of the data that we were going to present. So we did a lot of redundant assays. For the first part of the talk, some of the things you're going to see are repetitious, and the results are all the same. But what we decided to do was compare two different organisms with regard to toll-like receptor activation. One, of course, Leishmania, and the other, Rhodococcus equi. These are both intracellular organisms that spend the majority, if not all of their lifetimes in macrophages. But the one conspicuous difference between these two organisms, of course, is that Rhodococcus equi is a bacterium, so it's prokaryotic, whereas Leishmania, of course, is a eukaryote. And so the first thing we did was we infected macrophages with either Rhodococcus equi or Leishmania, and then look for NF-kappa B translocation. You all know, of course, that downstream of toll-like receptor activation, you get nuclear translocation to drive gene transcription. And for all of these studies, our positive control is lipopolysaccharide stimulation of these macrophages. And these are all human macrophages for the TLR. I'm sorry, these are all murine macrophages for the TLR activation that I'm going to show you now. And you can see that following LPS stimulation, you get good translocation of two of the REL family members, CREL and NF-kappa B P65. Similarly, when we infect macrophages with Rhodococcus equi, we get good nuclear translocation. But we don't see that nuclear translocation when we infect with L. amazonensis. This is immunofluorescence. where we're labeling one of the NF-kappa B family members, P65, in green. And you can see on untreated cells shown by these arrows here, the NF-kappa B is sequestered in the cytosol, as you know, by I-kappa B. And the nuclei are dark because these cells have been counterstained in red here. If we stimulate with LPS or infect with Rhodococcus equi, now essentially all the NF-kappa B rushes into the nucleus. The cytoplasm is now red and the nucleus is bright green. But if we infect with Leishmania, and even after a heavy infection, you can see that these nuclei remain dark red. They are not translocating NF-kappa B during that infection. We used a NF-kappa B dependent luciferase reporter, not a very good one, by the way. Nevertheless, when we stimulated these raw 264 reporter cells with LPS, we saw some luciferase activity, similarly with Rhodococcus equi, but we didn't see any increase in luciferase when we infected with Leishmania promastigotes, amazonensis. This is TNF alpha secretion from unprimed and gamma primed macrophages measured by ELISA. And in a dose-dependent response, you can see an increase in TNF in response to increasing amounts of live R. equi infection, but live L. amazonensis or heat-killed amazonensis failed to induce TNF alpha production from these cells. I will say over here, these are some of the positive controls, and we used a Rhodococcus equi acetone precipitated extract, which contained VAP-A. VAP-A is the virulence-associated protein on the surface of Rhodococcus equi, and that also induced TNF release. So then we looked at a TNF promoter activity, again in raw 264 cells, and once again on primed and unprimed macrophages. And we stimulate with LPS, we get good luciferase activity. Similarly, when we infect with Rhodococcus equi, we get good TNF promoted luciferase activity in primed and unprimed. But L. amazonensis failed to give us any detectable TNF promoter activity. Same thing with interleukin 12. Once again, in primed and unprimed macrophages. With Rhodococcus, we get a dose-dependent increase in IL-12 P40 release in both primed and unprimed cells, but we see essentially no IL-12 secretion in response to L. amazonensis or L. major infection. This is the IL-12 P40 promoter analysis, and once again, LPS and Rhodococcus equi give us good luciferase activity driven by the IL-12 P40 promoter, but L. amazonensis does not. This is IL-12 production by flow cytometry using an intracellular cytokine stain in red. And you can see, of course, that LPS treated cells and R. equi infected cells accumulate IL-12 in the cytosol, but L. major infected cells do not, no different from untreated cells. And these are the parasites stained in green just to show that these cells were in fact infected with Leishmania major. This is the production of nitric oxide by macrophages, and the story is the same. No nitric oxide by L. amazonensis or L. major in primed or unprimed macrophages, but good NO production in response to R. equi infection. And then finally, we looked at DC maturation. And this was CD86 and CD40. We see a good upregulation of CD86 and CD40 on dendritic cells that were stimulated with LPS and CpG oligonucleotides, but we don't see that upregulation on dendritic cells that were exposed to Leishmania amazonensis. So to conclude these early studies, we think that Leishmania enter macrophages via a quiescent mechanism, which fails to directly induce innate immune responses required for activation of macrophages and the induction of adaptive immunity. Now, of course, we know that this parasite activates innate immunity, and in fact, all the TLR knockout signaling studies are consistent with this parasite activating innate immunity. But we think That's due to the indirect activation of innate immunity by things like host cell death, the activation of the inflammasome, the migration of inflammatory cells, all of which we heard about in today's session. Now, I will give you a couple of caveats here, and that is that we didn't analyze L. brasiliensis, and that really was an oversight way back then. We didn't know we would be studying L. brasiliensis later, but you'll see later on in the talk that it really would have been good to have L. brasiliensis included in that analysis, because they seem to be much more immunogenic than the other parasites that we've had experience with. Then the other caveat, of course, is that the parasites that we were working with did not have any of the endosymbiotic LRV1 virus, which may change the way that this organism interacts with the innate immune response. I will say that we are a macrophage lab, so we're very careful about LPS contamination, and we virtually never use glassware that's contaminated with LPS. In fact, we almost never use glassware, we use all plasticware. And so we're confident that there was no LPS contaminating our preparations, and that may have been some of the confounding variables with some of the other data in this field. So now I'm going to go to some even older data that will never see the light of date of publication, and I think you'll be able to understand why when I show it to you. The first thing we did was we took that major virulence-associated protein off the surface of Rhodococcus equi called VAP-A, and we took that gene and transfected it into Leishmania amazonensis and Leishmania major. And you can see that these parasites express this VAP-A on their surface. I have to confess that these parasites don't look particularly happy, and the expression was in fact fairly variable. Nevertheless, we were able to use these VAP-A transfected parasites to infect murine macrophages. And when we measured TNF-alpha, we were able to see that the VAP-A transfected Leishmania major induced the production of TNF-alpha, albeit not as much as LPS does here as our positive control. But L. amazonensis was even a little bit better at inducing TNF-alpha, whereas the control parasites, once again, essentially failed to induce TNF-alpha. We also looked at nitric oxide production by these cells, and we saw a small amount of nitric oxide being produced by L major expressing VAP-A and L. amazonensis expressing VAP-A, a little bit more at 48 hours, surprisingly, than at 24 hours, which seems to be a long time point to us. And then we looked at the virulence of these VAP-A expressing parasites. This is in yellow is the VAP-A expressing L. amazonensis, and we put this into susceptible valve C mice. And what you see is that these mice got essentially no lesions with virtually no parasites in them. There's a caveat here because I told you we didn't think these parasites were all that healthy. These are the vector transfected parasites. They give large lesions with high numbers of parasites in them. We made the mistake of doing an additional control, which was 10 times more Leishmania amazonensis VAP-A parasites, and we infected the cells with the mice with 10 times more. And now in green, you can see that they do in fact get detectable lesions with a significant number of parasites in those lesions. So, you know, this was our first glance at whether the failure to activate the innate immune response would influence the pathogenicity of these parasites. We wanted to try a few other approaches, which I'm going to show you now, and all of them have their problems. Let me say straight up front. These were L929 cells that we transfected with GP63, the major surface protease on Leishmania. And you can see that these L cells express high levels of GP63. And then one set of L cells also was transfected with CD40 ligand. So we have CD40 ligand GP63 expressing L cells and GP63 alone expressing L cells. And then we vaccinated mice with these constructs, with these L929 constructs, and then we went back and infected them with Leishmania major and looked at lesion production. These are lesions in mice that got CD40 ligand with GP63 on its surface, fairly small. These are the ligands that got GP63 alone, and you can see the lesions, I'm sorry, not the ligands, the lesions are somewhat larger with larger numbers of parasites in them. I will say, however, that these lesion sizes are actually not that large anyway, 2 millimeters total. You can see conspicuously that we don't have a control here, which is non-vaccinated mice, to look for the relative protection of either of these two constructs. But we, undaunted, we proceeded and tried what was technically a little bit of an easier approach. We took soluble Leishmania antigen and we vaccinated mice in the presence of CD40 ligand transfected cells, or in the absence of these CD40 ligand transfected cells in gray. And once again, you can see that the parasites that were, the mice that were vaccinated with CD40 ligand and soluble Leishmania antigen had smaller lesions with lower numbers of parasites in them, whereas the mice that were immunized with soluble Leishmania antigen alone here in gray had large lesions with similar numbers of parasites to the control. That suggested to us that the innate immune response plays some role in this, that TLRs are important, although their effect is indirect, not a direct role on the parasite, but they play an important role in leishmaniasis. We think that Leishmania avoids TLR activation in order to establish successfully the infection, and that if you force toll-like receptor activation on these cells, as we show here in green, that they will become less virulent. And I think this has implications for vaccine development, et cetera. So those are our initial observations on TLR activation by these parasites. Now I'm going to go into some slightly newer data. And this is RNA sequencing to look at the transcriptome of L major and L amazonensis infected macrophages. And now these all, from here on in, we're talking about human macrophages. And the first thing I would like to do is call your attention to macrophages that were stimulated with LPS, these so-called M1 macrophages. And these are some of the most highly upregulated genes by these M1 macrophages. And these have been well described by us and by others. The inflammatory chemokines, CXCL9, 10, and 11. And this is the fold upregulation, by the way. They're hugely upregulated. Inflammatory cytokines, IL-6 and IL-12. Indolamine dioxygenase, which we heard about from Paul Kay this morning when he was talking about his Sri Lankan leishmaniasis. IRG1, and a few of the others, IL-1 and TNF and IL-12, et cetera. So these genes make essentially a signature for an M1 macrophage. And when we look at the genes that are induced in macrophages following L major infection or L amazonensis infection, you can see that essentially none of these genes are represented. There are no inflammatory chemokines, CXCL 10, 11, or 9. There's a little bit of IL-6 that's being made, but that amount of IL-6 is even less than what's made in response to infection by a bead, an inert bead. Same with L amazonensis. So really, none of these genes none of the genes that are induced following LPS stimulation, none of these so-called M1 signature genes are induced by Leishmania major. And of course, the Spearman correlation is a very low 0.25. There is one gene, by the way, that's commonly induced by all of these, LPS as well as L major and amazonensis infection, and that's IRG1. And we now have come to realize that IRG1 is a potent immunoregulatory gene. It actually can dampen cytokine production and inhibit inflammasome activation. And that's the one gene that's induced by all three of these stimulation conditions. Now, these are the genes that are actually induced in macrophages following L major or L amazonensis infection. And the gene names aren't very important. They're listed from 1 to 15, and this is the rank 1 through 15, and this is the fold induction. And you can see they're induced by the 100-folds, no longer by the 1,000-folds like we saw with LPS. Nevertheless, I think the point I want to make here is showing you that the genes induced by Leishmania major, the top 15 genes, are very similar to the top 15 genes that are induced following L amazonensis infection. In fact, 11 of the top 15 genes induced by L amazonensis are among the top 15 induced by L major. And the Spearman correlation is a blockbuster 0.78. So these two parasites induce a similar transcriptional program in macrophages. Of course, they are clinically distinct, these two parasites, but we think that that distinction is not because of a difference in what they're doing, at least directly to the macrophage. The other thing I will point out here is that these genes are induced by Leishmania infection are quite distinct from the genes induced by LPS. This is the ranking of these genes by LPS, and some of them are not even induced at all. Okay, now I'm going to shift gears a little bit and talk about some other studies that we've done on RNA sequencing of normal human macrophages. And on upper left, I'm showing you the top 10 genes that are induced by LPS, and we already talked about those genes. Several years ago, we identified what we called a regulatory macrophage, an immunoregulatory anti-inflammatory macrophage. And one of the ways we thought these macrophages were induced is by increasing the strength of signal. And so in this particular case, in this study, we stimulated the macrophages with LPS, and that's shown in red, but we increased the strength of signaling by stimulating them with LPS in the presence of immune complexes to ligate the Fc gamma receptors. And paradoxically, this turns these macrophages into an anti-inflammatory and regulatory macrophage. So let me just go through this data for you here. So these are the top genes induced by LPS in red, as I showed you: CXCL9, 10, and 11, IL-6, IL-12, endolimine deoxynase, RG1, et cetera. If we increase the signaling strength by adding immune complexes to this, now paradoxically, we turn off many of these potent inflammatory genes shown in blue here. Not all of them. IRG1, for example, is not turned off. But many of these inflammatory genes are turned off. If you look at the volcano plot, actually, most of the genes that are induced in macrophages following LPS stimulation are not changed. They're all the ones here in this massive yellow. There are just 632 genes that are downregulated and 925 genes that are upregulated by the addition of immune complexes. And if we look at these genes that are top upregulated, they're shown in this box here, and they include genes like matrix metalloprotease 10, matrix metalloprotease 1, matrix metalloprotease 3, activin A, IL-10, DC stamp, amphiregulin, OC stamp. Many of these genes that are upregulated by the addition of these immune complexes are genes that are involved in cell growth and repair. And that's why we call these cells regulatory macrophages, because we think they're involved during the resolution of an immune response and during wound healing responses. And we think these macrophages are very important in wound healing. And if you don't allow these macrophages to transition to this phase, we think you have problems with wound healing and problems with inflammation. Now, what I'm going to do is take this box that I just showed you and copy it into the next slide. And here's that box. And now here's a slide that I showed you 2 slides ago of the genes that are induced following Leishmania infection in macrophages. And what you can see, and you can judge for yourself whether this gene list looks anything like this gene list, although I have to admit that I'm helping with your judgment by adding all these arrows here. You can see this first most highly upregulated gene by Leishmania is also the most highly upregulated in immune complexes. Here we see CSF2, here's CSF2, OC stamp, activin A, MMP10, MMP10. So this clearly isn't, is just sort of a snapshot of the top 15 genes that are induced by Leishmania. And these genes are not only the genes that are induced by LPS plus immune complexes, but these are the genes that are most highly upregulated relative to LPS. So that a direct comparison is impossible. But what it suggests to us is that perhaps the Leishmania infection is priming macrophages to undergo or to start a similar regulatory transcriptional program, similar to the transcriptional program that I showed you on the previous slide, following the addition of immune complexes. So now I'm going to go to the much more complicated in vivo situation. And what I'm going to do is compare and contrast localized cutaneous leishmaniasis, which we did in collaboration with Edgar Carvalho and Phil Scott, and diffuse cutaneous leishmaniasis, which we did in collaboration with Fernando Silveira. And what we did was we took biopsies from these lesions and performed RNA sequencing on these biopsies. Now, one of the advantages of RNA sequencing is that you can map the reads to either the host genome or to the parasite genome. And when we mapped the genes in localized cutaneous leishmaniasis biopsies, we were surprised to see that most of the genes mapped to the human genome. There were very few genes that mapped to the parasite. In fact, less than 2% of the genes of the reads mapped to the parasite. So even though these can be large necrotic lesions, even though these lesions can persist to months to even years, there are very few parasite transcripts in these lesions. In contrast, in diffuse cutaneous leishmaniasis, these lesions are loaded with parasites. 10 to 28% of the reads map to the parasite. So these lesions are failing to control parasite growth, whereas these lesions are almost eliminating parasite growth. The next surprise came when we examined the human genes that were upregulated during diffuse cutaneous leishmaniasis. And that's shown here. These are the top 40 genes that are upregulated in these lesions. And this is the log fold change. You can see how substantially they're upregulated, over 1,000-fold. And 38 of these 40 genes encoded immunoglobulin gene segments. And the other two, shown in red, encoded B-cell genes. So we have always thought that leishmaniasis was a macrophage disease, obviously from our bias, but you can see that there are large numbers of B cells in these diffuse cutaneous leishmaniasis lesions. These are the first 10 genes from here, shown here, and I just amplified this so that you could see each of the individual patients, shown by each of the individual symbols here, and you can see that all 6 of these patients with diffuse cutaneous leishmaniasis, and I should say that we only had 6 patients. This is a very rare disease, but all 6 of these patients make a similarly high immunoglobulin response. And even more surprising to us was that the immunoglobulin that we identified was primarily IgG4 heavy chain. And that's surprising because if you look at the normal distribution of human immunoglobulin, it's dominated by IgG1 and IgG2, with IgG4 being a very small component of it. But in this disease, IgG4 seems to predominate. We think the immunoglobulin response in these patients is an oligoclonal rather than a polyclonal response. And the reason we think that is because we detected a limited number of J gene segments combining with a limited number of variable gene segments where all these others are not represented. And so this limited diversity suggests to us that it is an oligoclonal response, but we don't know the antigen that these hosts are responding to. So I'm going to take a little detour and look at the T cell responses in this before I get back to macrophages. And the first thing you can see was this high evidence for transcripts involved in cytotoxicity. in localized cutaneous leishmaniasis relative to diffuse cutaneous leishmaniasis. In localized cutaneous leishmaniasis, you have high levels of perforin, high levels of granzyme B, high levels of granzyme A, granulysin, and even CD8 transcripts as well. That was not a surprise to us because that had been reported by others in this field. In fact, others who are going to be speaking at this meeting. And when you look at inflammatory cytokines in the localized disease caused by L. brasiliensis, we see high levels of interferon gamma, high levels of TNF, high levels of IL-1. And in fact, you see a strong induction of transcripts encoding the Th1 transcription factor, master transcription factor TBET. So the localized disease is a pro-inflammatory disease characterized by inflammatory immunopathology that causes these lesions to persist for months at a time. And we think the cytotoxicity and these inflammatory cytokines are what are contributing to this disease. Now, then we move to the diffuse cutaneous leishmaniasis, and I have to say that it was a bit of a mystery to us. We assumed that we were going to see high levels of IL-4, IL-13, IL-5, and simply stated, we were unable to detect any IL-4, any transcripts for IL-4, 5, or IL-13. In fact, when we looked for GATA3, the master transcription factor, it wasn't induced at all in diffuse cutaneous leishmaniasis. This was surprising to us because others had suggested that this diffuse cutaneous leishmaniasis was a Th2 disease. We weren't able to show evidence for this. And in fact, when we looked at what we think are so-called canonical M2A responses by macrophages, and let me start by saying that there are excellent biomarkers for M2 macrophages, FIS1, YM1, mannose receptor, you name it. In the murine literature, there are excellent biomarkers for M2 macrophages, these so-called IL-4 treated macrophages. But in the human literature, there's a lot of debate as to what the best M2 macrophage markers are. We took markers from the literature that other people had used, and if you look at diffuse cutaneous leishmaniasis in red, there's no induction of any of these canonical or suspected M2 biomarkers. So we were unable to identify a Th2 or an M2 signature in diffuse cutaneous leishmaniasis. I have to say that we only had 6 patients, and these patients were all analyzed at one time. In most cases, it was substantially later after they had had the disease for quite some time. So we may have missed the Th2 response, but we were unable to convince ourselves that this disease is a Th2 dependent response. And maybe that's consistent with what we just heard from Nathan about his inability to report on Th2 in his murine models. But we did gain some, we think, important information when we looked carefully at the macrophage biomarkers in diffuse cutaneous leishmaniasis, and we compared them to the biomarkers in localized cutaneous leishmaniasis. Now here, the diffuse is in red and the localized is in blue. And on the left, we analyzed transcripts for what we thought were pan-macrophage markers, transcripts that should be expressed by all macrophages in the lesion. And these included the Fc receptors that are on macrophages, the complement receptors, CD11, CD11b, CD18, CD68, a well-described macrophage marker. And you can see that there's really no significant difference between the amount of transcripts for any of these pan-macrophage markers. So even though these lesions look very different, even though these lesions have very different numbers of parasites in them, our interpretation of these data on the left are that they had about the same number of macrophages in those lesions. But then we look at the macrophage activation markers. And I hope you can remember some of our canonical M1 macrophage activation markers, because in localized cutaneous leishmaniasis, we see high upregulation of these inflammatory chemokines, CXCL9, CXCL10, CXCL11. We see the upregulation in dolamine dioxygenase, the tryptophan catabolizing enzyme. We see an upregulation of NOS2, consistent with localized cutaneous leishmaniasis being an inflammatory disease and the pathology is being driven by immune pathology. When we look at diffuse cutaneous leishmaniasis, in contrast, now we see an upregulation of these matrix metalloproteinases, DC stamp and Marco. These are all transcripts that we had associated with our so-called regulatory macrophages. There's DC stamp staining by immunohistochemistry. And let me go back and say that we assume that the high level of immunoglobulin and the high number of B cells that we saw in these lesions was driving these regulatory macrophages. And we have some previous data that would be consistent with that. And that's what I'd like to just go back and revisit now for a second. This is localized cutaneous leishmaniasis caused by L. brasiliensis, as I remind you. And I remind you that there were very few parasite transcripts in these lesions. The highest ones, the parasite transcript positive individuals, we rated as having from 0.5% to 2% of the reads mapping to the parasite. The PT negative had no detectable parasite transcripts marking to it. And these PT intermediate, parasite transcript intermediate patients, 9 of them, had low levels of parasite transcripts up until 0.5% of the transcripts mapped to the parasite. And when you look at the immunoglobulin gene expression in these 3 different cohorts of patients that we arbitrarily, admittedly, separated, now you can see that the patients that had the highest level of parasite transcripts, shown in orange, had the highest levels of immunoglobulin gene expression. And so, once again, a direct correlation between parasite transcript number and immunoglobulin gene expression. And finally, I'm going to go back to some really ancient data. I should be a history professor rather than a scientist, going all the way back to 2005 to work that we did in collaboration with Selma Geronimo in Natal, Brazil, where we looked at Leishmania infection in BALB/c mice, and we compared it to Leishmania infections in BALB/c mice that were lacking the heavy chain, the J heavy chain, and so therefore we're deficient in B cells and immunoglobulin. And if I do say so, all these years later, I'm still fascinated by these results. In the solid pink line, this is Leishmania major infection in BALB/c mice. These are susceptible mice, as you all know. They get progressively larger lesions, shown in the solid line. And at the end of the experiment, they have large numbers of parasites in these lesions. In the dashed line, these are BALB/c mice from the same litter that got the same amounts of Leishmania major parasites, but they got anti-Leishmania major IgG at 3 different times post-infection over the first 2 weeks. And what you see is that that anti-Leishmania IgG does nothing. The lesions progress at the same time, at the same level, same kinetics, and at the end of the experiment, these mice have the same number of parasites in their lesions as these mice. So in this case, immune IgG did nothing to help these BALB/c mice. The JH mice was the fascinating story. These mice have no B cells and therefore should be technically immunocompromised. And yet when we infected them, now they got low lesions with small numbers of parasites in them. And then the real surprise came when we added anti-Leishmania IgG to these JH mice. Now, oddly enough, they got large lesions with high numbers of parasites in them. So in the case of these JH mice, the addition of anti-Leishmania IgG actually exacerbated disease. And so we went on to do a study with Selma Geronimo in Brazil. And Selma had many patients, some 300 and something patients with visceral leishmaniasis. 280 of these patients were anti-Leishmania antibody positive. And that meant they were making IgG 2 standard deviations over the mean of endemic controls IgG. And when we looked at these antibody positive patients, 90% of them were delayed type hypersensitivity negative. In contrast, she had 37 patients that were antibody negative. And when we looked at these patients, 84% of these patients were delayed type hypersensitivity positive. And if you look at the in duration, the size of the DTH, the antibody negative patients made large DTH responses. The antibody positive patients essentially made no DTH response because it has to be over 1 millimeter in order to be considered positive. So this is a real example of immune deviation. These patients either make an antibody response or they make a DTH response, and they don't typically make both. And then finally, what Selma did was to go on and follow these patients. At the time of diagnosis, they had high antibody levels shown in pink and low DTH responses. And as she treated these patients, the antibody levels went down and the DTH responses went up. So to conclude, in leishmaniasis, there's an inverse correlation between antibody and cell-mediated immunity. We call that immune deviation. I would suggest that would cause people to have pause when they're thinking about making an IgG vaccine against leishmaniasis, at least against some forms of leishmaniasis. In our hands, in murine models, and in this exaggerated diffuse cutaneous leishmaniasis model, the IgG seems to be more pathogenic than helpful. And then finally, in diffuse cutaneous leishmaniasis, the host makes the wrong immune response. It makes a humoral immune response. And we think those high levels of immunoglobulins promote the development of macrophages with a signature of growth promotion and angiogenesis, but not parasite killing. In contrast, the localized cutaneous leishmaniasis caused by Braziliensis induces a macrophage with a signature of an M1 macrophage, an inflammatory macrophage that results in parasite killing. We see very few transcripts in those lesions, and yet we see immunopathology because those macrophages can't turn themselves off. They can't regulate an immune response. And that's why we think this transition of macrophages from an inflammatory phenotype to this immunoregulatory immune resolution phase is so important. And we also think that that's the main role of macrophages, to maintain homeostasis. And somehow this brilliant parasite has figured out that that's the role of macrophages, to maintain homeostasis. And it has managed to find itself in these regulatory macrophages that promote growth and angiogenesis and fail to promote killing. These are the people in the lab that did the experiments. Here's my lab at the University of Maryland. And I'm happy to take questions from here. Thank you all.
Thank you, David. So we had 3 excellent presentations, and I'm happy to open. We are right on time, a little before the time actually, so we'll have more time to questions and discussions. So
that's because my English is so fast.
No, you guys did a terrible job in keeping the time. It was awesome. So that have a question from Leda to Nathan. Have you looked at TNF production in wild type and STAT6 knockout mouse on day 14 when you see more parasites in the STAT6 minus mice? In fact, what is the expression of TNF in this in the less and more permissive cells?
Yeah, I don't think I'm going to. I know that we looked at TNF both by flow and gene expression, but my knee-jerk reaction is that it followed the gamma fairly closely. But I'm not going to. Are you looking for TNF in the phagocytes, Lena? I think that's what maybe she's referring to. In general, the TNF follows the gamma very closely, but I'm not going to remember off the top of my head the answer to her question.
So Nathan, can I ask you a question about your talk?
Sure. David, you can answer my question.
No, I can't answer that question. I don't know the answer to that. But I loved your talk. I thought it was great. But there was so much data that it's very easy to confuse me, and I got confused. You showed that your monocytes are arriving in that lesion 18 or 20 hours afterwards, just after the neutrophils get in there. But the presumption is that those monocytes are not seeing interferon gamma yet, right? We don't have the interferon gamma that early, correct? And so is it the late monocytes that are responding to the interferon gamma, or do you think it's the residual innate interferon gamma?
So both sets of monocyte-derived cells can respond to the gamma, but the cells that are there longer, the monocyte-derived cells that are there longer, and in the time frames we were looking at, those cells would be the monocytes recruited during the innate interferon gamma independent recruitment phase. They've had time to mature and they become responsive. The ones that are non-responsive are the more recently recruited ones, and during that time frame that we're looking at, those are the ones that are being pulled in very robustly by the Th1 response.
But do you envision any interferon gamma over the first 3 or 4 days of this infection?
Well, yeah, you're going to have you've got innate lymphoid cells, you've got NK cells, there's probably some gamma kicking around.
You think that's enough?
In the context of what you get later on, it's probably pretty minimal. But is it tickling the site? I think so. When we think about the recruitment, there's innate monocyte driven recruitment. There's interferon gamma driven recruitment during that early phase. So gamma isn't the only thing that's going to drive these gamma inducible chemokines to bring in those early monocytes before Th1 immunity kicks in. Those are a lot of, I've pulled my hair out, looking at these sort of chemokine networks, and there's a lot of redundancy. But there is a little bit there in the beginning, but whether that's necessary to induce that innate monocyte recruitment, I would say no. There's other mediators at play, the MCP1, which is also gamma inducible, but can also be induced by other things. Fibroblasts can induce monocyte recruitments and chemokine production, etc.
Hey Nathan, I also have a couple questions.
Sure.
So those inflammatory monocytes, right? They're like immature cells. They will ultimately, once in the inflammatory side, they will differentiate to either macrophages or dendritic cells. So kind of like in the inflammatory side, transiently as inflammatory monocytes, as they receive signals that might drive a certain type of differentiation. So how do you see your this phenotype in macrophage in monocytes as they differentiate towards macrophage, especially during Leishman infection, because they kind of work as just as a transitory site for Leishmania, right? They ultimately gonna turn into macrophages.
That's right. I think the important, if I understand your comment correctly, I think the important thing to consider here is that when you look at 2 or 3 weeks post-infection, when you look at the total phagocyte population, it's very heterogeneous. But I would argue that a lot of those cells are infected when they are immature inflammatory monocytes. And that provides the opportunity for the parasite to make that niche. And then they subsequently mature. Some of them become very permissive host cells. Some of them become a TIP-DC and actually control the parasite. But it's when you look at them at 2 weeks, 3 weeks, and you say, oh, look, the parasite is in this cell, that's not necessarily the phenotype of the cell when that cell was initially infected. And I think what our data shows is that the cells are initially infected when they're that immature phenotype, when you're talking about the monocyte population anyway.
Because STAT6 seems to be regulating this M2 phenotype in the inflammatory monocytes, right? When I was surprised to see that when you knock down STAT6, you see an increase in interferon gamma, but then there's not much effect. And that, I mean, that it's eliciting more inflammatory monocytes in that site, right? So that should increase permissiveness to infection. But at the same time, without STAT6, you don't get that M2 phenotype in those inflammatory monocytes. So how come the parasites can proliferate more, right? Because you're showing in the beginning that STAT6, it's required for this M2 phenotype.
Well, there is this knock-on effect, though, of the STAT6 deficiency, and that is this robust IL-10 response that seems to kick in to control what is now becoming a very highly inflammatory Th1 environment. Mm-hmm. And I think that with all that IL-10 around, you get a sort of a secondary effect. So the killing and the recruitment may balance each other out, but at a single cell level, because in the absence of STAT6, you push things so Th1, it seems like the regulatory response is trying to control that, and the parasite at a single cell level can benefit from that. Those monocyte-derived cells are really, even though it's a predominant Th1 environment, they're still very sensitive, and that's, I think, one of the interesting takeaways. They're still very sensitive to the signaling induced by IL-4 and IL-10.
So we have a question from Herbert. So just raise the hand, and then after Herbert, you can open to Claudia. Good to see you here, Claudinha.
Me too.
And Nathan, congratulations for your talk. Fantastic. I like a lot your coffee espresso machine. Okay. Nathan, I have 2 questions for you. The first one is, if you have, for example, with the same infection, why the lesion progression in interferon gamma knockouts mice and white type. So, and if you wait more time, you can see white type resolving the lesion. It's a partially resistant model because you decrease the lesion, and then it'd be like chronic, and interferon gamma is still going up. So what's happening in this time, and what do you think that's happening that you start to control here, but you don't have any effect in the beginning of infection, but in this time, interferon gamma going up, interferon gamma knockouts going up, and the white types going down. I think we have to look at some regulatory process. So, but the second question is, did you look?
Can we do the first question first, and then?
Sure. Sorry. Yes.
Otherwise, I'll.
Yeah. Sure. Sure.
I have a tendency to lose my train of thought. I think the situation, what you might be seeing is there's enough interferon gamma to induce recruitment, but not enough to control or eliminate parasites. Later on, eventually you get to a point where you're both recruiting the cells that develop a killing capacity and also sufficiently activating them to keep a lid on the infection. And I'll just say we've only followed them out a certain amount of time, and now we're following, in the follow-up studies, we're following them all the way through, and those L. amazonensis infections in the ear of B6 mice are ultimately progressive. It slows, and I think that's probably what you're seeing in our data. Is in the wild type mice, it starts to slow. I just want to clarify that it's ultimately progressive.
I usually infect in the foot pads, and the foot pad we have the resolving. So
yeah, so
using several strains. If you use the foot pad, we saw the growing up the lesion, and then you start to resolve, but it's not complete. It's different from our major infection that's gone down like to zero, but in Leishmania amazonensis infection become chronic and still parasite. I follow more than 1 year and you still see, but it's chronic, you know.
Yeah, we find
pad.
In the foot pad, we find the same, that L. amazonensis, what we call it non-resolving. It goes up, stabilizes, and then just sort of continues on for extended period.
You use a PHH strain, right?
Yeah.
Okay. So, but it's the second question is the same question. Did you look at the interferon gamma receptor expression in your monocytes? Because I remember that in the past, maybe David can say the same, the infected macrophage decreased the expression of interferon gamma receptor. So maybe we have some points in the beginning, the receptors are down, and the presence of interferon gamma didn't have effects. But it's the modulation of the system, maybe you accumulating, how you said, accumulating interferon gamma producing cells, but at the same time, it's having some modulations that macrophages can increase the expression of the receptor. And in this particular time, you know, they start to clear or killing the parasite. So did you look at the expression of the interferon gamma receptor expression during the infection?
Yeah, definitely. And I know that on the total CD11b positive phagocyte population, that data is in the paper, in the supplement. And specifically on monocytes in the later studies where we started looking in the knockouts, I'm not sure. It may have been one of the ones we had to drop because we maxed out the flow cytometer. But the beginnings of that data are definitely in the supplement in the paper, and maybe take a look at that and then shoot me an email and we can discuss further.
Thank you so much.
Hi, Nathan. How are you doing?
I'm doing well. How are you?
I'm fine, thank you. Very nice talk. I'd like to know if you think about the vaccine against leishmania. We know that we need to produce a lot of interferon gamma at the beginning of the infection. So do you think in this case, this more interferon gamma elicit really fast, it's enough to kill the parasites and avoid the recruitment of cells? So I don't know, I think in Leishmania amazonensis is not the case, but I would like to hear your opinion.
Yeah, I think, you ultimately parasite dose will drive a lot of this. So, getting the parasite numbers down to a manageable level, whether or not you get sterilizing immunity or not, I think is good. And that requires, the data we're sort of generating now and have out for review is really showing the importance of a really hard gamma hit, like almost immediately, and how important that really is. So identifying the timing as a correlate of protection, more than all the phenotyping and all the this cell, that cell, this kind of stuff. But how important is time when those monocytes are exposed? Unfortunately, I didn't get to that data. So definitely those monocytes, the gamma isn't recruiting them directly. The gamma is inducing chemokines that recruit them, but then they really need to get a gamma signal almost as soon as they arrive in the site. And then you touched a little bit upon vaccinating against Leishmania amazonensis, and this is some of the follow-up work that we're doing. If you have pre-existing Th1 immunity, it provides beautiful protection against Leishmania major. What does it do against Leishmania amazonensis? And I will tell you that Leishmania amazonensis, the reason why we got into this is because, this amazonensis I view it as sort of a more complicated parasite and more difficult, and certainly in South America, Central and South America, way more relevant. And I can tell you that it's going to be a different bag for sure. Protecting against Leishmania amazonensis is really, really difficult, more than L major. Yeah. So I think we're in for a bit of a wake-up call in terms of just generating a great Th1 vaccine protects against major. We're seeing that amazonensis, it takes a hit, but it keeps on going.
Yeah, but I think this is the point that David also talked about, that is the problem that we have an anti-inflammatory response more than inflammatory response if you have a lot of immunocomplex, for instance. So it's really completely different, no? Even if you think about Leishmania major and Leishmania brasiliensis, for instance, that I think that it's not so bad to control. But anyway.
My comment there is that, these major L. amazonensis comparisons are probably some of the most difficult experiments to do, not because of all this complexity, but, just to get the parasites out of culture and have some sort of equivalence between what you're injecting. Because the metacyclogenesis, FV1 versus PH8, it's completely different, and it's so easy to say, well, I use 10 to the 3 and I use 10 to the 3, and you're not comparing apples to apples. So that's been very difficult. But we think we've gotten quite good at it. But in my sort of current view about what's going on with amazonensis is I think David's data really touched upon these comparisons between major and amazonensis. We just kind of killed ourselves looking for differences early, and I don't think there are differences early. I think it's how those parasites respond to gamma, and that the phagolysosome is biologically or anatomically is completely different. So I think it's later when these other things kick in, maybe like what you're talking about with the immune complexes.
Okay, thank you.
We'll have a question from Paul, the morning speaker. Welcome to the afternoon speaker, Paul.
Hi, Dario. Yeah, it was a question for David, actually. It was fascinating seeing your transcriptional profiles of infection with 2 species of Leishmania, giving this regulatory profile. Do you know whether that requires active infection, or is it a reflection of the signature on the surface of the parasites during the process of initiating phagocytosis?
I'm not sure 100%. What I will say is that those transcriptomes that I was showing you represent a very early response to parasite infection. And we used enough parasites that we thought most of the macrophages were going to be infected. But oddly enough, if you look 24 hours later, the macrophage looks very much like a quiescent macrophage that has never seen anything. The parasites are in there. The macrophage transcriptome has gone back down to baseline, and it's completely boring to us. So we looked at the early response, 4 and 6 hours after infection. I think most of those macrophages that we looked at were infected, actively infected. And by the way, we did have low amounts of complement in there to enhance the infection.
Okay, thanks.
But Paul, since you're here and since you're on, I would love to hear what you're thinking about with the L major human challenge studies that you referred to in the morning. Is it too early for you to talk about that, or can you tell us what you're thinking about?
Well, we've generated a GMP line of L major. That's been through all the safety screens and all the SAMFly differentiation assays, etc. And we're just started. We have a protocol approved by the IRB, and we're just starting to recruit, hopefully for the first volunteer infections early in the fall.
These are deletion mutants or wild?
No, these are wild type. These are wild type, a new strain isolated from Israel.
And you're just going to infect volunteers?
That's correct.
Oh, to be in the UK.
Probably the only way to do it, and to really not only understand this early pathogenesis of development of the lesion, but also to get a robust way of doing selection for candidates.
Yeah, good for you.
Cool. So another question from Leda, now to David. Why do you think primed macrophages did not make NO in response to Leishmania major, contrary to previously shown by Liu, Green, me, Veras, etc., etc.? Is that a cell line?
So so we used primary macrophages, but in fact, for almost all the studies we do, we first do a macrophage cell line just to appease our animal rights people, because they want us to know that we can eliminate the number of mice that we're going to use. But for all those studies that I showed you, those were primary macrophages. And we don't ever see NO production in unprimed macrophages. You know, it's very, very low. You have to gamma prime those macrophages in order to get NO production. And then if we infect with a bacterium, or if we stimulate with LPS, we see good NO production, but we never saw it with those two species of parasites. That's, you know, when we did those studies, it was in the height of the TLR craze. And that's why all that data has been sitting in my drawer for the last 10 years. I figured I could show it at a meeting like this, but that's why we never really published this stuff.
Yeah, cool. So there's another question for you, David. That's from probably, it's hard to say because the name in the YouTube is CC Afonso. So I guess it's Luis Carlos Croco Afonso, right? So that's,
oh, no, I can't answer those questions.
So he asked if you performed a comparison of gene transcription on IC treated macrophages versus Leishmania infected versus adenosine treated macrophages.
Yeah, so I was referring to those regulatory macrophages as, and one of the ways that we generate them is with immune complexes. But they have a very similar phenotype to macrophages that are stimulated in the presence of adenosine or macrophages that are stimulated in the presence of some prostaglandins like prostaglandin E2. And so we have sort of bent over backwards to try to figure out how closely related all these different so-called regulatory macrophages are. And the fact is they share some similarities, but you know, if you're just looking at Pearson correlations or something, it's not great. And so we're reluctant to say that, you know, that these are the same. And in fact, we think that every single macrophage is different from every other macrophage, depending on what, you know, what it sees. So there are some similarities. Both of those cells seem to promote angiogenesis and express some of the wound healing molecules, but it's a little bit of a stretch, and it's probably because I know exactly what genes to look for, and I'm biased about it.
Cool. So I have actually a comment about I thought it was very interesting, Nathan's, I mean, the way he started the presentation with in the absence of interferon gamma, the lesions are all the same until day 21, if I recall, something like this. It's very impressive, right? So there's a lot to discuss, and I think this roundtable events are kind of nice to discuss these things. And leishmaniasis has this, a lot of times we see that. So I wonder if somehow interferon gamma has maybe a detrimental role and a protective role during the first days of infections. And then as the one neutralizes the other one, so we have a similar lesion development and perhaps a similar parasite loads in the tissues. But then eventually, of course, interferon gamma is very important. So you start seeing the lack of interferon for restriction. And I think it's kind of a nice to discuss idea, because I think this happens a lot, not only with interferon gamma, but perhaps with neutrophils and IL-1 beta, whatever, inflammasome. So we at some point, something is very important, but can be good, can be detrimental as the imbalance goes and the infection goes on, and then you start seeing the absence of that determined molecule can lead to more pathology or more parasite replication. So I wonder your comment on this, if there is any possible detrimental role of interferon gamma in these early days of infection, where one may balance the absence of interferon by the protective side or the more pathological side.
Yeah, maybe I'll take a kick at that in the beginning. I think that's exactly the point. And I think one of the strengths of the postdoc who did a lot of this work was I don't know if Matthias learned this from his time in Leda's lab or not, but it's these kinetics, the number of mice that Matthias went through for some experiments, I was like, this is crazy. But the kinetics become so important. And, obviously you can't analyze every day. And there's some happy medium in the middle, but definitely, my view would be that if gamma, the knock-on effect of gamma early is chemokine production, and those chemokines can be very, very efficient at recruitment, but there's not yet enough gamma there to turn on those cells that are coming in, or you're not localized in quite the right place to get the nitric oxide production, then it can be, early on, it's quite detrimental, and the parasite has actually hit upon that. I think it's a great example of parasitism. Yeah.
Yeah.
So Nathan,
go ahead.
I wanted to comment on something you said in your talk, which was that you questioned the value of a hyperpolarized Th1 response, and I think you're right on target as far as that's concerned. We showed that in others, by the way, in Braziliensis, but I also think when you just look at just T cell biology, if you hyperstimulate a T cell, you induce senescence, which we're going to hear about tomorrow. You induce exhaustion, and you lose these polyfunctional T cells. So I think you're right. We don't want to hyperpolarize a Th1 immune response in this disease.
Yeah, no, I think it's potentially a bad idea. And I think if you look into the TB field, I think maybe Paul has a couple of things to say about this as well. In the TB field, they've got, we've got some great Th1 vaccines, like the work that Steve Reed did to generate these great Th1 vaccines they're fantastic. And I'd love to actually go back and do a little bit of a better job in my own work, even to see, we had some evidence that when we boosted with antigen and these adjuvants, that we're like, let's boost them before we challenge to really push things further. We totally overdid it. And Steve, I know that Steve was not very happy about how we did that, and a bit of a regret actually. But you push it too hard, then it's a bad situation, and which is why I really, obviously in the leishmanization camp, but even more so in these centrin knockout parasites that are initiating a sort of more natural infection that perhaps has just better balance. And if you come in with a hyperpolarizing approach, there's going to be overcompensation. IL-10, I think, is a great example of that. It's like, you didn't really think it all the way through. All that gamma around the immune system is going to be like, we can't have this. The skin's going to turn to mush. So it's going to regulate, and then you're back to the drawing board. So, yeah.
So just to add a comment with regards to the leishmanization, we don't think that a robust Th1 immunity is the mechanism behind the protection. It requires further studies, exploration, but we're just showing on the paper that there is robust type 1 immunity, which has been classically associated with protection in the chronic phase, but doesn't necessarily mean that is the same for the acute phase, and that's the only mechanism. I mean, I believe that it might be worth, studying what, knocking down centrin would, lead in order of, species, because clearly, I mean, they're so different, and it might lead to, different types of, it might require a different adaptive, response to protect, but it's not necessarily the type 1 immunity that's responsible for the protection. It's just it's one of the features of the leishmanization.
Yeah, no, that's a good point, and I think some of the data I would actually love to get my hands on the center knockout, because I think there is some divergence about what exactly is the immune response being elicited by those center knockouts versus leishmanization. You know, an argument's been made that there's a predominant role for neutrophils in the protection mediated by those center knockouts, and that's definitely not the case in leishmanization. You know, when you find parasites at a secondary challenge site, just in, you know, the basic B6L major model, it's the neutrophils are where the parasites that are left over are hiding. But I know that here is definitely making a case that with the centrin mediated immunity, that the neutrophils are mediating some killing. So there's a fundamental difference there. So I think still quite a bit of work to do, although the rationale to make the centrin knockout an attenuated parasite is certainly, you know, rooted in the practice of leishmanization. There might be some differences.
Ebert.
I have a question for David. David, great talk. I have two questions, sorry. The first one, when you look and you did see difference on macrophages from cutaneous and from diffuse cutaneous, can we say that disease is associated with adaptive immune response and the macrophages doesn't play the games of T cells? What do you think?
Well, I mean, I think that the way Nathan started out his talk was correct. And that is, the T cells are the major drivers of the specific immune responses, and all the other cells are going to sort of march in line with that. Although macrophages tend to be really promiscuous, they will change their physiology in response to anything in the tissue. They will respond to damps and PAMPs in the tissue. They will respond to complement activation fragments in the tissue, chemokines, et cetera. So I think they're a little bit more promiscuous in that they will respond to whatever they encounter. It just so happens that when you get, a clonal antigen specific T cell response, usually you get a flooding of a certain cytokine, and that's, the cells have no choice but to respond to that.
My second question, it's about the B cells on the lesions. It's very interesting. So when we look on XID mice, we see a difference in Leishmania amazonensis infection. So white types grow and the XID, there is a lower lesion. But when you look at the parasite load, there is no difference. The same phenotype was observed by Professor Sums, but using the GHD knockout mice. So no difference in the parasite. So B cell maybe is involved in the response or regulations. But when we look on these XID mice, we saw in the white type the presence of IL-10 producing B cells that we cannot see in these XID mice. Did you try to look at the B cells, IL-10 producing B cells, or try to associate it using the transcriptase?
Yeah, we, when we first identified these regulatory macrophages, we identified it, I'm embarrassed to say, on the basis of one protein, and that was IL-10, because that's what those macrophages were making more than anything, and it's such a potent immunoregulatory cytokine. We were a little bit disappointed. I guess you're not allowed to be disappointed about your data, but when we compared localized cutaneous leishmaniasis with diffuse cutaneous leishmaniasis and looked in tissue for transcripts, we didn't see any difference in IL-10 production. We were crushed because I have spent 5 years of my life trying to understand IL-10 and how it's regulated. In fact, we didn't see any difference in IL-12 either. But once again, I caution myself that those transcriptomic studies were done on patients at one particular point in their infection, long after they had gotten lesion development, et cetera. So it may be that we have missed certain things. But I continue to think, as I'm sure most people on this panel think, that IL-10 is a major determinant for disease progression in this disease.
Thank you so much, David.
So I think we have some questions, follow-up questions from Leda. I think she wrote this while David was replying about the nitric oxide production. And she mentions, yes, I'm asking about how much TNF do you had in the permissive and non-permissive macrophages? How much, and then she continues, how much TNF the macrophages are, the permissive macrophages are making compared to the ones that are not permissive to parasite growth? And if they respond to interferon gamma because they are, if they don't respond to interferon gamma because they're not making TNF.
That was to Nathan or to me? I hope to Nathan.
Yeah, I think so.
Yeah, I think that's so. the follow up to Nathan's reply.
Yeah, so I need to dig this up later. So send me an email later, and I'm pretty sure we have this data somewhere. So you're looking for the relative contribution or the role of the TNF in all of this, the synergy, et cetera, et cetera. So yeah.
Ebert, you still have the hand raised, or?
I can do a question for Thiago.
Great.
Thiago, congratulations for your talk. So when you think about neutrophils, it's a short lifetime cells, but so how using BALB/c and B6 mice, so did you see difference that can imply in pathogenics? So is that difference in net productions or inflammatory response through or by net comparing these two models?
Thiago, you are mute.
Okay. So thanks for the question, Habit. So there is recent data actually this being published in the blood, showing that in fact neutrophils they've developed better methods to trace down the neutrophil survival in the circulation using radioactive molecules. And what they show is that actually, although it's been thought previously that neutrophils, they survive just a few hours in circulation, in humans, actually, they can survive up to 5 days and in 2 days in mice. So that's not actually, it's being revisited, that information. And so what we saw with regards to pathology and inflammation is that if you digest those nets during the acute phase of the reaction, what you see is that there is a consistent effect in parasite dissemination, but there is a transient effect as well in pathology. There is slightly more lesions in the BALB/c, but there is a drastic decrease in the lesions in the B6 resistant mice by means that they increase the resistance. The mechanism, I cannot explain it to you right now, but I think there might be something related to the fact that there is increased diffusion of parasites to the lymph nodes. But I would need to, dig a little bit deeper into what's important of phagocytosis and how that will affect pathology, what is important, netosis and degranulation, all of the other mechanisms to be able to explain that as well. And of course, there is, during the chronic phase, there is a major role of the adaptive response. So like the interplay between neutrophils and T cells during this stage, it's so critical to understand better this mechanism.
Have you looked, Thiago, the paper that's talk about the physiological neutrophils in the tissues? So, because I think that's something important to see the impact of this. I don't know if there is neutrophils in the skin, in the physiological condition. I saw in the paper livers and the other tissues that there are some neutrophils that is part of the tissues. So did you see or have you looked at these Leishmania infections, the impact of these physiological neutrophils in the tissues?
This is actually a very interesting point. There has been recent data, mostly done by André Hidalgo's group, showing that there is actually subpopulation of neutrophils, because before what was established that neutrophils, they are basically just in the circulation, right, that they come out of the bone marrow, and then as they're dying, they return to the bone marrow. But there is actually multiple pools of neutrophils, and it's not under physiological conditions, under homeostasis. It's not very well understood what is triggering the migration of neutrophils there. And one of those sites, it's the lungs, the spleen. I think there has been some studies in the kidney as well. I'm not so sure about the skin, but I would not be, and the gut, but I would not be surprised, because I mean, the skin is one of the largest, like tissues in the body, and it's constantly, like receiving stimuli from skin microbiota, and the microenvironment that could potentially, like trigger some sort of, like inflammatory reaction that at steady state they would sustain, like neutrophils to come there and patrol. But I mean, if you just do like ordinary flow cytometry of naive skin, you can still detect some level of neutrophils in there. So whether they are actually in the skin or just like part of the circulation, I mean, there, it's not, I mean, I cannot affirm,
Thanks so much.
Thank you.
Herbert, you asked earlier about interferon gamma receptor expression on infected macrophages. I just looked on my list, and it looks like they go down from the transcripts go down between 6 and 10-fold following infection.
That's very nice to know.
Cool. So just one more comment about David's presentation. Was very, very nice, intriguing. And the data related to the reads in the lesions, the diffuse Leishmania amazonensis patients with the diffuse disease, it's amazing how many reads they have of the parasite, which I guess reflects how much parasites you have, how much more parasites you have in the lesions, right? And then if I recall correctly, you compared to the localized lesions of Leishmania brasiliensis infected patients. And of course, it's very hard to obtain samples from patients with localized Leishmania amazonensis. This is very clear. But so my question is, do you think if you compare, like, as these parasites are very different, like, so you think if you had the access to samples from Leishmania amazonensis infected patients with localized lesions, you're going to have so low parasite reads as well, or this would change a little bit the scenario?
Yeah, that's a good question. And by the way, that's the reason why the NIH decided to pass on funding those studies any further. We wanted to do that exact study, but we couldn't get support to do that. I do think Brasiliensis is uniquely immunodominant and immunopathogenic. And I think that's primarily because of the incredible cytotoxic T cell response that one sees in there. And you're going to see a senescence response in there that you're going to hear about from Daniel and the cytotoxicity from Claudia as well. I do think that Brasiliensis is unique in that way. So we were really sort of contrasting two, very, very different polarized responses here. An immunopathogenic response associated with Brasiliensis, and really a defective immune response associated with amazonensis. So it was a little bit of an unfair comparison, except that it really makes the differences very, very clear. Those are two very different diseases, and they have very different outcomes with regard to parasite numbers and growth.
Cool. And there is also a question from Patricia Veras. She asked to you, David, if that would be interesting checking if the RNA-seq of early lesions of DCL patients to further associate LGs to macrophage response in Leishmania amazonensis infection. Do you have access to those lesions?
Yeah. No, I mean, those patients are as scarce as hen's teeth. You know, they're really hard to come by. We were very fortunate to have developed that collaboration, and he was very generous to invite me up there to meet his patients, to take the biopsies and fast freeze them and bring them home. We, you know, and all we had was 6 patients. That's all Fernando has. So that would be great to do, but we don't have access to those patients.
Great. So I think we are going to the end of this roundtable. I'm going to open if anyone wants to comment on something else or discuss some specific point.
I would like to thank you all. It was amazing. And we know it's a hard day, that's all day long in conference, but for me, to hear about leishmaniasis and immunology was fantastic. Thanks so much all for participation.
Thank you.
All right, thank you. It's awesome to stay with you guys this afternoon.
Great to see you, Dario.
Thank you. Bye.
Thank you, guys. Thanks so much. Take care.
See you tomorrow, guys.
See you tomorrow.